and welcome to this Genomics England Innovation Showcase. In this session, we're going to be talking about the genomics of COVID-19, both the virus and the genomics of the human genome, to help understand how some of the work we've been doing at Genomics England together with people across the UK life sciences sector have helped us to understand some of the puzzles around COVID-19. I'm Rich Scott, I'm the Chief Medical Officer at Genomics England, and I'm joined with me in this session by two people. Firstly, by Professor Sharon Peacock, who is a Professor of Public Health and of Microbiology at the University of Cambridge, and in her role at Public Health England has established the COVID-19 Genomics Consortium, COG UK, which has been the driving force behind establishing the UK's potential to sequence the COVID virus itself. I'm also joined by Dr. Kenny Bailey, who is a research fellow and a consultant in anaesthesia and in critical care at the Roslin Institute and at the Royal Infirmary in Edinburgh. And as I say, we're going to talk about the work that we at Genomics England have been doing with Kenny, with Sharon, with many others across the UK life sciences sector, looking at COVID-19 and the genomic determinants of disease. We're going to be talking about that from two perspectives. Firstly, the genomics of the virus itself. So the COVID-19 virus has an RNA genome. That's the nucleic acid code that its genome is written in, and it's about 30,000 DNA or RNA bases long. Sequencing that, as we know, has helped us understand the biology of COVID. It's been really important in designing vaccines, and also, as we've all become intimately aware over the last 18 months, it's helped to understand where new variants of the disease have emerged and to track them. We're also going to be talking about the sequencing of the human genome and how that has helped us to understand why some people get severe COVID and some people get more mild disease. So reading, in that case, the 3 billion letters of the human DNA code in people who've had COVID. And in the case of the work that we've been doing here in the UK, comparing the genomes of as many as 20,000 people with severe COVID in hospital, in intensive care with COVID-19, with 15,000 people who've had COVID more mildly, and comparing those and understanding what drives those differences in terms of those participants' genomes. And that can help understand the biology. As you'll hear from Kenny, it's also been useful in making real decisions about working out which medicines might work better to prevent severe disease. So we're going to be hearing about that. We're also, I think, going to be hearing about how crucial collaboration across UK life sciences sector has been in this, and particularly at the beginning of the pandemic, how fast decisions and collaboration really set us on the front foot in the UK in terms of genomics. I think we'll also hear about the importance of the historical strengths of the UK in terms of genomics in putting us on that front foot. And that's not just the early discoveries that have been made in the UK in terms of genomics from Franklin, Watson and Crick, creation of new sequencing technologies, but it's also more recent investments and ongoing investment that keep us at the head of the game in terms of genomics worldwide. So, for example, us at Genomics England, having benefited recently from the sorts of investment that allowed us to build the infrastructure to deliver the 100,000 Genomes project, also to have that infrastructure to support the NHS Genomic Medicine Service whole genome sequencing service, and how we can use that infrastructure that was there and invested in to quickly adapt it to respond to the COVID pandemic. We'll also, I think, reflect on what the lessons we've learned are and what that means in terms of making sure we're prepared for future pandemics and remain ahead of the game in terms of genomics in this pandemic. So what we thought we would start with is going to Sharon. Sharon, I wonder if you'd like to sort of introduce yourself and then perhaps start by reflecting on and telling the audience some of the stories about the beginnings of the genomics response to COVID-19.
It's absolutely wonderful to be here, so thank you very much for the invitation, and I'm really pleased to be able to talk about SARS-CoV-2 sequencing in the United Kingdom, actually more widely, because it has been so important for our pandemic response. And so I'm a professor of public health and microbiology at the University of Cambridge, and I also direct the COVID-19 Genomics UK Consortium, which is conveniently kind of abbreviated to COG UK. And to date, that consortium has actually sequenced more than 1 million viral genomes, which is really quite an unprecedented number of genomes, which has helped to inform decisions, as you mentioned. But where did it all start? Well, it really started with an early conversation with Sir Patrick Vallance, who's our government chief science advisor, a very wise man. And we were saying that, really genomics was going to be key to understanding the evolution of the virus. Evolution is something that happens quite naturally. And we knew that after the virus emerged, that it would start to change. And we needed to be able to capture those changes so that we could understand the virus and its biological characteristics. Was it going to be more transmissible, or was it going to be more lethal, for example? So we needed that understanding. And so I've got a very nice image here of the initial, let me see if I can share my slides. So the early development, you can see a sort of pictorial representation here by a fantastic artist who in the bottom left-hand corner, following conversation with Patrick Vallance. Can you see those okay? Yep. You see the slides? Yeah, okay. You can see in the bottom left-hand corner that we called together mid-March 2020, actually, when there were fewer than 80 diagnosed cases in the country. We called together a group of scientists from across the United Kingdom who were experts in pathogen sequencing. And 20 people met in a room at the Wellcome building in Euston Road and actually discussed how we would put together a sequencing network. This was unprecedented times. We pulled together everyone who we thought could contribute to this. So there we are in the bottom corner. We put together over the course of a day a blueprint. So often when scientists get together, they'll talk about the nitty-gritty of science, but we talked about how could we practically put together a sequencing network for the United Kingdom. So within a few days, by the 18th of March, we had developed a blueprint which we passed to Patrick Vallance. And we were very fortunate to get supported by government funding through the COVID-19 Fighting Fund. And, the rest, as they say, is history. You can see in the image that we connected with the Lighthouse labs and other testing labs to get samples for sequencing. And we did the sequencing, which goes into a database, and then we use the genomes to understand how the virus is evolving. So this is what we developed. So if you look at the map, hopefully there's a sequencing facility near you, and you'll see that we have 16 regional sequencing sites across the UK, and they're in actually universities and the 4 public health agencies of the United Kingdom. And the Wellcome Sanger Institute were key for this too. They're a major sequencing site, and the project was administered in Cambridge. Now, you've mentioned that this is really important. Why do we need so many genomes? Well, actually, going back to the very first genome, in February 2020, this was a genome that caused an infection in a patient, or a virus caused an infection in a patient in China, and was released on the 3rd of February. And that was actually really instrumental for the very first vaccine design. So from that sequence data, you can tell a lot about the virus. You can tell what sort of virus it is, and it's kind of what sort of proteins it produces. And it was really important for vaccine development right from the very beginning. And we've already mentioned the fact that going ahead, this is really important for monitoring for vaccines, and that is very much the case into the future for as long as the virus is circulating. So I've got other slides that I can show in terms of how we've tracked the virus, but I think what I'll do is I'll stop there and perhaps come back to my slides about how we track the virus a little bit later.
Thank you, Sharon. I'm really interested by to know a little bit about sort of maybe what some of the history was that helped you when you entered that room and had those discussions and came up with that blueprint. What were some of the things that either had been done before or had been thought about before that meant you were able to make such good and rapid decisions?
Yeah. Well, I think you have to go back to 2013, actually, when there was an announcement about the 100,000 Genome Project by David Cameron. And that really raised awareness and started to build the infrastructure for sequencing. And actually, that was focusing on a combination of rare diseases and cancer. But a third work stream was actually pathogen genomics. I think at the time it was less well talked about, but that was focusing on pathogen genomics. And I actually chaired that group. And we made a set of recommendations about how we should start to use sequencing technologies for pathogens. And so we made recommendations that tuberculosis, the cause of tuberculosis should be routinely sequenced. It gives you really rapid information about resistance and also outbreaks. We also indicated that HIV sequencing, deep sequencing, that's having really detailed information about emerging resistance to drugs, going to be important. And also sequencing of hepatitis C also for treatment. So, right back in 2013, we were putting in place mechanisms to sequence pathogens. And I think following on from that, there was an awareness in certainly politicians' minds, policymakers, and that kind of drive also developed our capabilities. I think key moments were when Dame Sally Davis, our previous Chief Medical Officer, wrote her annual report, volume 2, and in 2 of those, she had one on genomics, which was really important for us in pathogens because we could highlight what we did. But we also got involved in the antibiotic resistance volume 2. And I think since 2013, and even before that, actually, we've been building our capabilities in the country so that by the time the pandemic came along, we had a really good group of experts across the country. We had the equipment, we had the expertise. We had to refine our methods for SARS-CoV-2, but it was like we were sort of in the starting blocks by the time SARS-CoV-2 came along, and were ready because of that history, actually, to get started with sequencing the virus. And by the time we got funding from the government, we'd already sequenced something like 250 genomes.
Fascinating, thank you. And I guess coming to you, Kenny, perhaps you sort of to introduce yourself and give a little bit of background both to the work that that sort of preceded COVID-19 from your point of view in terms of understanding determinants of people needing critical care for various reasons, and how again that set you and us as a country in a really strong position to respond rapidly to COVID-19 when it did arrive.
Well, thanks, Richard. So I agree completely with Sharon. I think that the key message, the key learning point from this outbreak is that what worked was what was already working. So we built on the foundations of existing activity, and without activity, infrastructure deteriorates. For the host genome sequencing, we already had a project operational in about half of the intensive care units in the UK, and it was ramping up. We started it in 2015, and the objective was to use host genetics to understand the complex biological problems that we face in intensive care medicine, including sepsis, flu, and emerging infections like SARS. So we were ready. Of course, when the outbreak hit, as with in every other sector, we had to adapt quickly and scale up but the infrastructure, the protocols, the approvals, and the people were already there and active. And that's really why we were able to scale up so much more quickly than any other country. So many other countries put resource into host genetics during COVID, but none have created as much genetic discovery power as the UK. In fact, the UK study that we've done in collaboration with Genomics England has more genetic discovery power than every other study of the host genome in the world put together. And that's fundamentally because we were able to get the right patients in very large numbers. I do have a few slides that I can show you about those results, if you like, Richard, and then go back to the discussion. So I can show you perhaps what we found. I think that would be of most interest. We were faced with the problem at the very start of the outbreak that we didn't know how to treat COVID. Before it was even called COVID, we'd had extensive discussions of what treatments might be useful, and I had the privilege of contributing to the setup of the recovery trial, which I think most in the audience will have heard of, led by Peter Horby and Martin Landry at the University of Oxford. When we first started choosing treatments to put into recovery, we had very little evidence for which treatments to use. We knew that the host immune system, your own immune system, is likely to be damaging the lungs. We now know that to be true, but we weren't sure that was the case. We weren't sure that immunosuppressive drugs would help people. And an even bigger problem is that the immune system is incredibly complicated and we don't know which buttons in it to push, which targets to aim for with drugs. And that's really where host genetics, I think, has most to offer. As most people will know, 6 of the drugs that we've tried so far in recovery have not been shown to confer a benefit on patients. And by a combination of good judgment and good fortune, 3 of them have, and together these have more than halved the mortality in critical COVID. 2 of the treatments that are currently being randomized right now, that we're currently testing in recovery, have been added to recovery, in part at least because of genetic evidence that we've provided from the genomic study in collaboration with Genomics England. And I'll show you the results just very briefly in 2 slides of genomics. So this is the result we got within 5 months of the first case hitting the UK. We had recruited 2,200 critically ill people, and that's important. The most extremely sick people with COVID, we recruited into the study and compared to the rest of the population. And this, I think I probably don't need to describe a Manhattan plot for this audience, but just for anyone who's not familiar with these plots, this is every chromosome in the genome lined up along the x-axis, and there are 5 million spots on this graph. The y-axis shows how sure we are that a given variant, a given genetic difference is really different between critically ill COVID patients and the rest of the population. And where it crosses this red line is where we agree that it's probably real. 4 of these, so you can see there are multiple hits and multiple points where it crosses the red line. We also set ourselves a second challenge, which is that we have to replicate them in a second group of patients. And 4 of these associations were robustly replicated, and we're very confident that they are real. And all 4 of them lead to therapeutic hypotheses, to ideas for treatment. Probably the most directly related to treatment is this gene type 2, which is one of the targets of baricitinib, a drug currently being randomized in the recovery trial. And actually, since we produced this evidence, that several clinical trials have been reported suggesting a benefit. And perhaps just as a teaser, this is what we found most recently. So that first slide had 2,200 patients included, and we'd used microarray genotyping, a cheap but fast technology to read the genomes. This is the results from genome sequencing in 7,400 people. So genome sequencing done by Illumina under the guidance of Genomics England. And this really shows the power of firstly getting large numbers of the right patients into the study, and secondly, the resolution that you get from genome sequencing. There are 25 spikes crossing the red line on this graph, and all of them are indicating important parts of the biology of the disease. this paper is under review right now, but it's in the public domain. People can see it on medRxiv. I'll stop there, and perhaps we can speak about some of the other aspects. I've got a few other slides I can show if needed.
Thank you, Kenny. So I think I was really struck at the beginning of the programme about how integrated the thinking was, in the same way that Sharon talks about some of the work that had gone together, and how those people together planned out a blueprint for the genomic response, particularly in the virus. The close working of recovery of your study, Genomic, of other studies, for example, ISRIC4C, the study you lead, looking particularly at clinical data of people with COVID. We've also been working with a number of others across the country. So, for example, in the work we do with you, Kenny, we're also working with the REACT program, so led by Paul Elliott at Imperial College, who are the team who people will have heard on the news, just as the ONS also do. They send teams to households across the country periodically, so that we can keep a track of the prevalence, the frequency of COVID in the population. We've worked with them to approach their participants to ask them if they wanted to join the same program, and so that we can then, alongside that network of recruitment that your team had already established ahead of the pandemic across intensive care units, across the NHS, and with so much input from the NHS itself, also recruit participants to the program from home who have had more mild disease, and in some cases long COVID, so that we can both compare their genomes, severe and mild, but also begin to understand potentially more about the biology of long COVID.
Yeah, I think the collegiate interactions between different studies has been one of the defining features of the UK response to the outbreak, and really why the UK has led the world in both discovery science and therapeutics in COVID. The other studies that we absolutely depended on were UK Biobank, and I've just realised that my little video is over the UK Biobank logo on our slides, but of course they provided the control group for our first genetic study, and that was already there. That's infrastructure that already existed and was generously shared. And of course the 100,000 Genomes Project, which Genomics England created, which provided the controls for the data that we have so far. For those 25 extra hits, I think can look forward to much more discovery. We now have 15,000 patients in the genomic study, and with REACT and Paul Elliott and the Genomics England team have recruited 15,000 individuals who are well matched to the critically ill people, both in age and sex and genetic ancestry, and had relatively mild or asymptomatic disease. So I think the discovery, the power of the study will more than double in the next few months when we analyse that data.
I wonder, it'd be interesting, in fact, later in this collection of showcase sessions we have today, we have a focus on diversity of data sets. You mentioned there, Kenny, the importance of the diversity of the COVID participants, and being able to make sure that those discoveries are relevant to everyone. I wonder whether you can sort of, again, give us a bit of a flavour of some of the challenges, but also the work that you have been doing with the team in the more recent work with the whole genomes.
Yeah, thanks. So I'll show this slide again because it contains a few hits that are exclusively discovered because we had both whole genome sequence data and a more diverse population than in the first study. Now, in genetics, everything, the discovery power is determined primarily by the number of patients, the number of the right patients, with the right clinical characteristics that you get into a study. And it's difficult to draw comparisons between a control group from one genetic ancestry and by that I mean a genetic heritage and a case group from a different genetic ancestry. So we're bound to compare the groups of people that we have the most of. And in the first study, that was white Europeans. Now, over the last 12 months, since we reported the results from those first 2,000 cases, we have several hundred patients from several other genetic ancestry groups that are common in the UK, and through the 100,000 Genomes Project and the direct recruitment through the Genomics England programme and REACT, we have people to compare them to. And that means that we're able to see signals, for example, if I zoom in here in chromosomes 1 and 2, TRIM46 and BCL11A, these are genetic signals near genes that have these names, probably mediated by these genes, that have been revealed because we're able to compare between ancestry groups. And particularly here, you can see that this is all one spike. It's difficult to tease apart genetic signals that are next door to each other on the genome. And the reason is that a whole block of genetic variants are inherited together, but those inheritance patterns are different in different ancestry groups. So having different ancestry groups helps us to tease apart signals that are right next door to each other in the genome. And of course, the other thing that you need to do that is really high resolution data on the genome, so the whole genome sequence has made that possible. So that's, it's extra discovery that we couldn't really get any other way.
Thank you. And I get it goes without saying, but crucial for both programs has been rapid funding decisions and sort of enthusiastic funding from multiple sources. So, for example, for the program we've been doing in human genomics of COVID, funding from the Department of Health and Social Care, from UKRI, which predated for the study and then supplemented that with specifics funding for the study, from NIHR, from LifeArc, and also contributions from Illumina have been crucial for us, for example, not just to fund the sequencing of the genomes, but also from a Genomics England perspective, one of the important things that we were able to do was establish a new cutting-edge cloud-based research environment that meant that the data were not just generated, but generated and made available with the right tools to support these rapid discoveries, so that the genome data were generated, the consent process that participants went through meant that they were agreeing to join our National Genomics Research Library. So their genomes sit alongside the genomes of the 100,000 Genomes participants, and participants or people who agree to a research consent offer through the Genomic Medicine Service and other research studies, so their data can be made in a de-identified way available in a trusted research environment that approved researchers can then visit to make these discoveries. And it's that support, and not just the infrastructure to be able to do the sequencing, but also to have the right tech and tools available so that we can get the right minds and algorithms there to make those discoveries. Sharon, I wonder whether it would be a nice time to go back to you to talk about the discoveries and the work in the viral sequencing program.
Certainly, yes. So perhaps I'll just share my screen again and show a couple of slides just as illustrations. So, what has sequencing done for us? I think at the very top of the list, we need sequencing for, I've already mentioned vaccines and therapeutics, but just to sort of dig into that just a little bit in a bit more detail. It's kind of, when you're looking at viral evolution, then it's survival of the fittest, and you have a fit virus emerging that can outcompete other viruses. It's a sort of simple philosophy that the fittest virus would be the one that can transmit the most, and or the one that can get around our immune system, dodge our immune system. And so looking at the vaccines that we have at the moment, their original development was guided by the first genome that was released. And at the moment, they're highly effective, actually. But looking at future viral evolution, what we need to be clear about is that our vaccines remain effective in the longer term. So it could be that we need to tweak our vaccines to make them as effective as they are today. And already vaccine developers have developed vaccines against specific what's called variants of concern. There are 4 circulating in the world, and these include variants that kind of partially dodge the immune system. But the other thing that we need to do is to watch for the virus evolving resistance to other therapeutics. Now, Kenny's already mentioned some therapeutics, but what's called monoclonal antibodies, these are antibodies that have been raised in laboratories, but that can help in fight infection. They're given in cocktails, but actually, and to try to help people who haven't got a strong immune system to fight against the virus and against the infection. But actually, what the virus will do over time is actually evolve to try and escape the effects of those antibodies. And so I think top of my list for sequencing now and in the future will be keeping vaccines and other therapies working effectively. And that includes actually antiviral drugs. So that's really important. Now, the second area that sequencing is really important for is it really does help us to understand the wider characteristics of the virus. I've already mentioned managing to dodge our immune system, which could lead to a risk of reinfection or infection after you've been vaccinated. But actually, if we can group the viruses into variants of concern, then what we can do is then study those, work with our biology colleagues to study them to understand whether they are more transmissible, for example. And so we detected the alpha variant first emerged in Kent, and we know that that's more transmissible than the original virus. And the second virus that's been problematic in the UK is Delta variant, which is more transmissible than the alpha variant. And so we can understand transmissibility, but also we can look very carefully at the variant versus disease severity. So we know that 2 variants of concern increase the risk of going to hospital, but not the risk of dying from that infection. We also need to check that our diagnostics are going to keep working. So as the virus genetic code changes, that can sometimes change the way our diagnostics work. Now, it's been fortunate that actually none of our diagnostics have been rendered not effective anymore. But there have been some areas where the diagnostics perform differently, and actually we've used that to good effect, particularly with the alpha variant. We were able to track the alpha variant through the PCR result. So we also use these to track variants coming into the UK and spreading in the UK. So what this wonderful graph shows is really the pandemic since the very beginning, from March 2020 right the way through up to the end of July. And each colour represents one particular version of the virus, or we call variant or lineage. So every colour is a unique lineage. And it's normalised to 100%. So at any given point, it's a proportion of what's causing infection in people in the United Kingdom at that time. Now, if you look at the kind of white blocks, what you'll see is I've put on there superimposed wave 1, wave 2, and wave 3. So what we can see here is that in wave 1, there was a huge diversity in the virus. There were a large number of lineages, and not one was kind of particularly dominating. This was before the variants of concern emerged. Then we go into wave 2, and we can see there's actually at half of wave 2, we've got lots of diversity. We have that red blob in the middle, a lineage called B1177, and that increase in the UK, it kind of got up to about 50% of all of our viruses are causing disease in people here, was most likely due to European holidays, travel. So it first emerged in Spain, and this lineage spread around Europe. Now, fortunately, it didn't have any special characteristics other than it was traveling in people who were traveling. And then you can see the alpha variant emerge, and you can see how it becomes completely dominant in the country for actually a period of months. And it's only kind of pushed off at the top spot by the emergence of Delta variant, which was introduced by travellers and then rapidly spread around the country. Now, this is really important for planning. So we need to know on a population level what variant is circulating, so we can then match our expectations for whether disease may be more severe or whether it's more transmissible, and there may have to be a titration of measures to try and stop the transmission, which we know is all to do with hands, face mask, space between people, and kind of being outdoors, but also vaccination. So this is how we can track the variant, and I think that this image is kind of really elegant description of the variants that have circulated, which have a direct impact on decisions around policy, etc. I think that that's my last slide, so I will stop there and hand back.
Thank you, Sharon. And I guess one of the things we've got a feeling for through talking about the human genomic work is some of the interactions with some of the other studies that are going on. I wonder if you can tell us a bit about how that data is being used and accessed, sort of both by the sort of core teams, but also how it's interacting with some of the other data sets that are available.
Yeah, certainly. Well, I would start by saying that as a community, there's been an immense support for data being released immediately. So all of our viral genomes are released into GISAID, which is a global database. So what we have, we share immediately. But you're right, they have used the viral data in numerous different studies. And so we've been able to, for example, sequence the virus in vaccine trials to see if people get infected with SARS-CoV-2 after they've been vaccinated, what variant is that? You know, is there kind of an emergence of a variant that's actually escaping that vaccine? We've worked with people who have been investigating, for example, outbreaks in prisons and schools and in hospitals. We've also sequenced for the ONS study, which you've already mentioned, and REACT, so that we can tell at any given moment what is circulating in the country. And we've provided sequencing for studies in care homes and so on. So we have shared our data extensively. We share it with biologists because they need to understand how a given variant actually behaves, you know, in laboratory experiments. And we share this with immunologists. Anybody who wants this data can have access to it. But in looking to the future, I think sharing of these data sets with data sets such as the human genome, and I'm sure we're going to talk more about this, actually what that's going to do is going to lead to really novel analyses where, you know, there hasn't ever been, to my knowledge at least, you know, 1 million pathogen genomes combined with a very large number of human genomes. And the possibility for discovery moving into predictive medicine, I think, is immensely powerful. So we're going to we've done an initial analysis of the virus, what variants are there, what impact does that have on control measures and policy decisions and vaccine development. But I think we're moving into a new era now of where bringing these data sets together means we can actually go into discovery science and really understand the interactions between the virus and the host in much greater detail than we've done for any other disease.
And for us, one of the really key things about the consent, what the participants are agreeing to in the human genome study is that ability to link data, because that adds, as you say, so much power to what can be understood. And for example, our teams working with the COG UK teams, working through just the practicalities of that linkage to make it work, so that we can, as you say, combine viral genome data sets and alongside those human data sets. And often it struck me through this, it's the unsexy stuff of making multiple studies work together and link well, and just the conversations that actually add so much value, so that what's delivered from the broader UK life sciences sector on this is greater than the sum of its parts, and it's that multiplicative power through the linkage. I wonder, I know, Sharon, you've been quite struck by the human side of that in the interactions, and I wonder whether that's something that you could expand on.
Yeah, well, actually the initial interactions with Genomics England happened from day one, and so Sir Mark Caulfield was at our brainstorming event actually back in March 2020, and ever since that time, COG UK have worked really closely with Genomics England, with ISARIC and others, and we've built those teams, and those teams have been there from the very beginning. And so working together, I think you're absolutely right. It's the kind of the practicalities of how do we link data? Where is it going to be housed? What's the process for accessing that data? It's that that kind of really underpins our discovery science, which is going to be so important in the future. But actually, that is also underpinned with regular interactions, good relations between people, and I think that the idea that we would bring viral and human genomes together, at the very beginning, March 2020, means that we've got plenty of time to plan and develop those kind of connections between us. So I think that this is kind of outside of my usual experience, actually, and I think it's a credit to scientists in the UK that they've been so keen to work across disciplines rather than stay in their own disciplines. And I think that's going to make a big difference for discovery science in the UK.
And Kenny, I wonder whether, I know you personally, but more broadly, the human genomics work has linked in with multiple other studies looking at different facets of the biology of COVID response. I wonder whether you can sort of share some of what's been going on and your reflections.
Yeah, I agree completely. What has really driven the innovation in the UK during COVID has been the boring but important stuff that we have been paying attention to for some time. So you mentioned the ISARIC study that builds on a consensus protocol for studying new diseases that we developed back in 2013 in the wake of the swine flu pandemic. The ISARIC consortium was formed, initially led by Jeremy Farrar, the head of the Wellcome Trust now. I chaired a working group that was tasked with coming up with an agreement on the data and sampling strategies that we should take for any new outbreak. And in fact, that was endorsed by WHO and established in multiple countries around the world. And that protocol was used for the very first description of the clinical features of COVID in January 2020 in Wuhan. In the UK, we established the protocol together with Callum Semple in 253 hospitals across the whole country, and it was there ready as a sleeping protocol, initially supported by Sally Davis when she was CMO, as the first urgent public health study. So when the first case arrived in the UK, we were able to recruit them into the ISARIC study, and since then we've recruited many patients. So we've obtained samples from thousands and data from 239,000 patients now. That data set is of great value for both public health, government planning, and research, and has led to many papers that I think many in the audience might be familiar with. But I think the real power of it is the ability to link to other studies, exactly as you're saying. So we've already linked the overlapping data from the COG UK viral sequencing study to provide additional prospectively collected clinical data about the hospitalized patients for whom we have viral sequence data. And of course, the host genome data we're now linking to viral sequence data. I think the boring but important element of that is that it is the human stuff that makes that work. It's we have extraordinary computational facilities. The UKRI funded outbreak data analysis platform here in Edinburgh sits at the UK National Supercomputer Facility, where there's about £100 million worth of hardware. But really what makes it work is humans linking the data sets fighting through the mismatching numbers, the missing data components, and the quirks that there are in the data, and building a data resource that is both comprehensive, which it already is, and usable, and that's an ongoing program of work supported by HDR UK. And actually, if there are people in the audience who are interested in using that data set, then they can apply for support from UKRI, HDR UK, and the Turing Institute through an open call right now. The data set comprises now 750,000 viral genomes, 240,000 clinical data sets, the genetic data from thousands of critically ill humans, and a range of other studies that have been generously contributed to the platform. That's a resource that's going to continue to become more useful as more people use it, because it's using it and working on it that makes it more accessible.
And on the theme of people power, one of the things that I've been really struck by and enjoyed during this process is talking to our participants in the National Genomics Research Library, who are at the moment predominantly people who joined through the 100,000 Genomes programme. We have, as I think you both know, a participant panel who are involved in our governance across Genomics England, including oversight of who has access to the data, but also the other decisions we make. And one of the things they've been both really eager to hear about, but also delighted and excited by, is the fact that, and you mentioned this, Kenny, is that their genomes that they agreed to be made available in this National Genomics Research Library could be powering some of these early discoveries. So they're people who joined our programme for other reasons. They may have had themselves a rare disease or a family member with, or they had cancer, but they want their genomes, they want their data made available in this secure way to drive exactly this. And it's that people power and the collaboration with others, you mentioned Biobank, etc. And it's the humans driving the really positive collaboration that we've seen, but also the people who are participants in these programs powering and actually really pushing us to make sure that the data's available and used to make really important insights.
Yeah, I think the interaction with patients particularly has really driven this work. One of the bits of advice that I got quite early in my career was from Steve Webb, an intensivist in Australia, who said, do the research that benefits your patients most, and all the papers and grants will follow from that. And there's nothing like being funded by patients to really force you to do that. And actually, I mean, you listed financially the largest contributors to the Genomic project when you were talking about funders earlier. And of course, they've made it possible. But the reason Genomic existed at all is because a small charity called the Fiona Elizabeth Agnew Trust, Sepsis Research, which is run by bereaved relatives of intensive care patients, gave us £120,000 to establish the study over 3 years. And they were very flexible about how it was used, and they didn't require outputs within the term of those 3 years. We told them it might take more than a decade to have impact. And it was that flexible funding that just trusting the investigators to take a risk and establish something that might take a long time to yield, that really made it possible to deliver Genomic. And of course, the source of the funding being from relatives of people we'd cared for in critical care really focuses everyone's mind on delivering something with real clinical impact.
And that actually moves really nicely to the sort of last area that I thought it'd be nice to talk about before we move on to the Q&A, which is about what some of the lessons are for the future. Sharon, I think you talked a little bit about some of the sort of historical legacies we've had that put us in a really strong position. Tell us, share some of your thoughts on on what this tells us for the future, and so we can make sure that we can respond rapidly to future challenges like this.
Yeah, thank you. Just before I do that, just going back to your previous point about the importance of people, I would say that something that we're very aware of in COG UK is that 1 million genomes is a huge scientific achievement, but each of those genomes actually relate to a patient who's had COVID-19. And we're always very cognizant of the fact that we need to think about the people who've been affected by that. So we have very much the same philosophy that Kenny's talked about, the importance of doing the right research for people, and we're grateful to have access to those genomes. Lessons learnt. Well, if there is one thing that I would say I would change about the pandemic if I could, and what I hope we will do well next time, is when a kind of unusual infectious disease emerges that could be the next pandemic. The lesson for me is that we need to study that very intensively. So we need to have mechanisms in place for clinicians to recognize that there's a kind of respiratory infection in somebody, but you don't know the cause. Could this be the next pandemic? And have the ability to sample that person to really understand what the cause is. And sequencing is only one part of that, but it's an important part because you need to understand very rapidly whether there's a new virus on the block that's causing an outbreak. Once you get that information, you can close that down really quickly and it doesn't become a global pandemic. So the very biggest lesson learned for me is that having the capability to diagnose people with sort of unusual or undiagnosed diseases is very important. And Kenny and myself and others have talked quite extensively about how you'd have that framework. Now, Kenny's framework does exactly that, but we need to actually have the ability to, as I say, sequence the pathogen. And if you know what it is, you can contain it very quickly. I think other lessons learned, the importance of team science has been really important. So scientists will often work in their own areas of speciality. I think working across disciplines, human genetics, viral genetics, studying the virus itself, virology, studying the immunology, that has really changed the way we think about disease. So I think a lesson learned for me is never just stick to your own tribe, but also look outside and look at what other people are thinking and saying. I think there are other areas. I think in terms of scale, I don't think that we will ever want to go back to very small scale sequencing because we've seen how useful it is. And so a lesson learned for me is we can use pathogen sequencing to great effect. And we need to now be nimble in thinking about how we're going to apply that going forward in relation to areas such as antimicrobial resistance control or the next pandemic. I think finally, I would say that I have become so much more aware of the importance of data linkage. And so as a pathogen genomicist or geneticist, I don't think that previously I appreciated the importance of data linkage so that you can link your genomes with your patient and with other data sets. And so for me, making sure that looking ahead, that the country is in the best possible position it can be in preparation for the next pandemic in terms of moving data across the country from patients into important data sets and then linking those together to make rapid kind of discoveries or impactful and actionable information that can help us contain and control the diseases. And I could talk. I could probably talk for a day actually on lessons learned, because I have to say that you have to have a sense of humble optimism as a scientist. I think humility because you can't predict what's going to happen next. For example, I can't predict what's going to happen with the virus next. But optimism because actually the science that we've developed, including the vaccines and the treatments, have been quite extraordinary in terms of their pace and scale. So I'm hopefully a humble optimist as we look forward, and that's also a lesson learned for me, that I can't know everything. That's okay, but I learn every day.
Thank you. Kenny, I wondered, sort of similar, your reflections on what the lessons are for the future and preparedness for the unknown.
Well, I agree completely with everything Sharon said, and I can't say any of that better than she did. So I'll only add that I think a lesson for me has been that there is a colossal amount of potential resource available in the academic sector. And when things really got difficult at the end of March 2020, the entire biomedical research sector in the UK certainly was able to refocus on a new problem, and that brought tremendous intellectual and practical resources to a problem, and it is one reason why we've made so much progress. I mean, we now know more about COVID than we do about any of the other diseases that I treat year in, year out in intensive care. So that adaptability is key, and what we already knew is that the outbreak you're preparing for is never the one that hits. So we were preparing for flu, and we got MERS, and we were busy with MERS, and then Ebola came along, and then Zika was the next big threat that came along. Completely different patient group and set of challenges, and then the one that we were all in the back of everyone's head since 2002 hit us in 2019. So I think adaptability is absolutely critical. Designing incentive structures for scientists to work together is a challenge that many people are engaging with, and that does need attention and leadership as it's getting from funders and publishers. The other thing that I think is maybe fits with my biases and I think should be a big lesson from this outbreak is the need to engage with clinicians. So Sharon mentioned that we need to empower clinicians to study what's wrong with their patients when they have unusual diseases. I think that has been a failing of research in many parts of the world, less so in the UK, but creating capacity for clinicians to engage meaningfully with scientists and study the diseases they're faced with, I think is deterministic for success. So critical care is the canary in the coal mine. It's the first place where all of the outbreaks that I listed before were detected. Unusual groups of patients presenting to intensive care, and it's I think through clinical insight that really we've been able to deliver so much in the host genetic program by focusing on patients with the right clinical characteristics to optimize genetic power. We're able to make more discoveries with 7,000 patients than other groups have made with 30,000 patients in other studies around the world.
And I guess one of the reflections I have on the discussion we've been having is actually how new a lot of the technology we've been using here is. What's really nice about the way we talk about it is we take it for granted that we have these next generation sequencing technologies that we wouldn't have dreamt of having up our sleeves had the pandemic had happened even a decade earlier. We would have been in a very different place. And I guess recognizing both that the investment to mean that we have the infrastructure there to respond rapidly, to sequence 1 million pathogen genomes, to sequence at a drop of a hat tens of thousands of people's genomes who have COVID, it has been absolutely crucial. And also that science, thankfully, is moving fast, and being able to use the very latest rapidly is really important. So that sort of remaining at the forefront in terms of genomics and other aspects of science and technology that are relevant to the pandemic also sort of feels like something that we in the UK are fortunate to, I think, sometimes take for granted, and that ongoing investment really feels crucial.
Yeah, I think that's absolutely right. That's such an important point. We do take it for granted, but if I look at the cost and the pace of sequencing, if we go back to 2001, 20 years ago, it would take 18 months and £500,000 to sequence one E. coli genome. That's a bacterium that lives in our gut. And now you can sequence, for a very low cost, very rapidly. So you can sequence a genome in under a day, and perhaps £100, or actually often less than that, depending on how many you sequence at the same time. And so we have to be, we have to be mindful of the fact that has been a revolution in our lifetime. But what really excites me looking forward is if we've managed to achieve that much in 10 or 20 years, what's to come? And so I'm really excited by the prospect of new technology that allows us to do new things faster. I hope that the trajectory of discoveries and improvement is as rapid as it has been in the last 10 years, actually. I'd just like to come back to one point, though, which I'll be annoyed with myself if I don't make. Going back to lessons learned, one of the things that has been really obvious to me is that public health agencies and academia, universities working together has really made a big difference. We need to try and conserve that model, because no public health agency can have all the resources it needs in times of absolute crisis like we've seen in the last year and a half. Universities and academics can make a really big contribution to that. And for example, the Wellcome Sanger Institute, it has done a large amount of sequencing. So I think another lesson learned is really being flexible. Sorry to squeeze that in, but I was sitting here thinking, I really must make that point, because I think for our future preparedness, that model is going to be key.
Thank you. And a great one to end on. We've now got some time to answer some of the questions that have come in from the audience. So we've got sort of 15 or so minutes for the Q&A session before we close. So perhaps to start with a couple of questions on sort of preparedness and the UK response. But here, Courtney Jones is asking the same question, but really from a sort of a global perspective. What is it globally that we need? And perhaps linked to that, Sue Martin asks about, just for a feel for what international cooperation has been like, what it's felt like. I don't know whether perhaps we'll start with you, Sharon.
Okay, thank you. Courtney, looking at the global viral sequencing, what I would say is that it's actually critical for us to have viral sequencing across the world, because actually in areas, particularly in areas where they have low access to vaccines and high disease rates, this is the sort of environment where variants of concern could emerge, because there's plenty of opportunity to infect people when the virus actually makes a mistake in its genome and develops mutations. It's only when it's replicating in people. And so I'm particularly concerned about the emergence of a new variant of concern in areas where we have limited vaccination and no sequencing. So it's vital that not only do we vaccinate the world, but we actually have sequencing across the world. Sequencing is actually quite patchy in some countries. I can see that for example, across Africa, there's a big initiative, people working together. There was a recent fantastic paper in Science where 112 different research groups came together to describe SARS-CoV-2 in Africa. But there really isn't enough, so we have to do more. And in COG UK, we have a training program called COG Train, where we're providing online courses to help people to be able to sequence, together with train the trainer courses and online classrooms. That's not going to solve the whole problem, but we're starting with kind of education. I think for Sue, in terms of international cooperation, there has been very good cooperation. And one of the key areas really has been that people have released their genomes, their viral genomes, into a global database for everybody to see and everybody to share. And so from that perspective, there has been very good cooperation. And there's been some strong leadership from WHO in terms of genomics. They've been very helpful in terms of coming up with a new naming scheme so that we have names for variants of concerns rather than the places from which they originated, which was actually very stigmatizing. And so overall, there has been good international cooperation. However, there's a lot more to do. So what we'll need to do, when we went into the pandemic, we didn't have an international structure for global pandemic preparedness, and that includes sequencing. And that is what we need to do. So we've done quite a lot in the times of emergency, but looking ahead, we need something that's much more sort of robust and structured. And you may well know that there are discussions ongoing at WHO and elsewhere to develop pandemic radars and hubs for pandemic preparedness.
And Kenny, I wonder, you alluded to some of the international collaboration and in fact, in the context of the power of the UK work for human genomes here. Give us a bit of a flavour of what's been going on.
Yeah, so I mean, I didn't mention this so far, but Genomic was always conceived as a global study, and we, from the very outset, took an open source model. So all of the study documents, the grant applications, the kind of source code that takes so much effort to write to make a study was completely open. And the intention was that intensive care doctors and clinical staff around the world would be able to pick that up and establish their own studies. And because we recognise that in order to tackle the really big problems in critical care medicine, we need to recruit over a very large area, and we need to recruit across the full spectrum of diversity of humans. So we are recruiting now in the UK, Ireland, Canada, and Brazil, and there are studies and set up across many other countries, including Australia, Pakistan, and Sierra Leone. I think the beautiful thing about genetics, about host genetics, is that the statistics force everyone to work together. You always find more if you combine your data. And so the genetics community have shared their data very openly, and been able to do international meta-analyses very quickly and efficiently. And really, there's the genomic study has been by far the largest of the contributors to the international meta-analyses, and there's been a very respectful and collegiate approach to that. I think in future, the founding principles of the genomic study that we state at the beginning of every collaboration are respect for sovereignty and capacity building, and I think they're the principles that we need to build on in order to engage respectfully with clinical and genetic researchers all over the world. Where we're building new capacity, it has to deliver something useful for the clinicians, scientists, and the population. So the approach that we're taking is to engage with an extraordinary group of people who lead an intensive care audit network, where they go to a new country where research infrastructure and critical care is completely absent, and establish at the beginning clinical audit, where all that happens is data recorded for each critically ill patient, and then that enables local intensive care units to compare themselves to their peers, see how well they're doing, and just the fact of doing that drives up the quality of care for patients. But of course, it also creates the infrastructure where that we can then build on for just getting a consent form signed on a single sample, which is fairly straightforward for a genetic study like Genomic. And then, of course, building on to doing randomized clinical trials. So building that infrastructure is possible all over the world. That's already been demonstrated. And what it needs is investment and momentum.
Brilliant. So I wonder, I'm hoping we might have a chance for just a couple more questions. So one that actually I'll take is a question from Mohalb, who is interested about how we ensure subjects' genomic privacy is maintained in the context of us talking about their whole genome, so the person's whole genome. So the approach we've taken in this study is the same as the approach we take generally across the work we do, where participants are engaged in a consent, a choice to join our research programs, and on the principle of people understanding what we do when we hold their data, it being their data. And what we do with the whole genome data is we de-identify it, so we take away sort of direct identifiers. Of course, we still need to be very careful because a whole genome is a whole genome. And the model that we use is one which we often refer to as being like a reading library approach. So we have a trusted research environment where the data is held in that de-identified fashion. Accredited researchers who declare what the research they are planning to do are able to access that data, to visit that data, and only take away sort of the metadata, the summary data that they've derived, the results they've derived, and so that maintains that control over what is very personal data. I just wonder, Kenny, there's one more question that I think people would be interested in about the relevance of this work. You mentioned that it built on previous work, including on Ebola and other emerging infections. How will the work you've done here help us respond rapidly to sort of other infections as they emerge, like that, or Ebola and so on specifically?
I hope that it will. I mean, it's generated momentum. So we now have particularly data infrastructure in the UK for linking the data sets that we've talked about, but also we've demonstrated that this stuff works. So we've demonstrated that genomics can suggest host treatments. We already knew from other diseases that more than doubles the chance a treatment will be effective. Sharon and the COG UK team have demonstrated the extraordinary power of of pathogen sequencing. So I think we've shown what works. In terms of infrastructure, we've spoken a lot about the UK. I think it's very unlikely that the next outbreak will arise in the UK. So there's a tremendous amount of work yet to do. Just as it's never the disease you expect, it never arises in the place where you expect it. So building both surveillance and operational research capacity around the world is going to be a major challenge, and we'll need continued leadership from WHO and an open and adaptable mindset from clinicians and scientists.
Brilliant, thank you. So we are just sort of coming to time, so I want to thank you, Sharon, thank you, Kenny, for joining us. I've really enjoyed the conversation and sort of been interested in where it takes us. I think the themes of collaboration across the community, I think that crucial theme of people power, not just the community, but of the people whose data, who've agreed to have their data included to power these findings, which as we've seen have, and we've heard in the news day to day how important these discoveries have been. I think it's so striking what has happened in this pandemic, and that humble optimism you talk of, Sharon, I think is a really nice and important sentiment to take away. I've also been struck, as we've talked, by the importance not just of the wise historic investment in infrastructure, but also the importance of ongoing investment in infrastructure, whether that's keeping us at the forefront of the technology, but some of those other aspects that we've talked about, so that we remain in the UK both a sort of a real hub for UK life sciences, of sort of genomics as a scientific superpower, but also so that we can respond rapidly in future pandemics. So thank you again. Thanks for joining us for this session. And for those of you joining online, there's more to follow in the chat related to this session, and also follow us on Twitter during the day about this session and about the others that will follow. Thank you very much.
Thank you.
morning. I'm Ellen Thomas. I'm a clinical geneticist and I'm the clinical director at Genomics England. And I'm delighted to be joined this morning by Professor Dame Sue Hill, who's the Chief Scientific Officer at NHS England, and by Chris Wigley, who's the Chief Executive Officer at Genomics England. Please do ask us questions via the Q&A function. We're looking forward to discussing as many of those as we have time for towards the end of this session. So this morning, we're going to be discussing the contribution of genomics to patient care in the NHS. And genomics is the study of our DNA. And we've discovered over the last couple of decades that in terms of the pattern of genomic variation that we all have, we all differ from each other a lot more than we'd previously anticipated. And a lot of this genomic variation is what makes us all different from each other, but it doesn't necessarily have a direct impact on our health. But there is some genomic variation where a particular variant or a particular combination of variants directly cause disease. And the goal of the Genomic Medicine Service is to use that information in the care of patients. So I'm going to ask Sue, first of all, to introduce herself and to talk to us a bit about the journey that the NHS has been on in increasing the use of genomics in patient care.
Thank you very much, Ellen, and good morning as well to Chris. And good morning to all of you listening to this showcase on genomic innovation. I'm the Chief Scientific Officer for England, as you've already heard, and my role in NHS England and Improvement is as the Senior Responsible Officer for Genomics in the NHS. And I just want to start by saying the NHS has had genetic services since its inception in the late 1940s, and our first laboratories came into being in the early 1960s. And they were, at that time, looking at whole genomes, but through looking at chromosomal patterns. They're important because the chromosomes actually contain our genes, and they are part of that essential instruction manual. And when things go wrong, they end up in causing disordered function and then disease. From an NHS perspective, we of course want to make sure that we have the best possible outcomes for our patients. We know that some people live with a rare and inherited disease, and sometimes it takes a long time for those individuals to present themselves to clinical services and to indeed to get a diagnosis. In terms of everything that we're pulling together at the moment, in partnership with Genomics England, we are trying to make sure that that diagnostic odyssey is minimised, but also that we expand what we look for in terms of the potential for genomics to influence the disease course for rare disease patients, or indeed to provide a diagnosis that may help families and other members of that family. But importantly, and something that's often overlooked, is that cancer is indeed the cause of a genomic mutation. And so part of our service is also to focus on cancer and to ensure that we undertake comprehensive genomic profiling of cancers to make sure that we can personalise and stratified medicines for those individuals. And finally, there are of course more common diseases in the population where genomics plays a part and where we need to find family members that might be influenced by that particular genomic mutation, or indeed adverse reactions to drugs that can cause quite severe reactions in some. So Ellen, to answer your question, and that was quite a long-winded answer for you, I think genomics is central to what we're doing in the National Health Service and is central to us improving outcomes for patients and their families.
Thank you very much for that comprehensive introduction. And I wonder, Chris, whether you could talk us through the contribution that Genomics England has made to some of the journey that Sue was talking about through the 100,000 Genomes project and now into the Genomic Medicine Service.
Absolutely. And as Sue was saying, the history of genomics in the NHS is rich and storied. I guess around the turn of the millennium, there was this big moment with the Human Genome Project. The first time that we had a full map of all of the 3.2 billion base pairs that are out there in the genome. And that, I think, opened up a vision of, if we can understand everything in the genome, how can we use that both for healthcare to treat patients and also for research to understand our bodies, what kind of drugs work, and so on. And Genomics England was created as a vehicle to really build a data set and a set of operational practices that would allow us to take that vision into reality in partnership with the NHS. And so, as many people are aware, the then Prime Minister in 2012-13, David Cameron, and his family, they had a kid, Ivan, who sadly had Ohtahara syndrome, which is a genetically based or driven disease. And like many families, they became deeply expert in this condition and the science around it. And in conversation with people like Sally Davies, who was then the Chief Medical Officer, and John Bell, who was John Bell, the Regius Professor of Medicine at Oxford, came up with this idea around if we could sequence 100,000 genomes, and doing just one as part of the Human Genome Project had been a couple of $1 billion and quite a long time, so this had become much more affordable and kind of rapid at that point. We can really start to understand those variants that you talked about, which ones are common, which ones are rare, which ones actually drive disease, and so on. And so, in partnership with the NHS, Genomics England went through this journey of the 100,000 Genomes Project, thanks to all of the people who participated and offered their consent for their data and tissue to be used for the project. Sequenced 100,000 genomes and through the NHS returned those results to the participants. And that really formed the basis of the whole genome sequencing part of the NHS Genomic Medicine Service today, which, as Sue said, is very wide-ranging and goes from single gene tests into exomes, which are all of the genes, deep read panels for cancer, and then all the way to whole genome sequencing today. And that interface of the healthcare delivery part of that service, and then de-identifying that data, making it available to research, and then using the insights from research to feed back into healthcare through the National Test Directory, which Sue and team own and update every year, means that we can really have a learning system that we can keep sort of learning, updating, and kind of moving those horizons forward for the benefit of patients and research participants.
Excellent. Sue, I'm aware that you've also been very involved in the mass testing within the NHS for the COVID-19 virus. And that's a context where we're also using DNA-based testing, but instead of testing the DNA of the people suffering from the COVID virus, we're looking at the DNA of the virus itself. And has there been an impact of that mass testing programme on the rest of the Genomic Medicine Service?
So from an NHS perspective, we've been both testing using a molecular test called PCR testing to actually identify whether the virus has been present. As part of the broader response to COVID, we've of course been using those sort of tests, that type of tests, for mass community-based testing. But one part that I've been really delighted to be part of is to establish the sequencing capacity that's been required to sequence the virus and to understand variants that may be circulating in the population, or indeed brought in from those people who've travelled abroad to countries where other variants may be circulating. And those may come back in and then start to be circulated within our population. It's required quite, us to think collaboratively, how we can work with the private sector, how we can stand up new facilities, but also how much we actually need to understand at a surveillance level the type of sequencing we need to do in terms of the numbers, but also how we support national studies. And those national studies are both reference studies within the population to help understand the relationship to vaccination or, for example, antibody studies, but also, and I'm sure something that Chris will come on to, is how it can be linked with eventually the whole genome sequencing of individuals that have been affected by COVID, that's been part of the genomic study. And we can come back on that hopefully a little bit later on. But to specifically answer your question, Ellen, which was about the impact of standing up that sequencing, and because of this rich history we've had in the NHS of using genomics, of bringing in new technology, of using whole genome sequencing now in a partnership with Genomics England. And part of the reason that partnership was established was because of the size of the data sets, the ability actually to analyse those 3.2 billion letters in our genetic code, and to do that in a way that's rapid and can be fed back to patients. So some of that expertise has been in the NHS has been used to help establish the sequencing capacity that we've needed, because we all know the number of positive cases we've actually had in the population.
And I wonder whether the greater awareness possibly across the NHS and beyond of the use of molecular tests in healthcare has really been driven in part by that mass testing programme as well.
I think there's no doubt about it. The British public now know about molecular testing, they know about PCR testing, they know about lateral flow testing, but they also know about the importance of sequencing to find variants. So although that might not necessarily translate across into human disease, I think with the outcome of studies like Genomic, and with our ongoing, I think, joint approach to making sure that we upskill the British public in terms of their knowledge of genomics and how it can contribute to health and also better health and outcomes, it's all coming together in a way to create a perfect storm.
Absolutely, and I'd really like to move on now to another context where we're using sequencing as an expansion area within the NHS, and that's thinking about cancer treatment and cancer care, where we are sequencing in this context. We're sequencing the DNA and the genome of the tumour itself, and it's the changes in that tumour and the changes in the genomic pattern within that tumour which may have driven or caused the tumour to spread. So in the context of cancer care, Sue, I wonder whether you could give us a sense of how genomics is having an impact on patient pathway.
So of course, from an NHS perspective, we have to think about how we can support the some 350,000 people that get a new cancer diagnosis every year, as well as those that get a reoccurrence of that cancer, and to think about how we best bring in genomics more comprehensively across that population. Part of what we've been doing, though, is moving towards, and Chris mentioned this earlier, which is to establish a much greater gene panel, so number of genes that we look for, or molecular targets, as we often refer to them in cancer in particular, that might be both DNA based, so part of that instruction sort of manual, but also part of the translation, which is the RNA side of that functional genomics pathway. How we do this in an affordable and more rapid way. So we've been introducing 500 gene panels that are being applied in what we call a pan-cancer approach. So they contain all the targets for all the tumours that we see within the NHS, and also all the targets that enable treatment to be stratified. So by that I mean that different treatments can be used for different people in terms of the molecular signature that they display. But what's really important is that focus is on genes. What we've known from the 100,000 Genomes Project is we have somewhere around 25,000, and there's always a number where it could be between 25,000 and 30,000 genes, but also the genome, with all those letters that Chris mentioned, that there are parts of the genome in areas that we call non-coding parts of the genome that are almost like areas that we haven't really read properly before. And so part of our strategy is to introduce more whole genome sequencing in cancer, because we think in addition to those 500 gene targets on panels, we think there's additional information that we can gauge from sequencing, whether that's in the sequencing we do today or whether it's in a different type of technology that enables us to look for very specific changes in that cancer tissue genome.
Yeah, and it's incredibly exciting now to be at a stage where we do have this kind of range of tools at our disposal, as Sue says, to kind of pick from depending on the situation. And just to pick up on the point about what we can learn from whole genomes in the sort of non-coding region, we're starting to understand that what back in the sort of 1970s and 80s was sometimes referred to as junk DNA, so this DNA which is not in genes and coding for proteins, which is 98% or so of the whole genome, that actually signals in that DNA, whether it's variations there or rearrangements and structural changes, can actually tell us a lot about whether an individual person is going to respond to a given treatment or what the best approach might be for them. And I think, like all of our work, it really comes down to the sort of human stories that we can influence and hopefully improve through this work. So it's incredibly exciting that today, through this partnership of the Whole Genome Sequencing Programme in the NHS Genomic Medicine Service, any kid or young person who gets cancer in the country will be eligible for whole genome sequencing. People with blood cancer, which is often sort of very aggressive and challenging, will be eligible for it, and other types of cancer as well. And with Sue and her team and the test directory kind of expanding the indications that are eligible for this, we're hoping that we can bring sort of diagnosis and personalized treatment to more and more people. And I don't know if we want to go into it now, but I think it's also a great example of where research and clinical activities are really kind of coming together.
So what I was going to, what you just prompted me to say and to tell the story of was an 11-year-old child being seen and treated at Great Ormond Street. And through whole genome sequencing it was a haematological cancer, a blood cancer. And that although other tests had been done and usual treatment given, the patient wasn't doing very well. And whole genome sequencing revealed a particular type of blood cancer, myeloproliferative blood cancer, that enabled that individual to access treatment they wouldn't have been able to access before, and they're doing very well. And we've had really good examples of children with brain cancer. Through the 100,000 Genomes Project, 50% of those 18,000 patients with cancer who participated had an actionable outcome. That was either about leading to a change in their treatment or accessing clinical trials. And I think that's a really important part of where we're going in cancer. But of course, from a National Health Service, we have to be in a position to offer the most appropriate test for that particular condition, and that's why we have experts who work with us to determine what what tests should be used for what type of, whether it's a rare disease condition, a common disease, or whether it's a cancer.
Yeah.
I'm very interested in what you've both said about the technologies that are being used in the cancer testing with a combination of the panel tests and the genome sequence tests, and the approach that the NHS is taking of really streamlining and mass producing the sort of wet lab phase of those tests in order to get the maximum value and the maximum robustness from those tests. But then that process of the expert clinical scientists within the NHS really delving into that data so that the data production becomes pretty routine, but the expertise required to delve into that data and to produce the personalisation and the individual insights is then the key value that the NHS is adding to that data, which is really contributing to patient care. And that's a very interesting model.
Yeah, and just to pick up on that, and this point that Sue and I have both touched on about, I guess, this innovation pipeline in cancer, it's a really good example of how the worlds of healthcare and research are so linked here, in that people who are coming through the whole genome sequence service within the genomic medicine service are asked if they would like to participate in research. And obviously, particularly for cancer patients, that can be incredibly relevant for things like clinical trials, like Sue mentioned. And we're finding that a very large proportion of them, the vast majority of people, are opting into that, which is fantastic. That then means that we can work with those participants to answer questions which are just at the sort of edge of what we understand in this field. And Sue and I co-chair a group called the NHS GMS Research Collaborative, which is quite a mouthful. But what that does is it brings together the work that's happening in the NHS with the Genomic Medicine Service, with the National Institute of Health Research, and with Genomics England, so that if researchers in academia or in biotech and pharma are doing work in this space, there's a single front door, so to speak, to come to be able to access those assets in an appropriate way, do the work on the research, and then bring those benefits into the clinic. And one proposal that we've had just recently, which is, I think, a really good example of this kind of exciting ability to innovate just at the edge of what we know, is exploring the question of, can we use whole genome sequencing insights to understand and kind of triage who, for a given cancer, will benefit from chemotherapy and radiotherapy, or who won't. And we know that from a patient perspective, many of us will know someone who's been through chemo, really horrible, long process, lots of pain and suffering involved. But if it's going to kill off the cancer, absolutely worth going through that journey. But if you're a patient who's going through that journey and it doesn't impact the cancer, that's just really painful and kind of unnecessary. And also, to the point about affordability, it's very expensive for the NHS to give these treatments if they're not actually going to benefit anyone. So, question mark, can we establish through looking at someone's genome, okay, you're one of the people who will actually benefit from this, and so it's going to be long, it's going to be painful, but it's going to be worth it. Or actually, you're not someone who's going to benefit from this, so we'll take a different approach. We'll maybe explore what kind of clinical trial we can get you onto. And that kind of, where we can see those insights from research coming straight into benefiting patients who are going through the service, I think it's just incredibly exciting time and kind of space for these technologies to be really coming to bear in the clinic.
I just wanted to bring up, I guess, a couple of points in relation to that, Chris. And I've made actually no secret that I've had breast cancer. I was a participant in the 100,000 Genomes project. I learnt that actually my breast cancer was genomically quite quiet. But as we move forward, what we have the potential to do, if we as we will be doing, saving those whole genome sequences and their analysis, we have the opportunity to go back and look at it again when further evidence becomes available. And that's the same for rare diseases and any of our common diseases. So that is really a positive benefit from whole genome sequencing in terms of it is something that keeps giving. But the other point I wanted to make was from the 100,000 Genomes project in particular, this alignment between routine clinical care in the NHS and research has so many benefits. In the 100,000 Genomes project, we returned 141 diagnoses to patients that wouldn't have had them if there hadn't been academics and researchers from industry looking at the more complicated variants that didn't necessarily have all the evidence to bring them into the clinic and be fed back to the patient at that time. There's another 131 still waiting for that evidence to be generated. But it just shows us what we can do as we go forward. When we have what we call variants of unknown significance, where we have to establish maybe other experiments to determine whether they are indeed pathogenic, they would cause disease in that patient, or to look at all the data that's been collected across a person's life course to understand whether it would fit with all the characteristics that they're presenting. And the other thing, Chris, and I think people often don't realise this, is how we've used the learning from what was the 100,000 Genomes project at that moment in time, but will be through this alignment between ourselves, through the NHS and Genomics England, how it's enabled us, for example, to design our rare disease gene panels.
Yeah.
What do we need to introduce in the NHS to systematically investigate, let's say, hematological rare diseases or those associated with cardiology or with neurological presentations. It's enabled us to understand how we need to standardise an approach to reporting and interpretation. And part of this, in the NHS, our principle is equity of access, driving the same outcomes for everyone, wherever they live, whether they live in Whitehaven, Penzance or Goole, for example. And I always pick quite extreme examples because I think that's what the National Health Service is about, and that's what genomics has to be about as we work together. Driving that levelling up agenda, but using everything we can to ensure we have the best possible outcomes for our patients and our population.
Yeah. And it's a great example, Sue, of how that kind of infrastructure and machinery means that this sort of learning system that we've talked about really kind of comes to life. And I think it's a place where Great Britain is really well positioned to lead because of the nature of our National Health Service. And it means that when a smart researcher finds something out in, could be Oxford or Cambridge, could be Manchester, could be Newcastle, could be Penzance, right? Could be, or Exeter.
Or Goole.
Or Goole. That research can feed into the system and then be available to everyone across the whole country.
But we have to do it in a controlled way, because what we learnt that previously, and one of the reasons we built upon the learnings from the 100,000 Genomes project and other evidence we had in the NHS, was that we have to be systematic. But we are unique. We're an integrated healthcare system, although some people would say it's semi-integrated, which in actual fact it probably is if we start to look down at a primary care level. But we have, all of us, we have longitudinal health records that can actually shape the type of characteristics we might present at any given time that will contribute to the overall relationship between, you know, what we see in in terms of genomic testing or our DNA or our RNA, and indeed what is presented by us in terms of abnormal function or other types of symptomatology, often referred to as the phenotype. So that genotype-phenotype interaction.
Absolutely.
I think that leads us on really nicely to the world that I inhabit mostly in my clinical life, which is seeing patients who have rare and inherited disorders. And I think one of the big developments that we've seen through the 100,000 Genomes project and into the Genomic Medicine Service is the ability for us to really cast our net widely and do a broad genomic test. So we often do what we call a trio exome or a trio genome. So that's where you often sequence a child with a developmental disorder, and both of their parents and compare either all of their genes in an exome sequence or the whole of their genome in a genome sequence. And that really enables us, when we strongly suspect that there is a genomic disorder, it enables us, but if we can't precisely pin it down by examining the patient and by talking to the patient, we can really do a broad test. And then once we've got the data back, that's the point at which the clinicians and the clinical scientists can get together and throw their brain power at that enormous body of data to see whether we can really pull out the high value healthcare information out of those broad genomic tests. And to be able to do that in the diagnostic context and not just when there is a research project that happens to be available to us in a particular place at a particular time has made a huge difference to the way that we look after our patients. And I know that, Sue, there are a number of different services within the NHS, within the Genomic Medicine Service at the moment, with specialist laboratories offering these sorts of broad tests in a number of different clinical contexts. And I just wondered whether you might tell us a little bit about some of those clinical contexts and what sort of impact that care is having and the personalisation that can really result from those tests.
Yes, so we of course have whole genome sequencing in rare and inherited conditions that is driving that triome-based approach and trio-based approach. I'm not making up a new word here, just everyone, just to be clear. But I think it's worth remembering that both in the 100,000 Genomes Project, over and above standard of care, so what had been, exhaustively tested for in the NHS, when patients came into the 100,000 Genomes Project with rare and inherited disease, we saw what we call a diagnostic yield, so a diagnosis returned in about 25%, but that went up to 50 to 60% in some conditions, so quite condition specific, which is what we would expect actually. Yeah. It's not unusual. Yeah. Now with the current live whole genome sequence service in the NHS, which is a world first. Nobody else is doing it in the world. And that's why this partnership is so unique between our two organisations. We are already seeing in some diseases like cystic renal disease, so a particular type of renal disease, we're seeing diagnoses being returned in one of two patients. I mean, it's just absolutely phenomenal. And that is going down to a diagnosis being returned in 1 in 3, for example, for some other conditions. But what we have done from NHS England is establish 3 national services. One is a national exome service for seriously ill children and babies, so they're often referred to as NICU and PICU, and that's a national service run out of Exeter, and we've had around 1,200 referrals to date, and we've got a diagnosis in about 40% of those, and that's resulted sometimes in changes in treatment that have meant that those babies or children have left intensive care units. Sometimes previously they may have been there for 2 years until this has been established. Really astonishing. We have a national fetal exome service that is really truly world leading, run out of Great Ormond Street and Birmingham, and that's giving families back diagnoses, again at that similar sort of level of 30 to 40%, where they can make very difficult decisions. The same with the NIPT, the non-invasive prenatal testing service that we're trialling in 3 organisations at the moment, that helps in terms of identifying whether a pregnant woman is carrying a baby with a high likelihood of Down syndrome. And I have to say I'm really grateful that we've done that, because one of my nephews, his wife has just had that test. And so it's a real comfort. Yeah. Or in sometimes, part of a heartbreaking decision, that those sort of tests are now available more systematically.
And I think, Sue, that focus on the human stories is so powerful, right? And I guess almost to place some of that in context, we've talked about doing trios, right? The patient themselves, their mum and dad, to try and understand what's happening. And indeed, this was actually the case with the Camerons, with Ivan and Otara syndrome. The big question was, if we have other kids, will they also have this combination? Is that 100% chance that we know that if we have another kid, they'll also have this variant in their genome? Is it a 50-50 chance, or is it actually what we call a de novo mutation in that genome? So it's actually a 1 in a million or less chance. And those kind of human decisions about family and so on are so profound. And also, to Sue's point about diagnosis, just a couple of, I guess, contextual points there. One is that the baseline before these kinds of programs was that people would often be left without a diagnosis for years, for decades, or for their whole life, a disease without a name. And actually just getting to a diagnosis is so powerful. And I remember one of the families from the 100,000 Genomes Project had a son, Owen, who had an undiagnosed developmental condition. And the family were kind enough to talk about their story on a video. And Owen's mum speaks incredibly eloquently about the power of getting a diagnosis, and it's again such a human thing. She says, "Oh, I was so relieved because I thought it was something that I had done wrong. I thought it was something that we had done wrong, and actually we know it's just this glitch in the genome." And as humans, we could just have this ability to kind of punish ourselves or to kind of tie ourselves up in knots. And actually having that diagnosis and saying, right, now we know kind of what the enemy is, so to speak. And that allows you to organize with other people who have the same condition, to create communities, to lobby for research into diagnoses. And of course, to, as Sue said, the benefit of having the whole genome on file and able to go back to through the sort of research library is what we sometimes think of as the kind of hopes and dreams factory part of what we do at Genomics England. That there are researchers out there working on these conditions who can then potentially a year, 2 years, 3 years down the line find out something about that case. And there are biotech and pharma companies working on developing treatments for those conditions. And that's hopefully how the magic happens, effectively, how we keep making progress against these terrible conditions.
I think though, Chris, part of this it's all come together, our ability to interrogate large data sets, to analyse them, the change in the technology. And so a part of this is how we ensure that the innovation is adopted. But also part of this is how do we spread this out across the whole of the NHS into all clinical specialties, rather than the specialists like Ellen who work as a clinical geneticist really looking at rare diseases. But what we know is 1 in 17 people in this country will develop a rare disease. I think that's an underestimate. Yeah. And so part of what we're doing in the NHS is to establish NHS Genomic Medicine Service alliances to really drive the embedding of genomics across the NHS, across all the different medical specialties, but using human stories, whether it's stories that you've told, whether it's about Jessica, who was another contributor to the 100,000 Genomes Project, and her mutation that showed she just needed actually just a special diet, or the people we're diagnosing with monogenic diabetes who don't need to be on insulin anymore and can resume their lives, as HGV drivers or whatever they want to do, or how we're linking inherited retinal diseases with the ability to treat with gene therapy so they can see again. I mean, this is just, we're at a moment in time where everything's come together, but the pace will continue to accelerate as we're able to do more analytical techniques that you and the team will be leading, Chris, whether we're bringing in new technologies that have industries being developed, and where we use people like Ellen and others to help us really understand what's happening, and that we convert those messages to people in our healthcare system who actually are not specially trained in very highly complex genomic conditions. And I think this is going to be an extremely exciting next decade, actually, and beyond.
Absolutely. And it's interesting, all of the data, the technology, the analytics and machine learning and so on, as you say, are all coming together in these incredibly exciting ways. And I was just looking at the laptops which are around us in this room and thinking the database that we kind of safeguard at Genomics England on behalf of the country, the National Genomic Reference Library, now has approaching 60 petabytes of data in it. And if we think about a sort of high-end laptop that someone would buy for themselves these days, maybe half a terabyte, so a couple of 100,000 laptops worth of data, which probably would stretch the length of the country. I'm trying to think how long that is. So the data sizes are kind of galactic, and we need to use all of these complex bioinformatics and machine learning techniques to spot that slightly longer or slightly shorter bit of hay in the haystack, or the slightly kinked bit of hay in the haystack. But then we really need to do a lot of work on how do we communicate these incredibly complex kind of statistical and mathematical insights in a way which is relevant to a clinical team, like a doctor, a clinical geneticist, a genetic counselor, or whatever, and also to a patient and their family and so on, so that people can actually act on this. And I think that we need to keep working on how we interpret, share, communicate these incredibly complex insights, as well as the kind of the wizardry that to some extent can sit sort of behind the screen. Yeah.
And I totally agree with you, Chris, because behind all of this, there is an individual.
Yeah.
There is an individual with a family, and I know we are all really totally patient-centric in what we do, and that's what we need to continue to do. Yeah. It's how we can influence individual lives and those of their families and carers.
And sorry, you probably will
sorry,
think some questions. We're getting over enthusiastic, but we should touch on trust at some point as well. I think that's...
Yeah, no, I was just going to actually bring in pick up on one of the examples that Sue used, which is the polycystic kidney disease example. This is actually very much at the common end of the spectrum of rare disease. Around 1 in 500 people in the UK have polycystic kidney disease, and this is a condition which causes patients to develop kidney failure and often need a transplant or need dialysis at some point around middle age. So it's a fairly significant cause of illness in the country, and I think it's a very interesting example of a lot of the things you've just been describing. The principal gene that causes polycystic kidney disease is extremely complex. It's technically very tricky to look at that particular gene, and that has really limited the number of people in the being cared for in the NHS who have a genetic diagnosis for their condition. We've assumed until recently that we needed to manage people without a genetic diagnosis in most cases. But that's really changed now, because we can examine the gene using whole genome sequencing, and so we're now really scaling up that testing. And that's been a two-way driver with the fact that there's a new treatment called tolvaptan for polycystic kidney disease, which can delay the progression of kidney failure in patients who have particularly aggressive forms of the condition. And the genetics contributes to understanding who will benefit most from tolvaptan, and the tolvaptan protects people's kidneys as they go through their middle years. So having those genetic diagnoses is contributing to that treatment, but it's also helping people to understand who in the family who in the next generation might develop kidney disease. We at the moment, if we can only scan people, then we have to wait till the age of 30 to know whether people are definitely free of the condition, and do regular ultrasound scans. And when you're talking to families who have small children, maybe primary school age children, and you have to say to them, well, we won't know until these children are 30 whether they definitely have this condition or not. That feels like a long wait to parents. And if you can do a genetic test to work out which children need their blood pressure monitored and need their kidney function to be kept an eye on, and which don't, then that makes a big difference to families. And also for people who are considering extending their families, it then opens some of the options that Sue was talking about, including pre-implantation genomic diagnosis, where you can only reimplant embryos that don't have the condition, which is also available within the NHS. So I think it's just a really nice example of the ecosystem of the way that the technology, the pharmaceuticals, the patient experience, and also the implementation within the NHS have changed in recent years. And And I'm just in the process of really handing over the management, the genetic management of the patients that I see with polycystic kidney disease back to the kidney doctors and the kidney clinics, who are picking up all of the basic elements of that management and taking on the outputs of these terribly complex genomic tests and really understanding what that means for their patients and how they explain that to their patients, and when they should phone a friend, when has this got complicated and we really need somebody who's more expert in the genomic aspects of this condition, and when is it actually we're fine managing this in the kidney clinic. So that just feels to me like a sort of vignette of really a lot of the elements that you've just been talking about.
And as is Alan, as you know, because we're working together on this, familial hypercholesteremia, which is an NHS long-term plan commitment, because this is a condition associated with a high circulating level of fats in the blood, associated with, an excess number of deaths, particularly in men below the age of 50, but that can be treated by drugs such as statins. And the community in general have not always access genomic testing, but also there's a big population out there who are sort of living with familial hypercholesteremia who've never actually presented into their health system. So we know that this we can test for it, we can treat for it, but it requires a particular way of working to ensure that we can find the case that has the high circulating fats in their blood, and then look at all their family members to understand whether they're carrying it, or indeed whether they can start early treatment, and that includes early treatment in childhood to prevent cardiac events. So that there are many areas that we can take that more systematic approach to.
And I think, Ellen, your sort of vignette also, I think, paints a great picture of what mainstreaming these kind of techniques looks like, right? You're saying, I personally don't have to do the details of each of these cases anymore. Actually, the mainstream clinical teams can deal with those. And I sometimes get asked about, okay, well, if we're talking about levelling up, we're talking about a national scale service. Do we have enough, clinical geneticists? Do we have enough genomic counsellors, etc.? The technology systems to some extent can scale to infinity, but there are actually a lot of really important humans in the mix as well. And Sue, this is very much your area, but I wonder whether it's worth touching a bit on if we're bringing genomics into the mainstream of health, how do we actually both bring the existing people with us, and how do we have enough of those people to actually do this at scale?
So, Chris, really good question, and I think we're having a great discussion here. We have, since the start of the 100,000 Genomes Project, actually, we've had under Health Education England, we've had a very focused education programme to start to develop resources to essentially upskill the current workforce in the use of genomics. But of course it has to be more systematic than that. We have to embed it in both the undergraduate and postgraduate training, particularly of doctors. And so we're working very closely with the Academy of Medical Royal Colleges to ensure that it is in the curriculum, the postgraduate curriculum for all the relevant specialties. But again, also part of this is bringing it to life, because if an individual clinician in a particular specialty is only going to see a patient with a potential genetic cause for their disease once every 3 months, but they need to continue to look for it all the time, then we need to ensure that the education and training is right for that particular intervention. So we're doing a lot of work at the moment on individual pathway pieces of work, so that we understand what both the just-in-time education is required, what sort of knowledge we can make available electronically, so people can go and get their quick fact sheet, but also how we can think about the type of support we need to give to patients or family members, especially when they're getting a rare genetic disorder where they might be the only case in the country, or in a very small number of cases, where they really don't know what the impact would be on having other children. Or it is such a sort of severe diagnosis, essentially, with you know, for example, you know, a child dying before the age of 5, which is what happens in many rare disease diagnoses, then we need to work on ensuring that our broader healthcare team have those genomic counselling skills. And so part of this is transferring some of that skill base into our nursing workforce, and that's why we've got a big nursing and midwifery transformation programme in NHS England. But we will never stop needing to do workforce education and training interventions. We're always going to need clinical geneticists because they need to help with those really complex cases and support the rest of the clinical teams. And that includes our clinical teams in cancer, because they've not always been used to receiving very complex genomic information. That means they may even have a choice of the treatments that they do. And we've learned a little bit about this by rolling out something called DPYD testing, which is testing to know whether patients who receive a particular type of chemotherapy called the fluoropyrimidines, whether they are going to experience a severe reaction and sometimes fatal reaction. And indeed, through the rollout of this across the NHS Genomic Medicine Service, we've stopped several potential fatalities in early testing, early understanding of whether those agents need to be used or not.
Yeah.
So this is such a widespread field, but again, just coming back to what I've said earlier, something that is life-changing both for the individuals, but can also be quite changing for the clinical practice of our clinical teams.
Yeah. We had Eric Topol on the G-Word podcast, who was lobbying for every medical student should sequence their own genome and use that as a sort of study aid through their course. I'm not sure if that's part of the vision for medical education, but I think it's an interesting idea.
It's an interesting concept, one we shouldn't actually throw away at all.
I wonder whether we could just come back to something that Chris said earlier about talking about trust in this area. And Chris, one of the big responsibilities of Genomics England is holding and curating the National Genomic Research Library, which holds the genomic and healthcare data of the participants from the 100,000 Genomes Project, from other research projects, and patients who are having their genome sequenced as part of the Genomic Medicine Service are offered the choice as to whether they would like to donate their genomic and healthcare data and join the research library. And you've talked a little bit about the size of the data and the challenge that poses, but I wonder what other challenges and also excitements really go with that data responsibility that Genomics England is holding.
Absolutely. Well, I think the first thing to say about the data that we hold is that it's not our data. It's the data of the patients and participants who've kindly volunteered to share that with us and through us with researchers in academia and in biotech and pharma, and that places a big responsibility on us. The reason that we've called it the National Genome Research Library is this metaphor of a research library. So at university, I did a slightly unusual combination of computer science and medieval history. But for the medieval history bit, I used to go to these obscure libraries where they have all of the old manuscripts, and you get given white kind of gloves, and you're not allowed to take pens in and so on, just a pencil, and you can't take the books anywhere. And that's very much the model that we have with the trusted research environment. You don't have to put on white gloves anymore, but you come in, you can study the data, you can run algorithms, you can do analytics, and you can take out your findings, you can take out the insights, but you can't take the data with you. The other analogy that we sometimes use is the aquarium. You can come in, you can admire the fish, but you can't take the fish with you. You know, it's not a fishmonger's. And so that model is one of the ways in which we try and act in a way which is trustworthy. Why should people trust us with their data? Because we care about it. We're thoughtful about the systems. We're thoughtful about the technology, and so on. The other big part of this is around empowering the people whose data it is to make decisions about who gets to access that data and on what basis. So we have a group called the Access Review Committee that, when we get a proposal from an academic or a biotech or pharma researcher wanting to do some research, that group includes representatives from the participants whose data it is, and they have an absolute veto if they don't feel that the researchers have demonstrated that they have the right ethics in place, that they're answering a question which is going to be relevant and meaningful. They can absolutely say, you know what, go away, sharpen your pencil, think about this research project, and kind of come back when you've thought about it, which is, I think, a really powerful model in terms of not just engaging with patients and participants, but actually putting them in the driving seat around how we do this. And that means that over the course of the 100,000 Genomes Project and the work that we're doing now with Sue and others, we've had more than 100 private companies access the data. We've had 4,000 academics register for research in there. And we haven't had, touch wood, and we're always working hard to sustain this, but we haven't had the kind of issues that we've seen in some other places of people being suspicious about motive or about the way that the research is being done, because we've empowered the people whose data it is to make those decisions. And I think that's absolutely vital, because to your point earlier about the ecosystem coming together, and we need to get both to diagnoses but also to treatments, we have to work with the private sector, as Sue was saying, to do that. Where do treatments come from? They come from biotech and pharma companies. But the how is as important as the what, and we have to do that in a way which is controlled and transparent, and where we're putting patients and participants, right in the driving seat of how we run those collaborations.
Of course, Chris, it starts with the patient, doesn't it? And their family right at the very beginning of their journey through a clinical service and into testing. And the work that we've done together, and particularly with Ellen here, we've done and spent a lot of time on ensuring that individuals can make an informed choice about both their data being shared to ensure that they get a diagnosis. If they are likely to be in a small group of people in the whole of the UK or in the whole of the world, to be able to get a diagnosis, it means that clinical data may need to be shared. And we've actually seen some really good examples where individual genomic data, especially in paediatric cancers, have needed to be shared with clinicians in Germany to understand whether they could access a particular type of inhibitor for that particular type of cancer. But it's both understanding that data will need to be shared for clinical diagnosis, but also to give them enough information to make an informed choice about their data being used for research. And we've done a lot of work to make sure that we've got materials that are acceptable, both to individual patients or, in the case of very small children, to family members. And we'll continue to need to do that. But I think what we've done with the rollout of the live whole genome sequencing service into the NHS, and with all of our trio exome services that we've talked about earlier, we've made time available through people who understand how genomics are going to be used in their care and indeed research, to have those conversations with them, to answer their questions. And I think as a result of that, in our live service at the moment, Chris, those who've had that conversation, 93% have given their consent. I mean, which is a tremendous amount. And I'm always of the opinion that people who access our health system usually want to not only get their own diagnosis, but help others. Yeah. And I think that's really demonstrating that, but also the power of having good information, good trusted environments where the data's going to sit, but also the opportunity to provide them with information and support. Yeah.
So I'd like us to move on now to address some of the questions that have come into us from our community today. So the first question, really, perhaps Sue, you could address this one first, is follows on very much from the conversation we were just having. Anna Dickinson and others have asked, what role will public trust and engagement play in ensuring that patients and their families actively participate in the service and research?
So I'm really delighted that question's been asked, because I realised I didn't say anything about it in my answers to date. But from an NHS England perspective, we've been very clear that at all levels of our system, so we have 7 genomic laboratory hubs, 7 genomic medicine service alliances, sort of then national sort of programme management type of committees. At all of those levels, we've insisted that there is patient and public engagement and involvement in all the governance groups, right up to the national governance group. And we have nationally a patient and communities forum that includes national organisations as well as representatives from Genomics England's participation panel. Mm. I think we can never do enough to involve the people who, as I said earlier, are at both the beginning and the end of this journey. Mm. Yeah. And so we have done that systematically, and we've insisted that in all of our contractual and other arrangements. But I think it's important what Chris has outlined, which is to ensure that access to the research environment also has significant patient and public involvement in the decision making there.
And I think that goes sort of right upstream as well for things that we're thinking about. So Sue's mentioned a couple of times the services that exist around newborn intensive care and pediatric intensive care, and about how these individual rare diseases are quite rare, but collectively they're actually not that rare. And one of the areas that we're jointly exploring is the concept of whole genome sequencing for newborns. Huge ethical questions to be asked, addressed, discussed, huge logistical questions about how you would do that. For those who are interested in this topic, there is a session with Sarah Jane Marsh, who's chairing the NHS steering group, thinking about this later on today as part of this innovation showcase. But as part of that thinking and design piece, with participants from ScienceWise, which is a Wellcome Trust initiative, we've had a series of public dialogues with new parents, expectant parents, young people, old people, people from different regions, midwives, other groups, around this concept, both the initial concept of we could sequence someone at birth to highlight if they have a serious and early onset and treatable condition that we can then get right into treatment straight away, but also this idea of having the life course benefit of having your whole genome on your record and using that to drive wellness or early interventions in other areas and so on. Huge questions. And what I find really inspiring is that often those groups are the ones who are telling us to do it right, but to absolutely get on with it. And these dialogues are quite intense and involve a lot of those groups learning about genomics, learning about these different questions. And what we're hearing from them is, if we can do this, if we can save lives, improve lives, get on with it, just do it right and do it thoughtfully.
Yeah, absolutely. And there's a follow-on question that leads on very nicely from that, from Megan Horton, which is that some individuals and communities are less likely to access genetic testing or consent to research due to a lack of trust or communication or understanding. And how do you think we can improve this so that we can provide more equitable care to patients?
At the risk of sounding like an advert for sessions today, we have our final session of the day is on the need for more diverse representation in the genomic data sets. So this big program around diverse data. And I think it's an excellent question. And we saw this particularly acutely through COVID as well. We saw differential rates of infection, differential rates of severity in different communities, whether that was different ethnic communities, communities in highly deprived areas versus more affluent areas, and so on. The mission statement of Genomics England is bringing the benefits of genomic medicine to everyone, and that final word, everyone, is really important. If we're generating insights which are based on research which is only done on European Caucasian genomes, because those historically have been the ones who've been most of the volunteers in those kind of large-scale biobanks. There's a risk that those insights are not relevant to different groups and so on. So there's a huge amount of work to do there. We've learned a lot through COVID about the very reasonable and often specific questions or concerns that different communities have, whether that's about vaccination, whether that's about participation in research. And I think there's an increasing sort of enthusiasm and belief in the whole community that we need to kind of, to some extent, turn on its head what has historically been the approach of, well, we just need to explain this better to people. Actually, no, we need to go to where those people are, understand their concerns, which are often really legitimate, and work together to come up with good answers to that. Yeah.
So, but in the NHS Genomic Medicine Service, I mean, we undertake somewhere between half to 3/4 tests a year. That is, not at the moment in partnership with Genomics England, not at the whole genome sequencing area. That testing, by and large, is actually representative of our populations. And we have to also, it's quite complex, because all the different clinical conditions we test for, there will be a different incidence and prevalence within different populations. And we know that from the 100,000 Genomes Project that the participation was actually reflective of the community makeup of our population in the UK. That having been said, there's always more that we can do, and the more we need to do. But the one thing we have to do is make sure we have information that's accessible to people whose language is not English. They have to be supported by people who can actually speak and interact with them in their own language. And certainly through our NHS genomic services, we have people who can support them, or they link with the broader support teams who support people where English is not their first language, and understand some of the sensitivities in some of the communities that make up our diverse and wonderful population in this country.
Yeah.
Thank you both very much. The next question comes from Neil Munro. I gather he's not the only person to ask this question, so definitely an important one to address. Please, can we have a little bit of information about how genomic investigation is going to address neurodevelopmental disorders, autism and the like? Are they included? So perhaps I'll start.
You start that one.
Yes, that's a really, really important question. That the conditions that you're referring to in that question are really a sort of group of lots of different conditions, and we know that some of the conditions which cause neurodevelopmental disorders and autism are conditions which are caused by a single faulty gene, and often in that situation that's a single faulty gene where the gene fault has happened for the first time in the affected child and hasn't been inherited from either parent. And that is the classic situation where the type of trio broad approach that we were talking about earlier in the conversation is the key to unlocking those. There are hundreds and hundreds of genes that can be involved in these conditions, so doing them one by one really isn't an option, and we've only really been able to offer routine diagnostic testing in this context since we've been able to do this broad trio approach. And the importance of the genome sequencing service in particular is the scalability of that service. We know that there are a large number of children with these sorts of disorders, and being able to scale that up is really important. Beyond the conditions which are caused by a single gene, there are many conditions which look very similar when we look at and talk to patients. But which are not caused by a single faulty gene, but may be caused by a combination, a sort of bad hand of genes of much smaller effect, but going together with other influences or environmental factors which we don't fully understand. And in that context at the moment, the level of diagnostic testing that we can do is relatively limited, really because we are still understanding the genetic and genomic architecture of those disorders. So it's definitely that there are definitely a number of these conditions which are in the phase of extensive research work and which are really important to research further and to understand better. And those I would say for those disorders really the diagnostic solutions for those disorders are still in that sort of more distant pipeline that we can see coming towards us, but which aren't fully implementable in the diagnostic context until we've got a bit more basic understanding of how they function in terms of their genomic architecture. But certainly intellectual disability is an important category of a test in the National Test Directory, which NHS England have produced to describe who who's likely to benefit from genetic testing in the diagnostic context. And we've seen around a third of the children who've come through that so far in the whole genome sequencing service have a diagnosis. So those sorts of disorders are definitely a really important part of the genomic medicine service and the whole genome sequencing diagnostic service within it. Would either of you like to add anything?
The only thing I was going to add, Ellen, was just around the National Test Directory. And this directory is the mandated directory for testing within the NHS. So what defines eligibility for testing, right up from single genes right through to whole genome sequencing. And the reason I was going to mention it is the way we've structured this is to be able to take evidence throughout the year and to make updates to that test directory at least annually, but more frequently if there are significant there's a significant evidence base that suggests we need to introduce it earlier. So these are the conditions that we're constantly looking at to understand whether we're close to being able to introduce whatever type of testing is appropriate.
And I think one sentence from me, and you can correct me if I'm wrong, which I might be, but I think I'm right in saying that intellectual disability is, if not the largest, one of the largest groups of participants in the National Genome Reference Library as well. So it's a very large research community out there working on a lot of these topics that we can then, through the test directory, kind of bring into clinical care.
It's also one of the areas where the research advances, I would say, are coming through most rapidly, and certainly in my own clinical practice where we've seen results coming back of the sort that we were talking about earlier, where a researcher has picked out something new or something particularly complex which wasn't picked up on the first round of analysis. It's probably the key area where those sorts of results are coming through most rapidly after the 100,000 Genomes project as well. Excellent. So perhaps we could move on now to a question from Carl. To what extent do you see a future of germline sequencing of inherited genetic profiles to predict and manage toxicity risks for individual patients or complex diseases? So I think, Sue, you talked a little bit about the DPYD program, which is within this area. But do you see that expanding and also expanding to complex diseases which aren't necessarily caused by a single genomic fault? So
if I could just stick, first of all, to sort of drug interactions. And I think what we know from what was an initial analysis of the 100,000 Genomes project participants, and around 8,000 of those whole genome sequences, is there were potentially 80% actionability in the drug gene interactions that have been looked for that might affect the way in which an individual receives that drug. It's not all about toxicity per se that leads to a severe side effect. It might be how a drug is metabolized and whether more is needed to be given, but it also gives us that adverse drug reaction, and remembering that we have about 1 in 15 hospital admissions every year associated with the adverse drug reactions. So At the moment, we still have more work to be done about how this would be applied within the NHS. We very much looked at the type of drug gene pair we might need to look at and introduce in the NHS, and we've got down to evidence that supports around 41 drug gene pairs. So it will be around classes of drugs like those to treat neuropsychiatric disease. And in NHS England, we're going to be working on a strategy for introducing sort of whole genome, for pharmacogenomic analysis into the NHS. But it will require further investment to do this over and above DPYD. And as I've said, we can start to look at how we use germline, whether it's germline sequencing, much more broadly. But as Chris has already alluded to, I think through the newborn sequencing programme, there's a real opportunity there to think about how it can be used and used in a systematic way. That having been said, we will look at this within the germline sequences that we've got within the current WGS Go Live system, and look at the evidence that we've got for the introduction. But it does all come down to evidence, and it also comes down to understanding how we're going to implement it in the NHS systematically, and not just for a few, and how much it's going to cost.
Thank you very much, Sue. I think that's an excellent way to close. That really sort of summarises our session very nicely this morning. So I'd like to thank Chris and Sue very much for all of your conversation this morning, and to thank our participants online very much for joining us and for submitting such excellent and interesting questions. Thank you very much.
Well, thank you, Ellen.
Thank you, Ellen.
Thanks, everyone.
Good afternoon, everyone. Thank you for coming along, taking part in the Genomics England Innovation Showcase. My name is Simon Wild. I'm the Director of Engagement at Genomics England. Welcome to you all. This session is titled Newborn Sequencing, the Value and the Vision. And for the next 90 minutes, with the help of a distinguished panel we have here, we want to tell you the story so far of the Newborn Genomes Programme, a new initiative that Genomics England has begun to explore the potential of using whole genome sequencing to increase the diagnosis of rare genetic conditions in newborn children. Please join in with your questions in the Q&A chat, and we'll try and answer as many of them as we can. Just before we begin, I want to emphasize that we're at a very early stage, that our work so far has been listening to and engaging with a wide range of different people and organizations, and that there will be a lot more listening, talking, and engagement before we are in a position to begin any kind of pilot research program to sequence newborn babies. As with any new technology or new use of existing technology, there are a huge number of scientific, technical, ethical, and societal challenges to overcome. But we'd like to describe to you today how we collectively might go about tackling these challenges and potentially realize the benefits that genomics might bring us. The other thing that I'd like to mention, for the avoidance of any doubt, is that everything we'll be talking about today is about postnatal sequencing and not pre- or antenatal genomic testing. In this session, we're going to be hearing from panelists from Genomics England who have been working on developing a vision for the Newborn Genomes Programme, and from those whose work has already had an impact or will maybe have their work impacted on or by developments in this area. So without further ado, let me introduce today's panel. They are Professor Nina Modi from Imperial College London, Rebecca Middleton, who is Vice Chair of our Genomics England participant panel, Catherine Joinson, who's Assistant Director of the Nuffield Council on Bioethics, Sarah Jane Marsh, who's joining us live from Birmingham, who is the Chief Executive of Birmingham Women's and Children's Hospital. Hello, Sarah Jane. And joining me from Genomics England, Rich Scott, our Chief Medical Officer, and Alice Tuff-Lacey, who is our Programme Lead for the Newborns Programme here at Genomics England. So, why newborn sequencing? The idea itself is not a new one. In fact, I learned only recently that in 2001, the same year as the first draft of the Human Genome was published, Francis Collins, one of the pioneers of the Human Genome Project, predicted that it would be feasible within 20 years to produce a kind of report card analysis from a baby's DNA sequence. Well, here we are in 2021, where our achievements in using genomics, and in particular whole genome sequencing, in the NHS you've been hearing about this morning. But without wanting to take anything away from Francis Collins' prediction, a report card analysis probably raises more questions than it answers. What might the possibilities of using data from our genomes to improve our health be? And in particular, what might be the benefits of analysing our genomes proactively at birth? We already have the NHS Newborn Screening Programme, which tests for 9 rare inherited conditions through a blood spot heel prick test just after birth. Can we, and if so, should we, be looking further than this? To help me explore the potential possibilities of using genomics at the start of life, I'm delighted to be joined first by Nina Modi, Professor of Neonatal Medicine at Imperial College London, who for many years has been involved in research and the clinical care of newborn babies. Professor Modi, thank you very much for joining us. So for the past 14 years, you've been running the Neonatal Data Analysis Unit. Can you tell us a bit about what the National Neonatal Research Database does?
Of course, thank you very, very much, Simon. It's great to be here. So my research group runs something called the National Neonatal Research Database, and also another database called the International Neonatal Research Database, 2 databases which are built on the view that families should not be asked the same questions again and again and again, but you ask a question once, record the information, and then make it available for appropriate use for all the other reasons that one might need information for. And also, busy clinical teams shouldn't be asked to record the same information again and again and again, and nor should researchers. So that's the very simple principle behind these databases. But what we've done more recently, which is why I think I come to be with my colleagues here today, is that we've also done exploratory work to obtain the views of families of linking this routine clinical data about sick and preterm newborn babies to DNA samples which are used for genetics analyses. And that pilot project, which we did a short while ago, has really, really, for me, opened up a really deep understanding of the possibilities of obtaining DNA samples from newborn babies and then storing it for uses such as whole genome sequencing and all of the other very many potential benefits that could come from it. Now, we have the wherewithal to do this nationally, and we pretty much have the wherewithal to do this beyond the UK borders as well. But I have to say that the UK is doing a great job in doing this cautiously, carefully, and taking into consideration the views of all of the various stakeholders involved.
Thanks, Dina. So you talk about possibilities here. What do you think the possibilities from collecting, analysing the genomes of newborn babies just after they're born might be?
So many, so many. So let me give you one example from our pilot project. We wanted to do the study both in order to obtain the views of families, but also to answer a really important research question. And the research question was, are very preterm babies enriched, is their DNA enriched for genetic variants that are known to be linked to later neurological disorders like autism spectrum disorder? So that was the research question, but we also wanted to get the views of families about how we went about doing this research. And the answer to the research question was yes. Yes, very preterm babies, in other words, babies born between 2 to 4 months earlier than their due date, are genetically, they do have markers that show them to be susceptible to a range of later developmental disorders. Now, whether these developmental disorders are actually going to emerge or whether it's just that the genetic markers are there is another series of research questions. And so research always opens up new frontiers, and you go from one bit of the jigsaw puzzle to the next bit of the jigsaw puzzle, and that's how science progresses, and that is why it is so fascinating.
Absolutely. And in terms of not just talking about preterm babies or babies that are born ill, expanding this out to a kind of wider cohort of babies that actually when they're born may not appear to have anything wrong with them. What might be the benefits both for clinical practice and for research?
So there's a lot of talk about the phrase is used, the life course approach to life, and that's a very appropriate phrase, I think, to use, because we are born with a certain genetic set of determinants. Whether or not those actually translate into real illnesses further down the line, in childhood, in young adult life, in old age, is uncertain because genes have to be expressed. So understanding the interaction between what you're endowed with genetically, what your subsequent life course exposures are, and how these all interact is one of the great opportunities for the future. In other words, all of us here want to have we want to be healthy in our old age. We are going to be susceptible to a whole range of disorders that increasingly we're vulnerable to as we age. But if our genes can tell us something not only about our original susceptibilities, but how those genes interact with the way in which we live and work, exercise, play, then surely it puts us more in control of having that healthy old age. And that, I think, is what no one would not wish to aspire to. So whole genome sequencing opens up a huge range of wonderful possibilities, not only about rare diseases which might affect a newborn baby immediately, or in infancy or in childhood, but also about diseases that may have their onset later on, and also about how we can take control of our lives and reduce our risks. So enormous possibilities, but it starts at the beginning, and it starts with engaging with families, ensuring we can actually obtain samples from which we can extract DNA and then sequence the genome and use it in ways that are wise, careful, considered, and done in conjunction with the families whom we care for.
Thanks very much, Nina. So you've started to touch there upon the kind of, the possibilities, but also the kind of fact that, if we're going to do anything like this, it really needs to kind of bring the families along with us with a careful consideration of all the kind of issues that are raised. And I want to turn now to Catherine Joinson here. Catherine, you are Assistant Director of the Nuffield Council on Bioethics, and you were part of the Nuffield Council team who have been looking at the ethics of whole genome sequencing. And in 2018, you published a paper looking at this issue in particular. What was it that prompted the Nuffield Council to have a look at this particular topic?
Well, that was part of a really long-standing interest for us in this area. In fact, our very first report 30 years ago was on genetic screening, as it was more called then. But what prompted us in 2018 to particularly focus in on newborn testing and screening was, I think we were moving there more from a sort of theory to reality. So we had the Chief Medical Officer's Generation Genome report, which set out a vision for genomic healthcare, had the 100,000 Genome Project, which was bringing whole genome sequencing into the NHS. But also, we saw a rapidly expanding commercial sector where, many more people were choosing to have their genetic profiling done. So we thought it was a good moment to take stock and set out what some of the issues were.
So tell us a little bit about what was in this report then. What were the main ethical issues that were identified?
Well, I'm going to talk a little bit about that, but I think as well as some of the things we've learned over the last 3 years since that report, and I knew you were going to ask me that question, so I've got 5 areas I just wanted to mention. But firstly, to say that none of these are particularly new issues, and none of them are particularly unique either to this area of healthcare. And also, I wouldn't want to suggest that the people working in this field aren't already very aware of these issues and working on them. So the first area to mention is sort of the big one really, which is how you get the balance right between the potential benefits of this technology and the potential harms and risks. I think it's generally accepted really, many countries across the world, that newborn screening should focus on serious, actionable childhood conditions. But that doesn't tell you everything you need to know, and different countries interpret that in different ways. In the US, for example, they screen for many more diseases. So perhaps does whole genome sequencing and also our increased understanding of rare genetic disease, does that mean we need to rethink how we approach newborn screening? The second area is around children and consent to screening, or any medical treatment actually. So this is proxy decision making. It's the parents that make the decision. So in order to treat those children and the parents with respect, we need to give them the information that they need to make those choices and support afterwards as well. So that's a really important area, and I think it raises questions around capacity and skills within the NHS in order to enable us to achieve that. The third area is around genetic information, which is special. It's very private, very sensitive. I think we're all aware that we need very secure data protocols here, but we mustn't be complacent. Data breaches happen, mission slip happens, that would have a huge impact on public trust. So we need to make sure that we are considering all the time how this data is being stored, and also that people who are signing up to be involved in research or having their genome sequenced are confident that their data is going to be secure. The fourth area is around fairness, and this is becoming a really important concept in public life. How can we make sure this technology benefits everyone? How can we ensure some people aren't disadvantaged? We live in a very diverse society, and families come in many forms. We need to make sure that we're thinking about everyone here. And we also need to think about fairness in terms of public resources as well. Is this a proportionate use of public funds? The fifth area is a different kind of ethical issue. It's around procedural ethics. How do we sort all this out? How do we decide what is the right way to go about this, or the best way to go about this? Whose voices are carrying most weight here? Are we being transparent in our processes? What factors are most important? So that's going to be really important as we go forward, to make sure we do this in the right way, and to make sure that we can make the best of this technology without over-promising and hype.
Yeah, lovely. That's a really, really useful kind of summary of the kind of main issues there. So I think 2018 was when the Nuffield Council report was published. Since then, there've been various documents that have been produced. Sally Davies, the then Chief Medical Officer, produced her Generation Genome report not long after then. And as a result of that, Genomics England was asked to kind of set up a task and finish group to look at the use of genomic analysis in children. And you sat on that group, I think, along with Professor Modi here. What were the kind of main considerations that the task and finish group considered?
Yeah, so that group looked at both the use of whole genome sequencing in acutely ill babies and also in apparently healthy babies, so the screening side of things. I think we quite quickly came to the conclusion that the former was fairly uncontroversial, and we focused most of our time on the latter. We concluded that we would be supportive of exploring the feasibility of using whole genome sequencing in newborn screening through a research programme. So that was, very importantly, the first step to look at it through research. But we highlighted that there were some important ethical and societal issues to work through, and that public and patient involvement should be core to the future of this.
Yeah. So you've mentioned public trust in your earlier comments, and you're talking about kind of making sure that the public are kind of consulted and involved in decisions that ultimately will affect them. So you were involved, and I'm trying to make this sound as though I don't know anything about this, but we've worked very closely over the last year or so to look at this. So we've created a public dialogue process where we went out and actually spoke to members of the public to ask them kind of what their views are around a lot of these issues around whole genome sequencing and its use in newborn screening. And at the time you were on secondment to the UK National Screening Committee. Why was it, do you think, that the UK NSC was wanting to be involved in the public dialogue?
Well, I think interestingly, both Genomics England and the Screening Committee think it was their idea, but I think it just came together perfectly well timed. From the Screening Committee's point of view, I mean, they've long recognised that genomics is likely to become a bigger feature of screening in future, and they also recognised that early engagement would be important here. So that was their real motivation. But the dialogue is going to feed into their work. Probably in a sort of closer time period than to Genomics England's, in using sort of maybe longer term work programme. It's informing decisions and considerations that they're having right now on particular screening programmes. But they're also, I think, keen to view this not as a one-off. The dialogue doesn't provide all the answers, we're done now with that. I think any kind of ethics and engagement reflection should be seen as a reflective, ongoing process.
Yeah, absolutely. And I think we're going to return to some kind of the ongoing engagement issues that in a bit later. So on the subject of dialogue and the public dialogue, let me turn now to Rebecca Middleton. I think we at Genomics England occasionally forget that Rebecca has a whole other life and job away from Genomics England. Rebecca runs a highly successful communications consultancy in the Midlands, but to us, as well as for this, Rebecca and her family are participants in the 100,000 Genomes project, and Rebecca is the Vice Chair of the Genomics England participant panel. Rebecca was also part of an oversight group that was set up to shape and inform this public dialogue project, and gave us some insights into the public perspectives on the implications for the NHS and society of using whole genome sequencing. Let me start with a question for people who might not be familiar. What is the participant panel and how does it work, and what's your role?
So we sit very much at the heart of Genomics England. The panel was formed in 2016 and I'm proud to be a founding member. We're there to ensure that the patient voice is represented at every single level of Genomics England and is heard when key decisions are being made, especially those decisions that concern our data, how it's stored and who accesses it, and to make sure that it is indeed safe. On the panel, we are all 100,000 genome participants, or our family are. So we all have our samples in the National Genomic Library, and we're all invested in the success of Genomics England and all of its projects. And we're a diverse lot. We come from every corner of the UK. We all have very different experiences as rare disease or cancer patients, and different experiences in life, which we all bring to the table, which ensures that we really do keep Genomics England account every day.
Absolutely. And I remember the phone call when I asked if you'd be willing to sit on the public dialogue oversight group. So we had a group of 20 to 25 people that provided input and, helped look at the materials and help us design the process of asking the public about their views and their aspirations and concerns about whole genome sequencing in newborn screening. Why were you interested in the topic? What made you say yes when I spoke to you?
I suppose I do have a background in communication, but I am a patient, and I have been through various kind of NHS pathways as well. So know what it feels like to kind of be on the receiving end and know how important it is to listen. And Genomics England really are a good example of how listening to patients and participants has changed the design of their service and made a real positive impact. So I wanted to kind of see that expanded and see that grow, because there really is power in listening to the patient and listening to the participant and ensuring that there is positive kind of change and impact from that.
Yeah, thank you. And so, a public dialogue process where you're going out to people, to members of the public, recruiting them to kind of join workshops to kind of discuss topics like this. There's obviously kind of a little bit of a difference between a member of the public who perhaps has not been affected or not needed to know about what genomics is all about and things like that. What was your role on the kind of oversight group in terms of what you did for the dialogue project and how you fed into it?
So on the oversight group, we were all there to ensure that we would guide and challenge the process to ensure that it was representative as possible, ensure that we had a good open dialogue. For me personally, as a rare disease patient, I wanted to ensure that the patient's views were represented in that dialogue. So let's take the case study from the slide pack and make it real. So we invited rare disease patients in the virtual room to share their experiences and share their thoughts and how they have been through a diagnostic odyssey, or they have been through kind of various NHS care pathways, what that felt like, and what they have learned from that. And the participants, the dialogue participants, were able to ask some challenging questions and help unpack and tackle some of those ethical questions as well. And the feedback that we also had from the dialogue was that really added to the richness of the dialogue, and that they really got to understand those topics and those issues. So it was really great to see.
And in terms of the kind of feedback, the dialogue report is available on the Genomics England website for anyone who's interested in finding out more about it. What for you were kind of the standout points that the participants in the dialogue, 130 of them, kind of came back to us in terms of what their kind of messages were around the use of whole genome sequencing in newborn screening?
I think it was really positive that they were positive, that they saw the potential and that they were optimistic about let's go forward and explore what this actually means, and that was fantastic. What we, as a panel, what we were also really excited about is that they absolutely recognise that we need to take our time to do this right. Not only going forward for a screening programme, but also now we're talking about the pilot programme, we're talking about the vision for the pilot programme. And they understood how important that was, to ensure that we get things right from the get-go, to ensure that we look at what resources are available, to ensure that we look at the training available. And they understood that the patient journey, if you like, from end to end should be thought through and considered. They also picked out trust, something that Catherine mentioned that is so critical, that we need to build that trust. And to build that trust, you need to let patients and participants in. And again, let them in from the get-go, which is fantastic, which is why we are here today talking about the pilot, and ensure that their views are represented again at every point of that journey. Because by getting them involved in the design of the programme, we are ensuring that we have transparency, that we're breaking down those borders, and we're exploring what's going right, what's going wrong as well, and having that honest debate. And if we can get all of that right, and if we can ensure access as well, ensure that every part of society has access to whole genome sequencing, and that experience is a good one, then they are positive. So yeah, as a panel, we were kind of delighted to hear that they'd picked out all these topics and issues that were very close to our heart too.
Absolutely. And I mean, going back to this issue of kind of motivation or positivity around this, I think, one of the, in making comparisons, say, with the 100,000 Genomes project of which you're a participant, and this potential scheme where we are asking families whose babies are perhaps otherwise healthy to have their DNA sequence in order to potentially find something wrong with them. Those potential ways into wanting to be involved in a project like this perhaps do vary between, healthy, for want of a better word, and patients like the participants in the 100,000 Genomes Project that have a kind of a reason, a condition, an illness within the family to take part. Is there a big difference between, you think, the motivations of participants and the general public? Does that?
Absolutely. We as participants joined the project because we either have or a suspected rare disease, or there was a cancer, and we wanted to kind of explore the genomics of that. The dialogue looked mainly at the public who had a wider experience of the NHS and perhaps didn't have that experience of living with a long-term condition. However, I think if you look at both kind of sets of groups, kind of participants and also the public, we all identified these universal truths or universal principles, really. And that's where we kind of share that common ground and alignment that, no matter what experience you have, we all, as patients and participants and public, want to be involved in the process. And they called for co-production. And again, it's great that here we are at the Vision launch, and that's exactly what we are doing as part of the pilot. We are co-producing this, and we are ensuring that we are listening, actively listening to patients and getting it right. So yes, there are, as you say, very different routes into kind of genomic testing, but we all want a good experience. We all want to ensure that everybody has access to this, and we all want to ensure that this is a successful program that delivers results for newborn babies and research going forward.
Thanks very much, Rebecca. If there's no other comments from there, we'll move on to Alice now. So for the past few months, Alice has led the team at Genomics England who are exploring the potential of the newborn sequencing initiative and who have been setting up programs of engagement and dialogue with key stakeholders from across the health and science community. Alice, how has the kind of the things that we've just been talking about now, the dialogue, all of the other initiatives, how have they kind of helped shape the work that you've done so far in developing a vision for the Newborns Initiative?
So I think for me it was a fantastic opportunity because I came into this role, I think it was around May time, just around the launch of the public dialogue, and it was brilliant to come off the back of a piece of work like that and really have that at the heart and the start of this journey. I think what came out strongly to me was quite similar to sort of what Rebecca was saying around that actually we need to take our time on this and think very carefully about how we do it, but we also need to make sure we're listening to all the different communities involved in this and engaging with them in the right way. And I think really, for every aspect of a programme, as you start to think about what a programme could look like in this space, you need to be thinking about it in terms of the different frames of both the scientific aspects, the clinical aspects, but also the ethical aspects, and also what the experiences are of the families and the sort of people around that will be going through the journey in this space. And so really to sort of draw on those kind of areas, we've built a team really within Genomics England where we try to bring in that expertise within our team, so that's kind of very central to how we're approaching this. But also one of the things we felt really important to start was to have a really strong steering group supporting this. And I know we've got Sarah Jane who will talk a little bit more about this later, but I'll put our hands up. It is a very large steering group, but one of the reasons for that is because there are so many different communities involved in this. So actually, we felt it was really important to have those voices heard and actually have that representation. And that steering group really will be supporting our team over the coming months and weeks to really start thinking about the shape of what the programme could look like and make sure we're asking the right questions. I think the other thing for us, it felt like it was very important, was to really, we've had the really strong signals from the public dialogue to start thinking about kind of their kind of excitement around this and where the potential is and their concerns, but actually to also really talk to all those different other communities. So we set out on doing a series of what we called expert stakeholder interviews, where we tried to pick people from the different groups that be involved across the pathway. So different healthcare professionals, people from rare disease charities, people with ethics backgrounds, really to kind of get their views and thoughts on actually both what the potential of what we could do in this space are, but also really what the kind of concerns and challenges are, and really start to think about some of the really big questions we need to be thinking about and asking. And I think, it's very much the start of the journey. These are only early insights, but it's given us a really good start to start really starting to think about the shape and feel of what a kind of pilot can look like in this space, and really thinking about kind of the vision of what we want to try and achieve over the coming months and years.
Thanks very much, Alex. So we've heard some of the kind of background to this initiative, and as I say, the team led by Alice has been kind of trying to flesh out some of that kind of fact-finding that we've been doing and the kind of engagement and dialogue that's happened. So we're kind of now in a position that we're able to kind of set out a kind of initial vision for how we want to take forward this work. Can I turn now to you, Rich, to kind of give us an overview of that vision that we've been developing?
Of course. Thank you, Simon. And really, I think the point to start on, which I think we've heard particularly from Rebecca, is about, you know, we're very much in a conversation here and at the beginning of a process. And we're very lucky to have had the chance to spend a good amount of time already listening in dialogue, the formal public dialogue that we launched this summer, and increasingly now engaging not just with the public, but increasingly, as Alice has said, with experts across the different areas and a very broad range of areas. And I think we're also lucky to have that time. Nina said earlier, the UK is in an unusual position here. I think part of that is that we've got the buy-in to take our time. And I think one of the very clear messages that we've had from the public dialogue, but throughout, is this is enormously exciting. It's important work, but do this right. Take your time. Don't rush it. And I think that's a really important message, and it runs through what we're doing. We're, just trying to advance my slides here. I may ask for the team to help. We've drawn out a representation of the process we'll go through. I don't think I'll call it a timeline even, because what's important is doing this in the right order and with the right steps. We're right at the beginning, and what we've been doing, and we'll hear more from Sarah Jane about the NHS steering group that has helped us, together with the public dialogue, develop a shared vision with the NHS on what the future might hold, what a potential research pilot could look like. But it's very much at the beginning. This is continually listening and developing and iterating that vision. We hope to publish that vision in the next few days so that people will be able to see that, be able to respond to it. And we've got various aspects of formal dialogue with people, but we really want to hear people's views broadly across the piece. So we're there at the beginning of this process. It's about not just dialogue, but also throughout the process, what's very clear, as Catherine said, we heard from the Task and Finish group that was set up in response to the Chief Medical Officer's request. This is about developing evidence, not making assumptions that you might be quite confident of if you have a particular opinion, but actually developing an evidence base that can inform in a stepwise manner, first of all, how you might deliver a research program, but secondly, then that it's a research program to develop evidence that can inform future decisions, whether or not this is something that should be adopted in mainstream care, should be a long-term service. And so it's putting in those steps and those processes and not assuming that you'll pass through. So what are the areas that we're exploring in the vision and we're anticipating exploring in this programme? So I think really there are three broad areas that we're looking at where we can see the potential value for whole genome sequencing sat alongside the conventional heel prick test to allow early diagnosis of treatable disorders, the sorts of conditions that are currently included in the heel prick test, but unfortunately one doesn't have the wherewithal with the technology before whole genome sequencing to detect them. So those sorts of conditions where, if detected early, there is treatment that could have a major impact on outcomes, and the sorts of things that would cause problems early in childhood. So that's the first area, and that requires the generation of evidence in terms of what that, not just what the potential impact is, but we need to know what the real impact would be, not just in terms of benefit to patients, also in terms of the resources required in the health service, the training required for different players in the health service, from midwives through to genetic counsellors and through to treating teams. What information you would absolutely expect if you were one of these parents receiving a result, the clarity of communication, how that would be delivered, so the feasibility as well as just the evidence. So that's sort of area one that we're exploring. The second area we're exploring is the potential, with the consent of families and participants in the programme, to develop, to explore the research benefits. For example, not just taking the benefits that we have today in terms of the knowledge of, for example, a particular condition that one might diagnose with a genome, allowing you to use a treatment we already have evidence for, but actually creating a platform where we can make improvements on the treatments, the diagnoses that we can make, and providing that platform so that we don't just stand still, but we improve with time, not just for the children of tomorrow, but also people who've joined our programs already. And then there's a third area that we will be exploring through this, which is the potential and the value and the ways in which we could use a genome and genomic data as a resource throughout the lifetime. And I think this is a really interesting and powerful concept. It's also one, I think, that we will take our most time to develop our view on how we do this. There are many questions that we'll be uncovering. So I think what we'd anticipate is that we'll be tackling these different areas at different paces through a programme. We'll probably find that that concept of the genome as a resource to be revisited is the one that we take slowest. And a lot of that is engaging in dialogue with people. As Catherine said, these aren't new questions. They're not things that people haven't thought of before. But that doesn't mean we know exactly what the right answers are either. And I think we have to be honest about that. But there is immediately, one can see the potential value of a baby, perhaps who's born, is well when they're first born. They don't have any of the maybe few hundred conditions that we know you can respond to, you can treat initially. But 6-9 months down the line, they get unwell. Maybe they end up in intensive care. Well, if their genome's there and available for analysis, then looking there for answers that they need to guide their care becomes something that I think we'll be relatively comfortable with. But there are many other questions that we'd want to explore. I think just to emphasize again on the sort of the feedback and those treatable conditions, as Simon, you said at the beginning, this is talking about this in a postnatal context after babies are born. We're not talking about antenatal care or prenatal care. We've already touched on, others have already, about some of the challenges. There are many, many questions here, and that is why we need to take our time as we go through. And today is too short to list out all of the areas we've already been talking about as an NHS steering group, but also more broadly. But just to sort of give a flavour and to touch on some of them, I think is useful. For example, we might say, well, we'll look at conditions initially that are like those that are returned on the current heel prick. We need to work through in real detail what like those conditions means in the context of whole genome sequencing. What are the thresholds for feeding those findings back to families? What do families expect? And so forth. So developing and evolving that approach is really important. I think one of the key things we've been talking about today, and we've been talking to people on the steering group and elsewhere a lot about, is capacity in the NHS. The NHS right now is stretched. The NHS is always stretched, and we need to be aware of that and ensure that part of what we learn through a pilot is how we work within the NHS, and what we need to do to expand capacity where we need to, and to make it part of sort of routine care so it doesn't create additional work where we can avoid that. And also what resources we need, not just for healthcare professionals, but for families as they go through this process at the beginning and throughout. And I guess it's worth also just being clear on what sort of program we're talking about here. As I say, this is, our view is still forming. We're in really active discussion, and there are lots of views on this. I think our experience in this country through the 100,000 Genomes Project and through the establishment of the NHS Genomic Medicine Service has given us a really good idea of the sorts of approach we could take. And our current view is that this would be a research program. As we've said, this is a research question at the moment. We don't have answers. This is research. So it would be performed under a research ethics framework. Families would go through a formal consent process, so they would choose to join it under that, through that process. But it needs to be embedded in the NHS for us to, first of all, learn what we need to learn, but also so that we begin to build some of the know-how and the future infrastructure. If the evidence tells us that this is something that should continue, it will allow us to embed that infrastructure. It's very much like the approach that was taken in the 100,000 Genomes Project. So you both learn from the NHS experience, but you also, through the process, help the system to learn and adapt as well.
Yeah. Yeah.
And I guess just a couple of things to sort of highlight on perhaps what this program is and isn't, and what we do and don't expect to do. We don't know yet how big a research program would need to be to answer all of these questions, but we know it may need to be up to 200,000 babies or families who join the program to give us the sort of weight of evidence. That's something that we'll learn as we go through the process, how many families we need to be confident of the findings. And it's very much about basing that in terms of the evidence that would be needed by the NHS, by the screening committee, by others to make the right decisions on the basis of the evidence. And I think just a couple of points to emphasise, as well as the point about this being a postnatal programme. We're not anticipating feeding back findings that aren't relevant in early childhood, certainly not findings that would be relevant in later life, for example. We often get asked whether we'd be feeding back things that might help understand whether people might be at risk of Alzheimer's, etc. Certainly not. And I think the key thing here, and it sort of goes back to the discussions we've been having earlier, this is about both being transparent as a programme and transparent with every individual involved. So this is a dialogue in terms of how we're developing a future programme, but also with all of the individuals involved and the families doing this with their information, with their buy-in, with their consent and with their guidance. So hopefully that has given you the
very much, Rich, and I think in particular that kind of question about what this is. You know, we've been using the word project, we've been using the word initiative, we've been using the, various different ways of describing this. It's a research project, but it's not just a research project. There are elements of kind of clinical development, but it's not just about that. And there are certainly elements potentially of NHS transformation, but it's not just that as well. A kind of hybrid of all three.
Absolutely.
Excellent. Good. Which kind of brings me on to the kind of the final, last but by no means least of our speakers. Thank you for bearing with us over in our Birmingham studio, Sarah Jane. Sarah Jane Marsh is the Chief Executive of Birmingham Women's and Children's Hospital NHS Foundation Trust. I don't know how she fits all of that on her business card, but she's also the chair of the NHS's Maternity and Young People's Transformation Programme, and she has very, very kindly agreed on behalf of Genomics England and NHS England to chair the steering group that Alice mentioned first in the project. I've been to one of your steering group meetings, Sarah Jane, and I've been bowled over already by the kind of energy and enthusiasm you bring to all of this, and trying to kind of herd all of these cats, for want of a better phrase, into kind of, you know, a way forward for the Genomics England programme. You haven't got a background in genomics. What was it that interested you in wanting to take part in this? How did we manage to twist your arm?
Thanks, Simon, and thank you. I've really enjoyed the session so far, and I've been learning along the way again. So brilliant contributions from everybody. I'm really passionate about the role that science can play in the future of healthcare. I think sometimes it can feel a little bit over there to people working in frontline services, but actually I think science could answer some of the challenges that we've got. We've already seen, as other speakers have mentioned, the impact that personalised medicine can have. So for me, you know, organisations like the one in which I work need to really grasp this and see it as, you know, something that we need to be very, very much involved with going forwards. It's really important for everybody right across the NHS to understand this and think through the practical implications. And, you know, I really want to bring that as well. There's no point having the best strategy in the world if you can't lift it off the paper and put it into operational reality. And then, if I'm honest, Simon, I think one of the other things was the people. I always, when anybody ever asks me to do anything, the answer really relates to the people that you meet and the sort of energy, enthusiasm that they bring. And whilst this is obviously a massively complex challenge on all fronts, when I met the people involved in it, I just thought, actually, you know, we've got shared values, we've got shared ways of working. We're going to listen to everybody along the way, you know, patients, families, citizens, clinicians, managers, whoever they might be. And yeah, that was what made me think, right, this can work. And then, of course, the lovely Sue Hill, who I worked with last year at NHS Test and Trace, wouldn't have let me say no, even if that had have been the answer anyway. So she did the final arm twist at the end. But no, I'm delighted to be involved. And yeah, just like everybody else, just hugely passionate about what we can achieve.
Thanks, Sarah. And yeah, I know what you mean about Professor Hill. It's very hard to say no to, isn't it? So what's the role of the steering group then? What do you provide to this programme?
I think there's a few things. I think the first is some structure around the conversations that we need to have. Others have alluded to the complexity here, but in some ways we need to try to remove some of that complexity and put some structure and some small decisions that we can keep making to move forward. So, as Rich said, we don't know the answers to all the questions, but we have managed to already redefine what some of those questions might be and narrow the scope of them, so that we're able to hold some structure and process around that. There are clearly huge ethical debates here, but there are major practical ones as well. And I think the steering group has got a range of people on it who are able to bring those different perspectives. So we've got people who absolutely understand the science and people who in the first meeting said, I don't really know what's going on here, but I know I'll have to live with, whatever the consequences are. So we had to go off and have a teaching session with Rich so we could actually learn what this was all about. And I think that's really a very strong thing to have. We don't want this to hit operational reality, a few weeks before we actually want to do it. So we've got that whole spectrum of people involved. I think I also see myself, and I know there's many other mechanisms in there as well, but just safeguarding the role of the patient and the citizen and the professional groups as well, and just making sure that there's a final check really that everybody has had an opportunity to have their say and be heard. That doesn't mean that we can always do everything that everybody says. The journey on the vision document has been interesting because there are some people who've got quite different views. I think that's very healthy. I see my job as, making sure all those views are heard and we come up with a fair way forward that people are comfortable with and feel able to support and put their name to.
Smashing. And both from your experience in your own hospital and also through the transformation programs that you chair, what do you see as being the kind of main potential benefits to maternity services from doing something like this?
Yes, so in my time, certainly in the Children's Hospital, we've got a major rare diseases centre in Birmingham. And I've met with many families who for years and years have been trying to get answers to questions about their child and family health. This obviously has a big impact on outcomes, but it also has a big impact on childhood. Children cannot be children. because they're constantly attending appointments here, there and everywhere, because we can't get a diagnosis for the condition, we can't get answers. And often by the time that children arrive in a place that might be able to help them, it can be too late, we can be far too far down the journey. So for me, one of the biggest potential impacts would be on the experience of children and families themselves. I absolutely believe in, that we need to put children at the heart of healthcare. It's a speech I often make, but, children aren't a specialty. They're a population group that we have to look after well if we believe in the future prosperity of this country. They're our opportunity to break cycles of ill health and deprivation and inequality. So I think the potential to impact on children and young people at the earliest stage that we possibly can and make a difference, to me, is the real driving force. I think there are going to be impacts in maternity services. I think we learnt in the 100,000 genome that whilst we could get, great outcomes from what we were doing, the sun's just come out to tell me that that the children are shining and supportive. Whilst there is, it's a great thing to do, there are practical implications as well. Maternity services are under a lot of pressure. Those that work in, more generally in maternity will know about some of the safety concerns, and Donna Ockerton's reviewing into Shrewsbury and Telford and so forth. It's really important, again, that we understand how we're implementing this in a broader NHS context. So I think the benefits are, worth it. We need to work through all of that as part of the steering group, but we do need to make sure that we understand the way that work will change and the additional pressures that it might put some of our services under. Certainly in the short term, where we're running dual systems potentially. Hopefully over time, it might make processes and systems a lot clearer. And actually, it could make for a more efficient and effective maternity and childhood journey, which would obviously be our hope.
And Sarah, as your kind of NHS colleagues kind of hear about the fact that you're involved in this group and are perhaps wanting to ask questions about kind of the programme and how it might work, is the feeling one of nervousness, excitement? How are people kind of reacting to the setting up of the programme and the vision?
I think that people can see the challenges, particularly some of the ethical challenges and the complexity of the implementation. But I think that many of us who lead in health services can sometimes feel very sort of under pressure with the day-to-day operational realities in the NHS. We're under huge pressure at the moment, serving our populations, urgent and emergency care, elective backlog, which is really important and so forth. I think having these projects that give us hope that there may be a different way of doing things, that science will be able to unlock answers to really important questions, and that we'll learn things that we don't even intend to learn when we set out, which I'm sure will happen, as it did with the 100,000 genomes. It was about rare disease, but we started to learn things about common disease along the way. I think they're the sorts of things that get people excited. So a few people have said to me, how did you get that gig? But in general, it's real enthusiasm and people really wanting to get involved. And I think that's another important role for, the steering group, is to make sure that it's just not a few people over there doing this work, that we are having those conversations and we're talking about this in the mainstream of NHS conversation. And I'll certainly be out and about on the sort of NHS conference circuit, really just trying to talk about this project as a really important thing for the NHS as a whole going forwards.
Thank you very much, Sarah. That's really inspiring. Thank you. Lots of questions, so many questions. I'm going to use my chair's privilege to kind of ask a couple of questions that I know that when we go out and we do some engagement that lots of people talk about. I think the first one, going back to those kind of the main aims around the programme, the first one about kind of looking for specific conditions, specific genes, specific variants to kind of make diagnosis from children area. A lot of questions around kind of, how will you decide, what conditions that you're able to look for? That I think people have done work on looking at, maybe up to 600 different conditions that you may look for. Some people say, well, it might be a lot more, it might be a lot less. How is the decision going to be made around kind of what's on that list of things to do, Rich?
So I think that's as with everything in this program, that's something that we will want to share our thinking on. We're developing a view. In fact, we've set up a little group from the NHS steering group to begin to think how we might shape this. Again, back to the theme of we're not starting from nowhere here. These are things that people have thought about long and hard. For example, the National Screening Committee has obviously thought for a long time, and many people internationally, about some of the considerations here. But there are obviously also aspects that are unique to using whole genome and other sort of particular aspects of what we're exploring here. So there will be, what we're looking to do is to develop a structure, as Sarah Jane says, to our thinking, and we can agree on some core principles. There is, of course, a lot of devil in the detail, and one of the things that we absolutely commit to is that we will, to every condition, every gene, every patient pathway, we need to give a fine-tooth comb level of detail about thinking about every step in that process. At the same time, and that's all the way from, do you include a condition, or what do you need to do before you're ready to start saying you'll feed this information back to families, because you can't start before you're absolutely sure you're ready. But at the same level, a lot of the questions, it's about simplifying them, if you like, as Sarah Jane says, thinking what the core of the question is, not sort of hiding ourselves in scientific detail, actually saying what are the really important things that participants and experts would expect. For example, when should these findings be relevant to families? Who should we be thinking about the benefits accruing to? We're talking about the baby's genome guiding their own care, and also what level of confidence do people expect before we start returning anything to them? Those sorts of big questions, which then you can help inform the really detailed view, condition by condition, gene by gene, family by family.
Great, thank you very much. Another question, because I know that there are a lot of people from industry hopefully tuning in today. Alice, the kind of second aim, which is about this, I think Rich called it a kind of platform for innovation. How will we work with industry? How will Genomics England work with industry in terms of kind of, what it is that they have access to, how they will be able to kind of create that kind of acceleration in new treatments and drugs that might have a benefit?
Yeah, so I think, one of the things about that second aim is there's a huge amount of potential in that. What we really want to do is help drive research into new diagnostics and treatments. However, I think we're all aware, and it's come up in the conversations today, that there are, public concerns around that use of data, who accesses it and how they access it. And I think what we have is a really strong foundation at Genomics England that we can build on in terms of our experience of actually ensuring that we put the right levels of controls and governance around how people access that data. But we also need to really understand what that looks like in this setting, because there are some differences in terms of the data we might be collecting and the potential of what we might do with that. I think, again, one of the things we've felt was really important is to have that kind of transparency and openness around that discussion. And so one of the first things we've done is set up a discussion which is actually happening this week with people from public dialogue backgrounds. Rebecca's joining us as well with her perspective as a participant, and some members from industry. So we can start that conversation about thinking around how do we look at what the potential of using this data could be, but also what are the considerations around how do we ensure that we both retain that public trust and confidence in what we're doing, but also thinking about the types of things we might think about in terms of that governance and control. So really it's kind of the start of the conversation, like many of the things like we've said today of where we're at. We're starting a conversation. We don't have all the answers yet, but I think in this space we have some of the benefits of building on the foundations of some experiences we have as an organisation, which we can take forward as part of that conversation.
Yeah. And Rebecca, not to put you on the spot at all, but what is the solution? There is this, as Alice says, a kind of a wariness amongst the public about access to their data, particularly by commercial companies. But on the other hand, we know, through talking to the participant panel, that actually, if you have a rare condition, then that's exactly what you want. You want, people to be looking for solutions, cures, whatever that might be. How do you kind of find that middle road that keeps everyone?
I think the two key words are transparency and benefit. So, in terms of working with industry, people automatically get scared and worried, what are you going to do with my data? But by kind of being transparent, by ensuring that, patients and participants are in the room when there's discussions are happening, that is a good way of kind of breaking down those barriers and ensuring that transparency. And when we've done public dialogue before, people do understand that you need kind of commercial partners to look at your data to find results which are not only going to help you, but families and other generations, and they're supportive of that. So explain the benefit of that and explain the benefit for all as well. So I think it's about having an eye-to-eye level conversation with the public and not being scared, kind of throwing open the lab door and letting people in and letting them see what you do and see how you use our data. It really shouldn't be magic, because at the end of the day, that data is our data. It belongs to people. It will eventually belong to babies who will grow up to be adults. So, let people in and be transparent about that process.
And Nina, your experience with the work that you do in the neonatal sphere, you talk about the fact that actually families are very willing to share their data for research. What's the kind of secret to doing that, if it is a secret?
Well, no, I would echo absolutely what Rebecca's just said. It's about being transparent and open and clear about how the data will be used, what it's being used for, who makes those decisions and how those decisions are made. But it's also, so it's about trust in that process. It's also about benefit. Now, benefits can be individual benefits, or they could be broader societal benefits. And of course, one of the dialogues that is ongoing, whether it's whole genome sequencing and the benefits that arise from that, whether it's medicines development, whether it's vaccines development, it's also about how will the benefits that a pharmaceutical company, say, or a commercial company, what is the balance of benefit to them, to their bottom line and their shareholders, versus the benefits to public and society? And I think that, I know, because this is what families say to us, they want to be reassured that the benefits are equitable, that it isn't a polarized set of benefits, so that, yes, a company needs to sustain itself to do the development work, the research and development that it wants to do, but also the public needs to be reassured that those benefits are translating back to societal benefit. And we've seen some of these discussions played out during COVID, of course, with vaccine equity and inequity. So part of the dialogue will be ensuring that the benefits are equally realised for wide societal benefit.
Thank you very much, Nina. A question from the chat. This is from Sarah Hunt, and it's a question around how the programme will work with the UK National Screening Committee. And I think maybe, Catherine, to you first to kind of talk about. So the Screening Committee have a set of principles about which kind of things, screening programmes should be available on the NHS, and then maybe tell us about kind of how a relationship might work with the Screening Committee going forward. So these principles that the Screening Committee have perhaps might be different from the principles around gene selection for the programme and things like that. How do we navigate that kind of differences, and how does that work?
I think actually the principles are broadly aligned. Actually, it's just there's room for interpretation between them. So, broadly, it's about having, serious conditions. There has to be something you can do about it. Has to be a fair use of public money. There's certain principles. I think, there's broad agreement. And actually, when we did the public dialogue, sort of unprompted, really, they all came out with very similar ideas, and were sort of signed up to that, and keen for that to continue to be the criteria going forward. I think what's going to be really important with this research programme is the evidence that's going to come out of it. That's often the barrier to getting a screening programme into the NHS, because they have, rightly, I think, a very high bar. They need to be sure that this benefit-harm balancing act is going to work, come out the right way. The benefit is going to outweigh the harm. They need to be very sure about that, and to be sure about that, you need evidence. You need to know what happens to families, what happens to children who get this diagnosis. Is it better for them to find out early about something than just finding out when the condition, comes to bear? Yeah, exactly. Why not wait? If it's something that tends to happen at 6 months or a year or 2 years, what is the benefit of knowing at birth? So that's going to be really important and really interesting, actually, to see what comes out of this research programme.
And it seems to be that a lot of the discussions and perhaps a bit of the heat is around the definition of actionability. So actionability in terms of what we can do about it, perhaps means different things to different people. Perhaps.
It does, although I should say the screening committee already does have a very broad view of that. It's not that they're just, it has to be, a medicine or some kind of therapy. Some of the screening programmes already in the NHS, are more about, diet or sort of lower level, cheaper kind of interventions. So they're definitely very open to that. But it's about just showing that this kind of early diagnosis is better than waiting for a little bit longer.
Yeah, thank you, Catherine. And Alice, in terms of going forward, how will Genomics England be working with the screening committee?
So I think it was important for us to have a representation on the steering group from the screening committee for that reason. So Anne Mackey from the UK National Screening Committee sits on the steering group. But also I think, as Catherine said, a lot of it is about when we go into this programme, we need to think very carefully about both the evidence we need to gather, but the evaluation also that sits around perhaps some of the big aspects. So there's evidence around actually, how many of these conditions you can find and how you treat them, but also actually evaluating some of the outcomes and thinking perhaps longer term also some of the data we might need to collect. And I think that very much having that input and that dialogue with them, so we really shape that design the right way to give them the information they need to then inform those decisions down the line as to actually what a potential clinical service could look like and how that fits in.
Great. We have a limited amount of time left, and I'm going to try and plough through as many of these questions as possible. This one might be for you, Sarah Jane. Outside of the consent issues, this is from Megan. Outside of the consent issues, one of my concerns is the additional resource we are going to need. More staff in the genomics workforce, more counsellors, more nurses and midwives trained in this area, more money for the genomic laboratories to actually be able to stand up this service. This could mean a massive increase in samples without the technology and IT systems to support this, and we have to provide for patients with clinical need. How will this programme manage what is achievable? Now, this is the almost the age-old thing about NHS resource and NHS change and steering the turning the supertanker and things like that. What's the magic solution, Sarah Jane, to how we do this?
Does Megan work for Birmingham Women's and Children?
Yes,
that's my first response, because I think what Megan said is what staff up and people working in the NHS up and down the country will be thinking. I think that's one of the really important things that we need to lead through the steering group, is that starting off small and learning as we go about what resources do actually need to be in place to make this work. The more efficient and effective that we can make it, the better. But we will need to invest in the services if we, and we need to make the case for that. And I think that's part of the work of the group. Now, I think some of the excitement that we've got so far comes from the fact that we would be potentially changing pathways of care. So, not all of it is going to be additionality in the end, but clearly there is going to be a period of time where we've got double running and, and so forth. So part of my role and part of the role of many of the people embedded in the NHS on the steering group will be to be honest about what those resources are, because if we're going to make the impact, we know they need to be in place. And I think that's, it was probably quite a brave decision to ask someone like me to do that, knowing that I would keep repeating this. You've got to give us the tools to be successful if we're going to do this well. So Megan, I will have your question and challenge on my mind, I think, in every meeting, every step of the way. To me, it's the crucial question.
Megan, we'll get your question framed and put above the in the boardroom. Okay. There is another question that I'm kind of looking at Catherine here, but it's about ethics. But obviously, you know, just because you've got ethics in your title doesn't mean you don't hold the card on this. What would the ethical impact be of finding an illness that would affect someone as an adult and not pass that on to the family, as it would not affect the newborn in their early years? Is it ethical to not provide that information as and when it is tested for? Could it be too late to find out as an adult and then act upon that information? Are we then restricting the choices they have for treatment? How do we deal with that? That was from Katie. Thank you, Katie.
That's a really good question, and I think brings in an important aspect that we've not really talked about today, actually, which is when you find out genetic information about a child, you also find out about their family. So yes, there's the question that was asked about whether, you know, that would have an impact on that child, but what about their family? If it's an inherited condition, it's possible that other adults in their family have that condition. What do you do? And there have been, you know, some discussions that I've been involved in at the screening committee of thinking about the established approach is you think about the individual as the beneficiary of screening, you know, whatever stage in life that is. So at this point we're talking about the baby, the child. Do we need to start thinking about the family as the beneficiary of screening? And I think that's quite a new question and quite a difficult question. So I haven't got any answers, I'm afraid, but just a really important question to flag.
Yeah. Rich, you're frantically signalling.
I think one other important way to sort of think about what the process will be is that the reality of what looking at genome data is, and therefore what, in inverted commas, answers one gets from it. So the genome is 3 billion letters, bases of DNA long. And the reality, just as when we do a newborn heel prick, a complex test is done and very specific analysis is pinpointed on the particular conditions we look for. The reality of a genome is give me a printout of my genome and I don't discover my future. I actually need to do a very specific analysis to ask some questions. And so rather than suddenly everyone knowing what one can interpret from the genome, actually we have the power to decide what questions we're asking at what stage. And so almost we have potential to do some things that might be relevant later in life, but we also have the power to decide together what people want to do and what people want to ask of their genome, if you like. So it's less, sometimes we feel like we're the victims of too much information. Actually, the reality is we're empowered to choose what we want to find out.
Yeah, smashing. Well, on that note, I'm afraid that we've run out of time. Thank you so much. It's a fascinating discussion, and I feel really privileged to be kind of working in this area. There is a webpage that we've just published on our website, which has got further information about the programme. As Rich said earlier, we're hoping to publish a vision document that we'd like people to have a read of and send us their comments on. There will be lots of opportunities over the next 18 months or so before we even begin a pilot to contribute, to come to events like this one, to feed in your views. And as I say, the engagement programme that we're standing up is an opportunity for us to do that listening and to do that talking. So it only remains for me to thank Rich and Alice from Genomics England. To a certain extent, they get paid to be here, that's all right. These other people have all given their time for free, and we're very, very grateful for it. To Sarah Jane Marsh over in Birmingham, thank you, Sarah Jane. To Rebecca, to Catherine, and to Nina, to Professor Modi, thank you all very much.
Good afternoon and welcome to the Cancer 2.0 session. We're really excited. We've got lots of people we want to introduce you to today, experts in their field, and we've got a lot that we want to tell you about what GEL is up to within this field. My name is Emma McCargo. I am the Cancer Programme Lead for Genomics England, and I want to introduce you to Parker Moss. He's our commercial officer, chief commercial officer, I should say, and our research tribe lead. Parker.
Thank you very much, Emma. So the first thing I want to say about cancer is how incredibly proud and excited we are to be collaborating with the NHS and the Genomic Medicine Service that the NHS have rolled out across the country. So for those of you who didn't hear what Dame Sue Hill said earlier about what we're doing in cancer today, it really is world leading. So for the last several months, we have been sequencing, whole genome sequencing across the country, all children with pediatric cancers, all sarcoma patients, and all acute leukemia patients. And we've just had approved and will be introducing in the coming months whole genome sequencing of triple-negative breast cancer patients, of ovarian cancer patients, high-grade serous, of the other hematological cancers. We'll be extending children to include young people, and we're going to be sequencing pan-tumor across metastatic and refractory conditions that are not well managed by standard of care. So this really is a world first. It's not just the number of indications that we're covering, but it's also that the NHS have managed to successfully set up the infrastructure, the clinical governance, the clinical buy-in, and the interpretation capability to roll this out at a national scale. And it's a tremendous thing for patients with cancer in the UK, so that's absolutely thrilling.
So, Parker, I wondered if you could tell a little bit more about what our vision is from a clinical, but also from a research point of view.
Yes, okay, so I'll start clinically. So cancer has been called the emperor of the maladies, and the reason for that is that it is an exquisitely complex disease. So cancer is multigenic, it's heterogeneous, it's metastatic. Cancer interacts with the germline, but it also interacts with the immunome, and increasingly it appears that it's interacting with the microbiome. So it really is a complicated disease to diagnose, to prognose, and to plan treatment for. And so what we want to make sure for the cancer patients that receive whole genomes is that we're taking the most comprehensive view of the genome to make the right treatment decision for them. And it's not just about the detail, it's also about a different way of looking at cancer. So because of this complexity that I laid out, it's very difficult for clinicians to be hypothesis-led, to start from the phenotype and make their treatment decision or their treatment planning from that basis. So we are engineering a system in collaboration with the NHS which is hypothesis-free. We're really unlocking the genome and allowing the genome to tell us the story about the patient's cancer. And we think that's going to introduce really exciting clinical trial opportunities for patients, particularly in a recurrent or a refractory setting. It's going to give us biomarkers that we can follow to track minimal residual disease along their journey post-remission. And there really will be a growing body of evidence that patients are going to benefit clinically. But getting the clinical service right is not just about accuracy and consistency. It's also about getting the operational details right. So when the first genome was sequenced at the beginning of this millennium, it cost over $1 billion, and famously it took many years to sequence that patient. Now sequencing can happen in less than a day, but the sequencing is just a part of the story. The whole end-to-end journey involves taking tissue from the patient, extracting the DNA, sending that to a central sequencing lab, running it through our bioinformatic pipelines, through decision support platforms, back through genomic lab hubs who combine other modalities like immunohistochemistry and imaging, and then back to genomic tumor board. So it's a long end-to-end process that takes several weeks. And we know that patients in England deserve better than that, so we're working very hard looking at new technologies, and you'll hear an expert panel talking about nanopore sequencing, which has a different form factor that will allow sequencing to happen much closer to the patient in the hospital, and we hope that that will demonstrate turnaround time benefits, and that we can eventually get sequences to be turned around in a week or a little bit more than that.
Yeah, it's wonderful, isn't it, for the patient? And what's the view, and where are we going from a research point of view?
Yeah, so from a research point of view, this is where I have to say how grateful we are to all of our participants. So we've sequenced 17,000 or more cancer patients as part of the 100,000 Genome Project, and now we're prospectively sequencing patients in the NHS. And today, prospectively, over 95% of those participants consent to allow us to have access to their data for research. So that's a tremendous thing. And what does that mean? That means we collect all of their clinical data retrospectively and prospectively, and you're going to hear in our digital pathology panel that we're also going to be collecting their images, radiology and pathology images. We have their full text pathology and radiology reports, their cancer regimen data, and that's through partnership with NHS Digital and Public Health England. And we have the ability to not just rephenotype those patients, but also to recruit them into clinical trials and to resample them to get precious human tissue for these functional genomic pipelines that can try to convert our data on associations between genotype and disease to actual causal relationships. So it's a data set that is a very, very powerful resource. We put all of our data sets, all of our data into Amazon Web Services today, and that's fronted by a terrific bioinformatic platform, which is run by a company called LifeBit, which is a great British startup that just was tremendously successful in a fundraise a couple of weeks ago. And that creates a really nice research environment, which is extremely secure and trusted, that researchers from around the world can come and look at. So today we have 4,000 academics, and some of the best ones are in the room with me today. And we have 35 commercial partners, and of those commercial partners, we have 8 of the top 10 pharma companies in the world who are researching our data and who are learning how to build better drugs, better diagnostic tools, who are doing these preclinical studies to prove causality, which de-risks clinical drug discovery and clinical development pipelines, and we're building real-world evidence tools as well, so that we can really demonstrate that the effects that drugs are meant to generate in very carefully managed clinical trial settings actually have that same effectiveness in the real world. So some of the strategies that we're doing to improve the experience and improve the quality of data for our research community is, number one, we're opening up our research pipelines to researchers who are working in the machine learning space. And you're going to hear later from Serena some of the work she's doing in homologous recombination and mismatch repair to introduce the absolute cutting edge algorithms that can look for markers of genomic instability. And we'll be incorporating those in our clinically accredited NHS pipelines. So that's one strategy. And another thing we're doing is we're going to be adding digital images of pathology and radiology into our data sets so that researchers can look at not just the molecular, but also the spatial context of solid tumors. So I think we're creating a really powerful world-leading research resource as well.
Wonderful. That's wonderful. So maybe I would hand over to...
Yes, let's call on the first panel.
To the first panel. Yeah.
Thank you. So I have on the stage with me today some of the real experts in sequencing in the wet lab, and I'm going to ask both of them to introduce themselves. Andy, please.
Hi, I'm Andrew Beggs, and I'm a professor of cancer genetics and surgery at the University of Birmingham.
And Greg.
Yeah, my name is Greg Elgar. I am Director of Sequencing Research and Development at Genomics England.
Thank you. So guys, we have been working for many years very successfully with the gold standard short read sequencing, which produces very reproducible and exquisitely accurate results. But we're now beginning to look at long read sequencing, specifically with nanopore, so nanopore sequencing for long reads and methylation. And maybe I can ask both of you, I'll start with you, Andy, why are we doing that now? What are some of the benefits?
I see long read sequencing as an evolution on existing techniques. It's complementary, but an evolution. And it allows us to easily identify things that short reads may have struggled to do in the past. Things like structural variants, and NTRK fusion is an example of that on DNA, but also turnaround time, speed, ease of accessibility. And I think the final thing that's most important is methylation. And we now know from several Nature and Nature Medicine papers that methylation could be used as a biomarker for deciding, if, for instance, in brain cancer, what the subtype of brain cancer is, which has profound implications for treatment.
Thank you. Greg, same question to you.
Yeah, I guess I'd look at this from a more operational standpoint. So short read sequencing has been very much the state of the art for a number of years now. But long read sequencing has been at the fringe, and Oxford Nanopore have really led in the UK the development of instruments and the technology itself. And now is the time, I think, that they are starting to look as if they have clinical grade applications and clinical grade instruments that could be used in future healthcare. So I think it's a really good time now. I think we really have to jump on the back of that and push the technology forward and work with clinicians, with researchers. We need to bring academic researchers together with clinicians, and GEL is a great way to do that. Genomics England is good at facilitating that. So I think that's what we need to do. We need to do it now. We need to try and get it into the healthcare setting in the next year or two.
Thank you. And Greg, having had a quick look at the audience that are joining this call, there are quite a lot of clinical scientists and technical people in the room. So maybe you could just go into a little bit more detail. What has changed now in terms of quality and throughput that makes nanopore sequencing kind of ready for prime time?
Yeah, I mean, there are a number of things we have to consider. First of all, the economics of it. So long read sequencing has been very expensive. Nanopore have driven down the cost, especially at scale, and I think it's now competitive with short read sequencing, and we mustn't forget that that's a serious consideration. But at the same time, they have made technological advances that have made the sequencing more accurate. They have enabled us to sequence a whole genome on one of their flow cells, so the capacity of the sequencing has increased. And they've introduced instruments in the last year or two that can really match any of the short read instruments in terms of scale and capacity. So the current instrument that we're using, the PromethION, in our lab, can sequence 48 genomes simultaneously, and we can get really good data from that machine in about 24 hours for those 48 genomes. And that's really up there with the capacity of other technologies.
Okay, so I think it's just such an exciting opportunity to introduce this complementary technology into the lab and ultimately into the clinic. And so maybe, Greg, you can tell me, Andy, you can tell me a few kind of patient examples of where you feel this might make the difference to patients in the clinic.
I think before just the patients, the speed of being able to get the data to a patient is fantastically important with long read sequencing technologies. So an example is in brain cancer. It's very important to be able to get a rapid clinical diagnosis, both in adults and children. And one of the advantages of long read sequencing, you can provide the structural variants, the copy number variants, the methylation, most importantly, to accurately subtype a patient's tumour and take it forward to a diagnosis so you can pick the right treatment. Another example is sarcoma and hematological malignancies, but also it can be pan-cancer. So, for instance, NTRK fusions, larotrectinib has been licensed now for NTRK fusions across the whole spectrum of cancers. And one of the advantages of long read sequencing is we don't need to turn to RNA and RNA probe bases. We can go straight from DNA using long reads. And that means you can wrap everything up into a very neat package with a single assay.
Okay, I mean, that's extraordinarily exciting. And maybe as you run a lab, as you both run labs actually yourself, you can tell us a little bit about the footprint and the form factor of these new devices and how that might change things operationally.
I mean, one of the great things about the long read sequencing, the nanopore devices exist, they're very portable. You have the smallest one, the MinION, which is about the size of a big USB stick, but the PromethION, which is what we both use for clinical grade type sequencing, I have literally picked it up and put it under my arm, and it is durable and robust, and it makes it a lot easier to take this type of sequencing to the patient, which in the past the sequencing has had to go to the machine rather than be a bit nearer to the patient, and that allows us to speed it up and really democratise it, widen access and participation for all patients.
Yeah. Greg, we're in the middle of a kind of validation program in which you're testing against gold standard. Perhaps you could explain a little bit about that program and some of the challenges that we're facing operationally and technically and with the infrastructure to make this live and real.
Yeah, absolutely. So the backbone of a pipeline is similar to that of short read sequencing, but the challenges are more significant in certain areas. So the extraction of DNA from high quality tissue presents two problems. One is getting the high quality tissue in the first place, and we need to work very closely with surgeons, pathologists, cancer clinicians in order to expedite the movement of that tissue to a central lab where the DNA can be extracted, or it needs to be done locally to a high quality level. And once that's moved into the lab, the sequencing part now actually has become pretty straightforward. The machines are more reliable and more robust, and we have a pretty low failure rate if we get reasonable quality DNA in the lab for long read sequencing now, and that's really important. Then there are also some challenges in terms of interpretation, because I think certainly at the outset we would probably be looking at using short read sequencing and long read sequencing alongside each other, because they both have their strengths and weaknesses, and so it really enriches the data that you can get from a whole genome when you use long and short read sequencing together. But we haven't really worked out the best way of actually merging those two sets of data. And there are various ways we can do it. And then leading on from that, we then need to have clear interpretation that we can then hand back to clinicians. And that's also an area where we have to really work things out. So we're going to get long read data, short read data, methylation data, and we need to put that together so it doesn't conflict, so it actually supports each other, because they are in a way they're validating technologies. So long read sequencing and short read sequencing are done completely differently, are using completely different methodology and technology. And so if you find a mutation in both, then you've immediately validated, or each validates the other. So there's some real advantages, but we need to work that out. It's also a really rapidly evolving technology still. And so in order to stick to a specific end-to-end pipeline, we need to make really important decisions about what we take on board and what we leave behind.
Yes, this is a real challenge, isn't it? Of course, in the scientific method, you want a stable baseline, but the baseline is shifting every month. Right. There's the quality of pores and the chemistry, but also the quality of the machine learning algorithms for base calling improve. And so that must be a very challenging thing to do. And we've talked a lot about the long read, but you've both mentioned methylation. Andy, maybe you can tell me a little bit more about the clinical application of methylation and whether you really think that nanopore sequencing could be a substitute to orthogonal methylation testing.
So my laboratory's undertaken quite a lot of methylation-based testing recently using nanopore, and to be honest, it's a killer application for the technology. It beats everything currently going, all the old techniques using short read sequencing and other technologies like pyrosequencing. We see base accuracies comparable to whole genome bisulfite sequencing. And so the advantage of that is we can use that very rapidly and validate it very rapidly into clinical practice. Certainly it's transformational for us. I mean, one of the other exciting things is, for the technical minded on this discussion, is that we can phase methylation as well. So we can actually work out the maternal and paternal contributions within the cells and various things. And that is fantastically interesting. It's something we've never been able to do before. So it's unique to long reads, and it's mind-blowing in terms of what it can open up for us in understanding how these cancers work.
And in terms of this question, and for the less technically minded on the call, maybe both of you can explain how the principle of using a pore for sequencing differs from sequencing by synthesis and chopping up the genome, and why, in some ways, that how that affects our view of ground truth and the operations.
Well, it's very clever. It's basically a protein pore in a membrane, and you detect a voltage as the DNA strand goes through it for nanopore sequencing. And that has advantages and disadvantages. The advantages is you can stick anything through it, not just DNA. Theoretically, protein can go through it as well. And from the voltage change, you can understand whether a base has been methylated or unmethylated. With short read sequencing, it works by adding coloured nucleotides onto a flow cell that shine when the laser shone on them. And that is very good and very accurate, but it doesn't allow you to get access to things like methylation.
Yeah, fascinating.
Yeah, I mean, and then the real reason for that is you are sequencing the real genome with nanopore technology. You know, it's the DNA that's come out the cell is going straight through that pore and being read. Whereas with other technologies, quite often you're reading secondary copies of DNA. So you lose a lot of that really vital information that could be really key, for instance, methylation, but other modifications as well. DNA carries lots of other modifications besides just 4 bases. And potentially, if you're reading the native DNA molecule, you can read any of that information.
So isn't it exciting that we have these new players in this sequencing ecosystem, and not just on the infrastructure side, but also on the software side? I'm aware that we're evaluating various variant callers at the moment, and from what I understand, both of you are working closely with Google and their Pepper deep variant algorithm, which uses deep learning to actually take a visual impression of the genome. Who would have ever thought that Google would have been one of the leaders in variant calling in cancer sequencing? Maybe you can tell me a little bit about what you're doing with them, Andy.
Yeah, so it's an amazing approach. It completely turns on its head variant calling that we've seen in the past. In the past you use mathematical approaches to match the sequence you get off the sequencer to the reference human genome. But what Google have done is turned that on its head and turned it into a human method. Essentially, it looks at the genome and it puts it as a picture, and you see a map of both the genome and the sequence genome, and the machine learning algorithm works out where it should go, but like the human eye would. So it's completely different from any mathematical approach, but it has the advantage of being incredibly accurate. They've taken how a human would do it, sped it up, and used machine learning to leverage the accuracy. And so we've seen substantial increases in accuracy using the Pepper deep variant algorithm within our own laboratory, and I know that there's further to go with it.
Anything on variant calling or base calling?
Yeah, I would probably add that this is, again, a rapidly evolving field, and it is great to see completely different players using quite really novel approaches. And it's partly because of the way the signal comes off the machine. It's completely different to the somewhat synthetic approach of other methods. You know, it is the real raw signal, and that gives you the opportunity to play with it. And it also has given Genomics England the opportunity to really engage with top academic researchers as well, who are really keen to develop algorithms to read these sequence traces better. So it's a really exciting collaboration for Genomics England, for clinicians, for academics. It pulls everyone together, and I think that's, it really makes me working in GEL feel like a real enabler of this kind of work, and it's great fun.
So we have one question from the floor, and I think this question highlights how technical our audience is. So you can choose who takes this one. So here's the question. I thought methylation was stripped before the gametes were formed, so how can it be phased?
Good question.
So DNA is methylated all through our life. So yes, there is a stripping and methylation, but we methylate our DNA both naturally, and also we smoke cigarettes sometimes. That can sometimes add modifications to our DNA that we may or may not want. So our DNA is being modified all the time. And that's actually one of the key things in cancer, because in cancer, our methylation status changes. So we can actually classify, stratify different kinds of cancers by the different kind of methylation that you see within those cells. So every cell may contain differently methylated DNA. So that's true, but this is the exciting thing. We can literally look at the living representation of a cell's genome by looking at methylation. It's probably far more representative of some of the activity in the cell than just reading the bases themselves, which is the same in every cell.
Well, I think with the launch of the GRAIL methylation test, which has been rolled out to 140,000 patients, that's bisulfite methylation, I think we're going to be hearing a lot more about methylation in the coming years, pan-tumour.
Bisulfite methylation is pretty expensive. You get methylation for free with nanopore sequencing. So that's worth remembering.
Okay, so there's really, really a lot of promise out there. Yes. Good. Is there anything else that you would like to say about what is happening in your lab and what you're excited about beyond nanopore sequencing?
I think it's the ability to rapidly achieve patient benefit. I mean, short-read sequencing is great, but, there's always a but, it needs a bit of time to batch enough people together to get it going to make it cost-effective. With nanopore sequencing, we can turn genomes around really, really rapidly, 24 hours or less, depending on what the application is. Something like methylation, you don't need a high read-depth coverage, you just need enough to do the methylation call. And so by using this technology, we've been able to access samples much quicker and get much more information. The data is fantastically useful. I think the other thing that's most exciting about nanopore, it must be about the only technology in sequencing where when a better base caller comes along, you can reanalyze the data set you've got and get more accuracy and more insights. You know, retraining, like Greg talked about base modifications. One of the things we're working on at the moment is developing a better base modification caller. So we can access some of these base modifications that have been out range of conventional technologies like hydroxymethylation in the future. So I think this is a technology that's got a long way to go in that there's a lot of stuff we could do with it we haven't been able to do before, and that's very exciting.
Again, one last question from the floor, and I think that's all we're going to have time for. This is a great question. Something that we're not very involved with yet in Genomics England is cell-free circulating tumour DNA. And of course, it would be incredibly valuable to take a long read or even a short read whole genome sequence from circulating tumour DNA. But is that achievable? Are these short fragments of DNA sequenceable?
I'm going to give away the trade secrets now, but I suppose people won't mind. So one of the really exciting things about the latest iteration of nanopore technology is the library preparation kits. I'm sorry if the audience is going to get very technical, can accept a very low input, femtomolar inputs, which means when you've got short DNA fragments of sort of 100 to 150 base pair long, that is compatible with putting it straight into the kit. So it is possible to input ctDNA directly into a nanopore reaction. Now, I know sequencing, it does require optimization, obviously, and there's a long way to go, but the technology is expanding that, yes, we are actually, we've done it, getting ctDNA onto nanopore technology just by direct ligation. So yes is the answer.
Fantastic. Bring on femtomolar samples. Exciting. Okay, well, thank you very much to both of you. And can I please encourage the audience, you can see the quality of the answers you can get on technical, but also non-technical questions. So please do ask your questions. I've got these coming up live in front of me right now. And if you two would kindly leave the stage, I'm going to bring on my next panel. Welcome. I'm very proud to invite to the stage Darren Trina, who's one of the leading pathologists in the country and perhaps our very own pathologist from within Genomics England. Please can you introduce yourself a little bit more fully than that, Darren?
Thanks, Parker. Yes, I'm Darren Trainer, a pathologist in Leeds. I'm one of the doctors who diagnoses cancer in the lab. But my other job is to be the director of the digital pathology program called NPIC, National Pathology Imaging Cooperative.
And how many hospitals are in your NPIC?
It's pretty big, so the ambition is a national central system for digital pathology to which other scanners can be added at other sites. We have at least 20, possibly 30 or 40 hospitals by the end of the programme.
That's quite an achievement. Perhaps please introduce yourself.
Hi, so my name is Prabhu Armugam. I was a pathologist and I'm now one of the directors here at Genomics England, and I head up the multimodal imaging programme.
Perhaps you'll always be a pathologist. Darren, can we first just start clinically? You've been one of the leading lights, not just nationally, but globally in digital pathology. I think you introduced it in the UK far ahead of other UK hospitals, but also international hospitals. Perhaps you can tell us what digital pathology is doing for our patients and doing for the operational workings of cancer diagnosis in the clinic today.
Sure. So it's a really very simple technology. Digital pathology is a technology that has taken its time to come, and that's the reason being is because the images that we take down the microscope are enormous. So we pathologists have been working for decades using microscopes to look at patient samples on glass slides. And what the scanners that we use in digital pathology allow us to do is fully digitize that entire little piece of glass. So a single patient's sample might be maybe a centimetre or two in size on our glass slide, but when we scan it, it creates an image the size of a squash court. And that is a lot of data. That's the reason why digital pathology has taken a little bit of time to come to the mainstream. It has a variety of benefits of digitizing our samples in pathology labs. The most obvious ones are that we're no longer tied to that physical glass slide anymore. So we can review it in different rooms, in different hospitals, in different regions, or even different countries. And that really opens up lots of new ways for us to work in pathology. We can get second opinions more rapidly. We're not relying on samples to be sent in the post to other laboratories for their opinions. We can collaborate more readily with larger groups of people. And then in this era of artificial intelligence, having the images digitized means that our images are available for image analysis for using AI, which is a very exciting new area for us.
So we're going to come on to AI, but I just want to go, I've done a tour of your lab, which is really inspiring. And when you mentioned an image the size of a squash court, you weren't actually being metaphorical. Can you describe for us what you've built and how that helps you in an MDT session?
Yeah, so because our microscope images are taken at such high resolution, the typical scanner that you might have in an office is about 1,000 dots per inch to generate an image. The scanners we use in our pathology labs are 200,000 dots per inch, much, much higher resolution. So as a result, each image file from each patient is very large. And some of the early work that we did at Leeds was to evaluate, well, if you're looking at such a big image, what's the best way to do that? Because it's a hard thing to do with the computer. So we eventually decided that what we needed was very large displays with many megapixels or tens of megapixels, initially experimenting with a wall of displays which had 50 megapixels in size, and we found that was a really good way of interacting with the images. As time has gone on and computers have got faster and displays have got better, we found that it's very possible to essentially replicate the microscope diagnosis on a desktop display, which is a moderately high resolution today.
That's really impressive to see. So you've been speaking about really the clinical implications of digital pathology, but perhaps what we're working on in Genomics England is the research potential of not just digital pathology itself, but combining that with the molecular modalities and also the clinical data. Maybe you could just describe the program that you're responsible for.
Yeah, okay. So we're incredibly privileged at Genomics England because our consent is quite broad, so our participants are very generous in terms of the fact that we're allowed to access not only the data from their genome, but we can also access clinical data about those participants and also now the images. So what we're trying to do is link all these genomes, the images, and effectively the digital pathology and radiology for all of these genomes. The focus for us is cancer, so we're working on the idea that there's about 15,500 what we call solid tumours. So we're not really looking at hematological or blood tumours, and we're trying to digitise all the images that we have for all of those participants. So at the moment, that is very much a logistical process. So we're going back to each NHS laboratory, asking for the physical glass slides and then sending them up to Darren and his team in Leeds, who are digitising those images for us. And then we're also doing the same with each of the NHS sites to retrieve the radiology image as well. So the first part of our project is effectively creating a very large data asset or a library.
And before you go on to the second part of the project, can you be clear, are we just collecting the index slide, or what are we collecting in the pathology setting?
Yeah, so the 100,000 project was slightly complex in terms of we created a tissue pathway. So we had the fresh frozen section, which we call the index slide. And then that participant would then go on and have what we call the standard of care. So they would have their full specimen resected and excised and create FFP blocks. So we're actually going to retrieve the index slide and the whole excision specimen. So for instance, in the colorectal tumour, we'll have probably about 15 or 20 glass slides per participant for that excision, and we're going to do that for all of our participants. So the library and the image library that we'll have potentially could be one of the largest libraries in the world of linked images to genomic data.
And clinical data. So we will really understand the full spatial context of the tumour.
That would be the aim, Parker. I think the problem is obviously at the moment no one's really had this amount of data, and that's always been the challenge, is actually having access to this kind of data. And I think because of the privileged position that we have, we're able to do that. So we can provide researchers access to images, to genomes, to the data to actually try and work out what these changes mean, what do these mutations that we've seen in the genomes that we've got a sense and an understanding from, what does that translate to? So can we really see, an ER change from a kind of a genome? Can we see that, an H&E stain on a glass slide? And can we see more? What can we understand from, survival, drug discovery? And I think that's where we need to go to next, really.
Okay, and then the second part of the program, which is very much about bringing machine learning tools to the image, what are we doing there?
So Genomics England at the moment have what we call a trusted research environment, or TRE, and at the moment we keep all of our genomes and our clinical data within that, and we invite users to kind of what we call a reading library. So you enter our research environment and you review the genomes and the clinical data, and we will also add in the images into that research environment. But what we'd also like to do is adding tools to try and interpret the images alongside the genome. So what we'd like to do is provide researchers access to, say, 2,500 breast genomes, their comparative excision specimens, so that could be 10, 15,000 images, and the tooling to try and understand where the tumour is, where the stroma is, where the kind of infiltrating cells are, what that means in terms of the mutational changes alongside those what we call morphological changes down an H&E or on an image.
And I know from conversations that both you and I have had with the machine learning community that it's increasingly possible to vectorize both the spatial and the molecular context into a single vector and run that through a neural network and really get an unsupervised view of the impact of those multimodal features on outcomes such as survival and prognosis in response to treatment. So I'd love to ask you about that,
Yeah.
Darren. What are you seeing out there?
Yeah, I mean, that's really exciting. I think the benefit of having the pathologist in the loop, so to speak, and having the images is that we can actually close that loop and make sense of it. So when you come up with predictions about survival or about maybe newly discovered subtypes of cancer, you can say, well, is there something to be seen when you go back to the images? We've seen early examples that are already in colorectal cancer, where one group has described a different prognostic subgroup within colorectal cancer, and then when they worked with the pathologist, they found some morphological features down the microscope or on a digital image that were associated with that. So it's a very exciting time. We can even start with simpler things though, so simply just confirming with an expert pathologist that this is cancer and that there may be some pre-invasive cancer alongside it. Cancers look different down the microscope, so when we take maybe 5 or 6 samples of a tumour, it's often surprising how different it can look from place to place, so we can inform a little bit of the understanding of the genomic profile that comes out of that. And then simple tools for calculating, how much DNA might I get out of this? Is this viable tumour? Is it necrotic tumour? Really simple basic things that we can provide with DigPath. And then, as perhaps I said, we then have this really unique, globally unique data set that allows us to build on that in all sorts of ways using AI in future.
So in such deep specialisms, there's a risk of people working in a siloed way. Do you think that having the data from the genome and the image integrated will actually help interworking between geneticists and pathologists?
Yeah, good point. Yeah, I think it's essential, really. And, when I think we're only just at the beginning of this field. It feels like there's a new crossover field that needs to be emerging. We have a fellow that's going to be working with Genomics England, Charlotte Jennings, and her initial work for us is just sort of discovering what work has been done in pathology genomic correlation in the past and where the opportunities might lie. So even mapping that out is an interesting task in its own right. I hope to see a generation of clinician scientists working in this area following that sort of pathway. So yes, I think communication, I think involving pathologists in these discoveries is very exciting as well. So it's really good to see for the first time in a long time pathology images in Nature Medicine papers on a regular basis now because of this amazing new technology and simple technology of digital pathology. Hopefully it will bring it will improve the quality of cancer research by having that additional multidisciplinary view on things.
And a question for both of you. I'm seeing more and more exciting papers in increasingly credible journals that suggest that you can identify genomic features, genomic phenotypes in tissue, like HER2, like macrosatellite instability, for example. So do you think that the pathologists are going to put the geneticists out of business one day?
That's often a question posed when AI is sort of showing these superhuman abilities. I think we take a more real-world, nuanced view on it. So some of those discoveries are really very exciting. Some of them are based on kind of hunches that good pathologists would have had already, that, as I said, tumours can look different from sample to sample. So some of them are confirming that we knew. Some of them are really exciting new findings that we would like to see verified in larger settings with more diverse data. Perhaps the short-term value of it will be in helping us to excitingly discover new subtypes, new prognostic subgroups within cancer, but also just simple workflow improvements. So perhaps rather than putting the geneticist out of a job or not doing an assay, we might see it as maybe ordering the right test a bit quicker. Yeah. And we've seen 2 groups demonstrating that in prostate cancer already, where a H&E-based AI is looking at the original biopsy, is deciding whether or not it should order some immunostains, and it's saving a few days, 4 or 5 days on the prostate cancer diagnostic pathway for those patients, which is really valuable. So it's I think the real world is more nuanced and complicated, but in some ways better because we do it that way.
Well, you mentioned hunches, but hunches lead to anecdotes, but it's only when you do something at a national scale that you can convert those anecdotes into evidence base, and that's what we're trying to do, really to industrialise the good work that you and your colleagues in pathology and geneticists have been doing for many years. So I do have a bunch of questions that have come in from the floor, in no particular order. Darren, this is another one for you. This is actually a bit of an infrastructure question. So what are your thoughts on centralized storage of images and how best to ensure collaborative learnings from algorithms?
Yeah, that may come from a pathologist, I'm not sure. But yeah, we on the NPIC programme, National Pathology Imaging Cooperative, we took a very strong view that there was an opportunity for the NHS by centralising. So we are building and we'll have a very large central data store available for any hospital to plug a scanner into. And the reason for that, a variety of reasons. You get economies of scale, you get better contracts by doing that, you can have an expert team running it. But in terms of AI, there are two key advantages. One is that you can then gather data in a structured, standardised way across multiple sites. And an argument, a failing of AI at the moment is that we're just not gathering broad enough data sets, particularly from non-teaching hospital settings. And then by having a centralised infrastructure, you can deploy AI much more easily, potentially at the flick of a switch by installing code. So within NPIC, what we envisage is that once we've got to the point where we want to evaluate an AI algorithm in the real world, we install it on that central system, and that allows it to be used in all of the hospitals that are using our system. And perhaps as importantly, we'll have the staff centrally who'll understand AI deployment and evaluation, so that that burden of understanding that is taken off some of the hospitals using the AI. It becomes much easier to deploy. So a bit of a long-winded, complicated answer.
But it's a hard question. Thank you. I've got a question for you, perhaps. So this question is, are we only focusing on immunohistochemistry? And I can answer that quickly by saying, no, we're focusing on H&E images, which is the bulk of our collection. But why don't we try to address this question? Why are we not doing immunohistochemistry, and what is the challenge of centralizing IHC images?
We're not saying we're not doing IHC. So at the moment, the focus and what we have funding for is for H&E slides. We will also ask most of the NHS sites as we retrieve for the immunohistochemistry, but the problem is that will also include multiple levels, multiple sections, and actually the number of slides we potentially could have could almost, go up 50%, and actually that puts an impact on our storage, things like that. So we're not saying no to that. I think within reason we are asking for immunohistochemistry slides, because actually I think there is a real value to having the immunohistochemistry. So, if you have an ER expressing stain, you compare that to the genomic markers as well. I think actually that could be very useful to do. So I'm not saying no to it. I'm just saying within reason, as long as we can do that, we will do that.
And clearly many of the driver proteins show up in IHC. So Darren, what are some of the challenges? Because immunohistochemistry is less standardized than H&E. So operationally, do you think that could be a challenge for us to make sense of kind of decentralized data?
No, I don't think so. I think it's quite doable. It's easier to image in some ways, yeah. I mean, ironically, when we started in digital pathology 20 years ago, the only image analysis that you could do was based on immunohistochemistry, because the images are very simple brown or not brown images, and deep learning has changed that. So as a result, all the image analysis now we see is on H&E stains. So I'd be pretty optimistic about including IHC. Yes, there are challenges with every lab having their own protocol and the results not necessarily being transferable. But if you need to do some just discovery-based work or comparing H&E with IHC, I think that the data that's in the 100,000 genomes data set is probably of some value. And I think we should make the most of it if we can.
And that optimism is why you're running National Digital Pathology. So a question for both of you from the floor. Can digital pathology images also be taken of metastatic lesions, solid metastatic lesions? What's the opportunity there for longitudinal digital pathology?
So at the moment, we're only consented for what the participant from 100,000 Genomes project has consented us to take a sample for. So at the moment, that would probably be their primary tumour. If it is their metastatic lesion, then yes. The aim and the dream would be is that you have the primary diagnostic tumour. You then also have a digital image from the metastatic lesion if, unfortunately, they have a further lesion. That will be our aim. If you can then also relate that to multiple time course of genomes and then radiology, you've suddenly got an asset that will be incredibly unique and incredibly kind of data driven that I don't think we'll ever no one will ever be able to have. So it would be ideal, but at the moment we're only going on what we have consent for.
Yeah, okay. So I'm going to switch focus entirely now to a kind of operational set of questions. I think it's a tough time for the pathology industry. We have a massive shortage of pathologists. Maybe you can talk about that. And COVID has been very difficult as well. But maybe you can, Darren, you can talk about how digital pathology has changed, not just the logistics of the discipline, but also the lifestyle for new recruits and students.
Yeah, and I think for pathologists that are listening in, it's probably worth hearing that, yeah. So the places that led the world in the initial days of digital pathology when it was first developed were those that really had acute shortages of pathologists, in Sweden, northern parts of Canada, where they really wanted to try just keeping a service running. It's coming a little bit later to the NHS and to the US as well. But absolutely, I think one of my colleagues, Bethany Williams at Leeds, has written a very comprehensive review of the reasons why you might want to go digital. And there are all sorts of strategic reasons for the NHS, operational reasons for hospitals and laboratories, and individual reasons for pathologists. And if you just focus on the pathologists, lots of compelling reasons. I don't know whether perhaps it's developed a sore neck yet, but most pathologists have got some sort of musculoskeletal problems due to being hunched over a microscope for most of their life. Pathology is a job that sometimes is chosen because of the flexibility of the career offerings, and I think digital pathology has definitely enhanced that. So at Leeds we've had 3 trainees joining our programme out of 9 1 year who were coming because of the technology. We've had 1 consultant who now works a significant amount remotely from home, which completely enables their life to continue, their working life to continue. And we've obviously during the pandemic been very fortunate that we've been able to keep looking at patients' images and showing them at MDT meetings, and our trainees have continued to be able to look at images and be trained when times were such that you couldn't sit beside each other at a multi-headed microscope. So lots of key benefits, and I think that young pathologists coming into the profession see that. It's not an overnight switch from analogue to digital in pathology. It's going to be a multi-year programme, but I'm hoping that it'll attract people into the profession, make them feel they're doing something exciting and developmental. And perhaps we should say that they're not going to have their job replaced by AI now today. It's going to be very exciting because they'll be using AI to do some really cool stuff in the future.
Yeah, I completely agree. It's not man against the machine. And perhaps, so far you've spoken mostly about what we're doing with our retrospective pathology data, but could you also clarify prospectively how we're going to support the NHS with prospective sequencing of patients in the GMS? And then maybe Darren could go on to tell us a little bit about the NHS's prospective plans.
Yeah, so the Genomic Medicine Service, at the moment we have a series of cancers that we're kind of standard of care commissioners, and we can, those participants that have a research consent again, we will be making requests in a similar way for those images as well. So we'll ask the sites that will have the glass slides to digitise them. Some of them may have scanners, and so we'll be asking for the digital scans for those participants as well as their radiology images. So the idea would be is almost near real time as that sample is being processed, we'll be also able to digitise those images as well. And that probably feeds quite nicely into Darren and his national strategy.
Yeah, so I think we've been working closely with Genomics England for a few years now and saw an opportunity to align our vision with what you're doing. So as part of our ongoing funding of NPIC, we propose to develop 2 national networks, one in paediatric tumours and one in soft tissue and bone tumours. These are led by Neil Sabir in Great Ormond Street and Adrian Flanagan at Royal National Orthopaedic Hospital. And through those 2 programmes, we have a really very engaged community of pathologists who want to be part of that, and our funding allows us to put scanners into a lot of those sites to enable synchronous gathering of the digital pathology data alongside the genomic sequencing that's being done. It also brings other benefits to those communities. So Adrienne and her colleagues, only 5 of them in the UK are able to diagnose bone tumours or are certified to do it. So it enables that very small community to work together, get the right diagnosis quicker for patients. So we envisage national MDT meetings discussing patients, collaborating amongst that pathology community. in addition, so there are operational benefits in addition to the strategic benefits of scanning the digital pathology at the same time as the sequence is being.
So we can have a real-time MDT for example, an osteosarcoma, which is such a specialism and such a horrific disease.
Yeah, I'm quite excited about
that's
that.
Just wonderful. And one thing I'd like to draw attention to, which I've read many times over the years, is that you did an extensive business case for why it makes sense economically for a hospital to introduce digital pathology. I'm sure you were anticipating many of the lifestyle benefits that then became essential during COVID, and you've made that available on your website. I know that there are a lot of startups and other companies that have benefited from that. Maybe you could tell us a little bit about how the economics of this works in a hospital.
Yeah, and I think at Leeds we're always very cautious about the technology not being mooted as simply a cost-saving device or a centralisation of pathology labs device. So if you read the couple of papers that, again, Bethany Williams wrote about the future of pathology and how we could support it with digital, it's very nuanced. So yes, there are settings in which it's mandatory to have digital pathology because you've got a remote laboratory, you've got remote pathologists. But as I said, the benefits are much more complicated than that at different levels of the health service. And it also depends on the health service's willingness to deliver services digitally or otherwise. I think digital pathology in the NHS has definitely hit the tipping point now. So people are coming to us saying, we are going to go digital, tell us how to do it. Whereas 3 years ago it was, well, tell me why I should go digital. So the case for needing to be accepting of it is being made already. It will take time to establish that it's not going to overnight replace the traditional microscope and paper processes that we have. But what you find is that once you've gone digital, the benefits start multiplying, like all digital transformations. They take some years to come, but once you've done it, you couldn't possibly go back. And I think that's what we're finding so far. This is one of my jobs, is to encourage both the pathologists and the laboratories to consider the move to digital and support them in doing it, but also to influence those who are making big decisions, that they should be supporting the transition to digital, both financially and emotionally, I suppose.
Yeah, absolutely. And perhaps I'd like to ask you a little bit more about the machine learning program. You're not a machine learning expert yourself, but you are trying to orchestrate the coming together of this platform and tool set that will really allow different modalities of research on genomes and the spatial context. Maybe you could tell us a little bit more about how that program is going and what you envisage the research experience to be like once the product is built.
I mean, the sky's the limit, so I'd like to say the research experience will be phenomenal. There'll be no questions about that. But I think at the moment we're working with users, so we need to understand this is ultimately going to be a very novel platform. This has never really been done at scale. No one's ever handled this much data. So we need to really understand what are the questions our researchers want to ask and how is that going to be done. So is it going to be very much a code-centric front end? Is it going to be point-and-click interface? And how much kind of input do we need to help them with that? The tooling is ultimately out there, like if you just want to look at a whole image and just identify tumour versus non-tumour, those tools, those modules, whatever you want to call them, already exist. But I think the challenge we now face is kind of integrating, this is tumour, this is probably an ER positive tumour, or this is a HER2 negative tumour, and what does that morphologically look like down a nuclei is actually a combination of a series of different kind of analysis tools. And that's what we're looking to try and bring in together. So it's all going to be ultimately a cloud-based research environment. All the images are going to be cloud-based. I think the big issue, or potentially one of the biggest challenges, will be that kind of computation element. So we talk about the GPU usage. The storage isn't going to be too much challenge, but that compute and the need to analyse thousands and thousands of images and genomes together will be a real challenge for us. But I think actually AWS are being incredibly generous with their time and with their efforts to help us with that. And I think we can kind of work towards them in delivering that. I think in the next 6 months will be really interesting. I think we're looking to do a few small pilots with a few small use cases with our users, and actually working with a group in Leeds to see if we can do some small cases in ovarian cancer. And I think ultimately we'll work towards what is best for us as users, and ultimately we have an asset that we need to be helping as many people as we can to learn from. I think that's a real focus here, that we have something that we can almost push the UK kind of research environment a few years forward with this asset. And so that's the way I see it, is that how can we help as many of our academics and our stakeholders as possible.
Yeah, I agree with everything you said there. And if I address the audience for a moment, so Genomics England's research strategy is very much to be an enabling platform for the global research community, but for both academics and for biotech and pharma to come and use our data. Now, this is a cutting-edge field, multimodal genomic and image analysis. And I think we have quite a clear understanding today, as Darren's explained, of the clinical benefits, and we can also see companion diagnostic benefits out there. But we know also that there are drug discovery benefits to this approach. We know that there's going to be kind of digital target ID being done. We know that there are more biomarkers out there that will help to classify, stratify clinical trials, and we know that we can reverse engineer these algorithms that predict response or predict prognosis and look for the drivers of response in the features of these combined genomes and images, and then use that as the starting point for new discovery programs. So I would ask the biotech companies and the pharma companies who are interested in that to reach out to us, because we are really co-creating this platform around your requirements, and we need to have those. This is not something we have in-house expertise on. So a call to industry: come and work with us and help us create the largest national scale multimodal research platform on the planet today. Good, thank you. I think we should call on our next panel. Thank you, Serena. Thank you, Alona. So I'm absolutely thrilled. I have two real experts on the stage today. I'm going to ask them to introduce them to you individually, but this topic is on bioanalytics. And Alona, maybe I can start with you. Please introduce yourself.
Hi, I'm Alona Sosinski, Scientific Director for Cancer at Genomics England.
And I'm Serena Nixonal. I am a CRUK Advanced Clinician Scientist in Cambridge. I'm also an Honorary Consultant Clinical Geneticist in Addenbrooke's.
So Serena and Alona are both very humble, but I need to just explain a little bit more. So Alona has handcrafted all of the clinically accredited cancer biopipelines and diagnostic tools that we use to create lists of pathogenic variants that go out to the genomic lab hubs, and has really innovated in partnership with the Dragon team in Alona, abilities to predict productive fusions from short reads, and it's incredible what you do for us. We're very lucky to have you in-house. And Serena has been an extensive user of Genomics England data. Serena recently won the Steiner Award for Cancer, which is a bit like winning the Field Medal in maths. And so we've been very, very lucky to have Serena, who's gone deep into our cancer data set and created mutational signatures and callers for homologous recombination and mismatch repair, and a whole pipeline of new algorithms coming. So we're delighted to have you here. Thank you for coming down from Cambridge to join us today. So first question to both of you, this is a conceptual one. I'd just love to hear both of your vision for cancer diagnostics in the next 10 years. Maybe we'll start in-house, start with you, Alona.
Thank you, Parker, for this nice introduction. Yeah, it's quite a task to fit several years' vision into 2-3 minutes. I will try. Yeah, our current approach to the clinical interpretation of the genomic test is still very single mutation focused, and we are looking for these small variants, structural variants in a specific genes which can drive development of cancer. And at the moment we are working a lot with clinical scientists, with clinicians on supporting them in this journey. So we can have national platform which would support interpretation of the genomes, because genomes can be overwhelming. So we need platform which would help to sift through the thousands of the mutations, find the one which can drive cancer. So this is good. But these driver mutations, they can be missed sometimes, and sometimes they're very difficult to make interpretation of and find if they're pathogenic, not pathogenic. So this takes us to the next level of interpretation. If they do pathogenic, then they disrupt some functions, molecular pathways in the cell, and they leave big footprints in the genomes, hundreds and thousands of mutations with some specific patterns. So we want to recognize these patterns because many of them are linked to the patient response to treatment, to prognostic markers. So this is where the strengths of the whole genome sequencing come apart. And there is a lot of buzz now in the research community about these markers of genomic instability, but then the question is how we translate them from research space into the clinical space. And obviously, the easiest way is to do clinical trials using whole genome as prognostic markers, prognostic tools for these clinical trials. And thanks to the genomic medicine services, whole genomes are now covered for a lot of clinical indications. So we can develop here a new generation of clinical trials, which are based on the whole genome sequencing that used genomes for diagnostic of patient stratification. But so far, these patterns, they usually come from our previous knowledge of genome biology. And now we have probably enough data in our National Research Library to feed into the modern artificial intelligence, algorithm, deep natural learning, all this buzzword. You know? And until recently, we just didn't get enough biological data to use the whole strength. So now probably we are at the stage, and we mentioned before, like collaborations with Google and how we can use the strength of these algorithms to discover new patterns without previous knowledge. The next level is combine genomes with all multimodal data, and we probably already spoke enough about combinations with image data, biochemistry, knowledge about patient history. So to make it short, in, I don't know, 10 years, artificial intelligence learned platform, which is trained from our multimodal data from the National Research Library. New patient arrive, you run it through this trained system. It extracts the most relevant bits of patient information from genomes, from images, from biochemistry, put it together into integrative markers for patient prognosis, whether the patient is likely to transform from primary to metastatic, what are the treatments that patients are likely to respond, and everything is presented into this interactive and intuitive way on molecular tumor board. So we support our molecular tumor boards in making decisions about patient treatment.
Wow, that's a very comprehensive response. Thank you, Aletta. Very good. Serena, how would you like to add to that?
And I hope that happens in our lifetimes. Yeah, I'm not sure there's a huge amount I can add to that, really. I suppose I could come at it from my perspective. We've been very fortunate to have had the opportunity to use the data that's been in the research environment. As you said earlier, over 17,000 whole genomes. That's the largest collection of whole genomes in the world. It is the most comprehensive data set worldwide, and it's been an enormous privilege for my team and I to be playing in the environment. And I think there's hundreds and hundreds of people who are doing the same. We've been sort of searching for patterns of mutagenesis. Alona's already mentioned them, these signatures, because we feel that they tell us something about the history of the cancer, how that cancer has arisen. And if you understand each person's cancer, then you might be better placed to treat that more efficiently. And so, we've, as you've mentioned already, we've come up with a couple of algorithms that help us, or help clinicians in the future, use genomic data in order to inform the treatment decisions going forward. This is only the surface that I'm talking about here, because as we've already heard from the other panels today, there's just a potential a huge amount of additional information that could come from digital pathology, from imaging that we could also gain from. But just from this little area, which is just somatic genomics, we've come up with a couple of algorithms that help us to look at whether a tumor is defective in a particular DNA repair pathway, for example. But just to give you one particular example, we did work in the lab. So, then there are plenty of scientists out there who are not data scientists who work in labs, and they'll find things. Now, if we all had access to the Genomics England data, it's nice and accessible, we could validate our findings very quickly. And in fact, what was mind-blowing for my team was that there was also clinical pathology information available. There was immunohistochemistry data there, and we were able to train our algorithm, validate it, and then publish it together with Genomics England, do a piece of IP together. And the whole idea then is to put it back into into the National Genomics Pipeline so that everybody has access to it, not just us in Cambridge. You know, it is available to all the 7 genomic medicine services throughout the country. And you know, it's just such a privilege to be a data scientist working in that system and environment and know that you can put it back into the national system for equitable access throughout. I think a really important point to make is that the insights we make are at population level. They're clinical or biological insights of a whole population, and it's anonymized data. And I think that's super important to realize. All of this is done in purely anonymized data, but the benefits are going to be felt at a patient level, at an individual level. And so I don't think people need to be afraid of their data being here in this environment. It's perfectly safe. I don't need to know who you are or where you live or anything about you. I just need to look at the genomic information and look at some of the clinical information in order to gain population level clinical insights in order to then apply for the rest of the population. So It's been a fabulous privilege to be working with Genomics England.
We're glad to have you working with our data. And I'm going to ask you a little bit more about the science in a minute and what a mutational signature is. But first, I would just like to ask, in the context of you being a clinician, you're not just a data scientist, and I know you've seen the impact of some of your algorithms and some of your work on actual patients in the clinic. So perhaps you could kind of lift this out of the detail for a minute and just tell us how patients are being affected by this.
So every genome is, really individual. Everybody's genome is just specific to them. And historically, we treat cancers as a collective. So, if you have an ER-positive tumor or triple-negative cancer, everybody more or less gets the same treatment. But individual patients may have something very specific wrong with their tumor, and then you'd want to treat that patient to the best of your ability. We still have a way to go before we can actually we'll need to go through accreditation, all those sorts of things, demonstrate prognostically that it can, it says something about outcome, demonstrate predictive properties. But we have had an impact on an individual, a couple of individual patients, for example, and that's been in the UK. So a patient in Hull, nowhere near Cambridge, who had a metastatic cancer for whom whole genome sequencing revealed something very specific about his tumor. And that meant that his tumor may have been sensitive to a particular drug. In this case, it was an immune checkpoint inhibitor, but he was metastatic. It's not licensed in the UK necessarily, so you have to, as a clinician, you'd have to go through trying to get IFR, individual funding requests, done. But in fact, it was turned down twice. So there's a piece of work that I think we need to do together with sort of legal frameworks and regulators in terms of how we can modernize how we use genomic data and data in Genomics England. But long story short, he got the treatment in the end, and it had a fantastic outcome for him. And you'd like to think that in our lifetimes, we will see, well, hopefully, within the next 5 or 10 years, we'll be able to get to a point where a genome sequence together with a pathology or radiology would help to inform decisions for individual patients as individuals, as opposed to as a sort of a collective bucket of diseases.
Yes, I completely agree. And perhaps you could talk a little bit about what you consider the role of Genomics England to be in the whole cancer pathway.
There's no really other site in the world that has the centralized infrastructure that Genomics England has. It's done it in a remarkably short period of time, there's things that will need fine-tuning, but the infrastructure that has been built is pretty phenomenal, really, and I seriously hope that carries on. There's no other system that allows us to also recruit patients from all over the country as well, and be able to return then that information and that knowledge back all over the country. You'll need Alona's pipelines, you'll need input from Dragon, etc., to be able to get it better and better and better and return it back equitably. So I hope that Genomics England carries on with a sort of centralized way of being a place, or that data can come through, that also permits innovation, helps to develop some of the relationships with pharma, with regulators, to enable us to be able to do clinical research more effectively, and then hopefully develop the infrastructure, the interfaces that permit everybody to do clinical research, not just people with big IT teams. That just shouldn't be the case. It should be that anybody and everybody who's got questions, and there's lots of very smart kids coming through the system, right, they need to be enabled to do clinical research. And hopefully that will come through Genomics England.
Yeah, I hope so too. I think we have made a lot of progress, and part of that is thanks to the push from scientists like you and Alona, but also part of it has been thanks to the pull from the NHS, who we just collaborate with and entirely depend on to drive this into clinical practice. And we are really impressed that even during these hard times for the health system with COVID, that the NHS is still progressing actually really quite rapidly and ambitiously, kind of driving genomics, not just whole genome sequencing, but also large panel genomics into the NHS pan-tumor. So Alona, one of the questions that just came in from the floor, which I think would be a good one from you, but feel free to answer as well, is about metastatic lesions and metastatic predictions. So really the question is, can we achieve with genomics that dream of thinking about cancer as a chess game where we can diagnose not just what is happening today in the body, but also predict future metastatic and future clonal evolution?
Yes.
Good.
Yes, I think I want to talk a little bit about real-world data that we managed to collect in our research environment, put it side by side with genomic data. We were very lucky in this country to have these centralized sources of patient clinical information, like a patient electronic record. So we can put side by side genomes with patient diagnosis, histology, pathology reports. Well, they're still in the free format for many clinical indications, but still you can extract pieces of information from there. Information on patient treatment, information about patient mortality, some information about patient progression. At the beginning, there was a big lag between patient diagnosis and the time of this information arriving. Now we are probably down to 3-4 months with a lag time. So basically we can follow up patients almost in real time, and for some of 100,000 genomes patients, we already have more than 5 years of follow-up. And we can see whether patient's disease transforms from primary to metastatic, or whether the patient is still alive, whether the patient is still free of disease after surgery. So we can start to put together this information with genomic information, compare cohorts of patients with transformed to the metastatic disease very quickly, patients which are still free of disease after 5 years, and start extracting these genomic markers which would help us to diagnose this patient, to prognose this patient when we see it at the diagnosis point, and predict whether the patient is likely to transform into metastatic disease, and when is it happening.
So I just want to add for the audience a little bit of context to what Alona said about real-world data. So we have integrated in near real time, I think we have about a 1-month lag with the cancer registries, which are hosted by, curated by Public Health England and hosted by NHS Digital. And what that's allowed us to do is really quite remarkable work with real-world data. So any of the listeners out there who are interested in this, an example would be a study that we did with PIK3CA, which I think is the second most prevalent oncogene. TP53 is the only more prevalent tumor suppressor gene out there. And it is a well-established fact, I think, it was published in 2012, that in breast cancer, the PIK3CA wild type ER positive, HER2 negative women conveyed a real survival benefit that you could see very clearly in the Kaplan-Meier curves. And we were able to recapitulate that with real-world data very, very accurately to a very low error margin. So there's just one example to show how real-world data that comes from the cancer registries, that's annotated from remission and relapse data that we have in our longitudinal data, can recapitulate RCT data, randomized controlled trial data, which is really, really very, very useful for people who want to retrospectively study subgroups.
Yeah, can I just add that on this recent American Association of Cancer Research conference, there was a lot of presentation around genomic instability, chromatripsis and how it can be, it can work as a predictive marker of the quick transformation into metastatic disease. So now we can use our genomic data, our real-world data, to confirm these findings.
Okay, thank you. We've got a question here that's come in. It's about tumor normal. So do you also sequence normal tissue to differentiate between rare germline and true somatic mutations with a matched tumor normal workflow, or do you rely on tumor-only variant calling? So I can quickly say we for every single cancer patient, we sequence the germline at 30x and the somatic tissue at 100x, and maybe you could both speak about why it's important to have tumor normal matching. Serena, do you want to lead with that?
So, whole genome sequencing has always been based on the premise of tumor normal matched sequencing, and yeah, their data is fantastic. High coverage. I think I don't think you do any tumor only sequencing, do you, in the?
Not at the moment. We are under a lot of pressure from hematological community to start sequencing tumor only for some acute leukemias, just because at the time of the diagnosis it's very difficult to collect germlines. We have a pilot with the skin biopsies, but it's not likely to work on the national scale. You're able to adopt to it in a big research hospitals, but probably not in every hospital. And of course, we want our service to be very inclusive. So we did quite a lot of progress in returning results for tumor only on hematological cancers, with expectation that once patient goes through the treatment, we will be supplied with germline as well, clean germline as well. So we will have both tumor and normal for research.
It's a good question, though. It actually shows what the potential is. So I've talked a lot about somatic mutations only, but there is a second genome there, which is the normal genome that anybody and everybody can use. And when that data is there, that's just the most powerful thing. And over time, it's only going to get better. It's only going to get more and more. Every time your data increases, your ability to learn something new just goes up and up. So you can do anything with it. Look at the germline more, look at the somatic bits more. So, the world is your oyster.
So size does matter.
It absolutely does for data.
I do feel that I need to ask this question because this question has come in from a patient. So this patient says, can those of us with metastatic cancer have our genomes altered to get rid of the cancer? Perhaps you could both talk about some of the strategies that are used for metastatic disease. We are not in the business of base editing genomes, but there are increasingly rich strategies for managing metastatic disease, and perhaps you can talk to that.
So as I understand it, you will be taking in patients who have got metastatic disease, isn't it, as part of your next phase. So I think we'll be learning a lot more about metastatic cancers. It's true that currently the vast majority of the data in Genomics England is on primary tumors, but I think that's going to change. There are certainly activities going on elsewhere in the world as well that are looking at whole genomes of primaries and of metastatic cancers. And actually all the data is very valuable, Genomics England together with all those other cohorts. But I think we're going to learn a lot more about metastatic cancers. As you say, we're not quite in the business of being able to change anything just yet. But yeah, I think we'll be learning a lot more.
And before I hand on to you, just for clarity, so about 70% of our cancer patients that we sequenced were sequenced treatment naive, so before treatment started, of the primary tumor, but they were stage 4 cancer patients, so likely already metastatic. Alona, do you want to make any comment about the metastatic setting?
Yes, I don't think we can edit genomes which are already affected by tumor disease, but we can develop targeted treatments which would only kill metastatic cells where we see these genome damages.
Okay.
So that would be the way of getting rid of metastatic tumors.
Yeah. A question's come in that I would like to answer myself, actually, and this one is, the question is, how does the wider healthcare professional or researcher access the GEL data set? So I think this is a really important part to introduce. trust and validation of the researchers that have access to our data. So we have a very, very rigorous way of managing who has access to the cancer data. First of all of our data, cancer and rare disease, lives in a trusted research environment, and it can never be removed at a patient level from that trusted research environment. It sits behind AWS's firewall, Amazon Web Services, which is extremely secure. They invest billions in security, and the information governance is very tight. To get access to the data, whether you're a researcher or a biotech or pharma company, you have to apply for access to our Access Review Committee. And the Access Review Committee is entirely independent of Genomics England. It's made up of professors and also participants who literally have skin in the game, who have their samples in our biobank. That Access Review Committee assesses, number one, is this the type of research, is this the type of company which is bona fide, which is qualified to have access to the data, and then they have to explicitly agree the research study that they choose, that they've written to study. And there's a technical summary and also a lay summary of every research proposal so that our participants can review and approve it. And then at the completion of the project, if they want to take aggregate results, so algorithms or GWAS results out of the research environment, that has to go through an airlock process. So our environment is not connected to the internet. The only way to get things out is a manual approval process by our airlock team, who literally review manually every single extract request from our environment and ensure that there's no kind of nefariously or accidentally re-identifiable patient data kind of hidden in those results. So it couldn't be more secure, and we're very, very grateful for our participants in making their data available for these research purposes. I hope you don't mind me covering that for you, but I think that's a really, really important thing to say. I've got a question that's come in. Do we have plans to extend genome sequencing to other ethnic groups? And perhaps Alona, you might want to talk a little bit about our diversity program. I can support with that if you want. And Serena, you can talk about the importance of diversity in genomic research.
Yes, of course, Parker. So this is the program that was recently launched in Genomics England, our diversity program. The expectation is that we will learn more about population frequency of the mutations that we rarely, or any type of variations that we rarely see in our collection of the 100,000 genomes. So it will inform our interpretation of, for example, tumor mutations, and do they look like the population-specific common mutations, or they're rare and they can be pathogenic causative of the cancers. So it's very important to have this background information on prevalence of the mutation that you see in the patient genome, prevalence in the background population. So this is probably one of the drivers.
Okay. And do you want to add to that at all?
No, I think it's really important for us to reach out to all the communities out there in the UK. A lot of research, I guess, has been rather focused on sort of a white Western population, but the UK is becoming increasingly diverse, so we need to learn more about across the populations as possible, and hopefully something like this, where we come to a point where everyone's tumors are going to be coming through gel, we will learn from the whole population.
We will, and actually in a panel, I think coming up next, we have an explicit session all about diversity in our diversity program. A question that has just come in, and this one I think is for you, Serena, and it's great because it gives you a chance to talk about your mutational signatures. So the question is, can you give more detail about structural variation and that you can discover through your research? And perhaps you might start off by talking by explaining what a mutational signature is and how that allows you to look at genomic instability across the whole genome.
Okay, so as your cells, you start off from a single cell, and as you become human, your cells have to divide lots and lots of times, and you can make errors during the process of copying your DNA, and you can acquire mutations. And you will have heard about mutations from COVID, for example. Now, cancers have historically been believed to have been driven by these driver mutations, these few events that confer some kind of selective or growth advantage. 1, 5, maybe 10 driver mutations per cancer, and people are really focused on that. But actually, as your tumor develops, it acquires thousands and thousands of mutations, and sometimes hundreds of thousands of mutations. And those mutations aren't random. They actually tell you something about the biological processes that have been going awry as that cell has turned into a cancer cell. And so mutational signatures are these patterns, are the patterns of mutations that arise because things are going wrong in your cells. And that can be from anything. That can be from within your cells. You could have be producing something in your body that's damaging your DNA, or you could have a repair pathway that isn't working, or you could be a smoker that damages DNA. So things from outside the body as well as inside the body, all of it can leave these patterns that we call mutational signatures. And when I was at the Sanger Institute, that's where we came up with this concept of mutational signatures, and we showed that you can see it in cancers. Historically, and that was just technological, we started with single base changes because that was the easiest thing that we could call. And today, people are starting to look at indels and structural variation as well. So it is one of the things that's amazing about whole genome sequencing. It's that you can see all these structural big pieces of DNA that are swapped around or moved to other parts of the genome. There are patterns in them as well, and those patterns tell you something about the biological processes that have been going wrong. Sometimes those structural variations can produce a driver event, like we've heard already today, NTRK fusions, for example. But sometimes they can produce patterns, and those patterns say that a repair pathway isn't working, for example. So that's how structural variations are starting to be used, I suppose, in whole genome sequencing, and actually already has clinical applications in some certain tumor types.
It does, and we're planning on incorporating your algorithms into our environment, which we call microsatellite instability, or mismatch repair, I should say, the precursor of MSI, and also homologous recombination, which is really one of the major forms of DNA damage repair. And for those of you who haven't read Serena's paper on mutational signatures, it's quite stunning to see actually how beautiful some of these signatures are. You can see these very smooth melanoma signatures where you can see the damage comes from UV light fairly consistently, whereas in your colorectal cancer signatures, you can see those subgroups of mismatch repair and non-mismatch repair patients. So it's something which is, I think, really helps to visualize the heterogeneity and really the consequences of these mutational pathways on patients. Is that a fair description?
Yeah, absolutely. Always ask someone else to explain your research for you, because they always do it better.
Well, they're also very colourful, which is really, really nice. Okay, I have a very technical question, which has got your name on it, Alona. So here we go. Could tumour-only variant calling be used to investigate why double-agent genes act as oncogenes in some cancers and tumour suppressors in others?
Yeah, I suspect it's come from some research.
I would have thought so.
So as I explained, this scenario for this tumor-only pathway that we're developing now for the NHS and for genomic labs, we vision to process tumor only, mainly to return a small set of clinically relevant results back to the genomic lab, back to the oncologists. And then once patient is treated, we expect to receive germline sequenced coupled with the tumor and put proper tumor normal matched pair into the research environment. So if you're talking into the more research questions, then we would still stick to the proper tumor normal pair.
Okay, I think that's a good answer. I've got a question that very much has your name on it, Serena, because it's about breast cancer, which is a topic we talk about a lot with you in Genomics England. The gist of the question is, what are we going to do about curing breast cancer? I think this has come from a patient, and I would love it if we use that question to explain the insights that you found into DNA damage repair, and perhaps to address the biotech sector out there about the opportunities for addressing more responders to kind of BRCA and other pangenomic features.
So this is an enormous question, and certainly not one that any particular individual would be able to answer, and will require the input from the community and from the patients and from all the clinicians to be able to get to. So first, I think we'll need appropriate sort of infrastructure to permit us to be able to play with the data and the data that's there, and invite other people from outside, other researchers, other clinicians and biologists as well, right? We need people to ask the right questions of the data, and data scientists may not have all the questions, and that's why the interaction between all the parts of the community is very important. We'll need pharma input to be able to help us perhaps develop those studies, those trials, because playing with data is one thing. You develop an algorithm, that's something, but to make it clinically useful, you have to demonstrate that it does something either about outcome or that it predicts something. So to do that, you will need trials. You will need to put it in a context of a study. And so we need and actually we already have a sort of a setup, which is the Genomics England setup, but we need to be able to invite others, other clinicians, other researchers, other clinician scientists and pharma to engage in order to use this environment, to use the connections and the GMSs across the country in order to be able to recruit patients into appropriate studies that can be cascaded nationally. I know that sounds almost highly ambitious, but I don't think it's beyond the UK to be able to achieve something like that.
Yeah.
And I think over time, because this is the other thing, over time you'll be accumulating data on those cases in patients, and the knowledge you'll gain from that is massive. So we'll need infrastructure to be able to do that sort of thing. We'll need interaction from the community. We need infrastructure that permits analyses across sites as well. Sort of more federation analytics. You've already touched on companies like LifeBit that are in that space. I think we need to be able to do more of that sort of thing, because of course there are other data sets that have been created there's Interval, there's UK Biobank, there's the NIH Bioresources, huge numbers of TREs across the country. We need them to be better connected to enable people to do research more effectively. Just needs to be fast and quick and efficient. And I think nationally there is a drive towards trying to build up federated analytics. And then we need to have the infrastructure to permit people to explore the data a bit easier, because right now it's still a little bit limited to people who have IT teams. We need to have the ways of biologists and clinicians who can kind of go into an interface and kind of explore data and how they want to. So I think infrastructure, community interaction is terribly important. And we need buy-in from the whole country. And this is something that we should all be proud of, and we should all want to be part of and contribute to and recruit patients to, etc. So I think it's wonderful to be part of the innovative bit of discovery and finding new things. But we need to, if we want to put it back to patients, we need to give it enough time to consolidate as well. So give our GMSs, our genomic medicine services, our clinical scientists enough time to develop the workforce, train them, educate them, put in the infrastructure, get the accreditation, have the rigor, because of course doing something as an academic is quite different to doing something in the clinical context. But once we have that, we've put in the consolidation and we have the innovation infrastructure, the world is your oyster. Then in the UK you can really start to explore things. So in breast cancer, for example, 70% of breast cancers are cured, but there's still that 30% that we don't understand. How do we now find those 30%, use this infrastructure that's been developed across the country, interact with the right pharmaceutical companies, and try to understand the natural history of the disease, try to understand how to treat these patients better. I think it's kind of an ambitious sounding thing, but I think it's something that can be, that is achievable in the long term if you have the right things in place.
It is ambitious, but cancer is an aggressive disease and we need an aggressive response. And I think what these three sessions with three experts from the wet lab, from pathology, from molecular biology show the importance of is really the convening role that the NHS and the Genomics England have to bring scientists, to bring pharma companies, ultimately have the drugs that give the kind of clinical pull to push patients through clinical trials. And of course, what we can always depend upon is the courage of the patients who are prepared to put themselves out there and try new things, not just for themselves, but for the benefit of future patients that present like them. So I think at that point, I'd like to end the session and just thank you, Serena, you, Ilona, and all of the previous speakers for coming down to London to talk to us about cancer today. Thank you.
Thank you.
Hello and welcome to the last and probably best session of the day. Thank you for agreeing with that. We are going to be talking about diverse data, health inequalities, genomic health equity. I appreciate that some of you may have been here all day or you've come in especially for this session. So grab your cup of tea, or because it's so late in the day, your G&T. No one's here to judge. Sit back, relax, get really involved in this meaty discussion. It's an absolute honour to be here in 3D. It's nice to be able to kind of see and smell and touch all the panellists. Smiling in a good way. You're looking really nervous, Sufyan, there. We've got a panelist for a really varied background, lots of different disciplines, clinical, ethics, anthropology, research, and so be prepared for a very, very diverse discussion. I'm going to take Chair's privilege to give us a lay of the land, but before that I just want to kind of introduce who I am. So I am Maxine McIntosh. I'm the programme lead for diverse data at Genomics England. We're a programme that aims to ensure that genomic medicine works for everyone, and I'll give you a wee bit of detail a bit later in terms of how we're kind of delivering on that ambitious goal. So obviously, we have a long-standing history and problem with diversity in medical sciences. So science has been colonised by weird countries, Western, educated, industrialised, rich and democratic countries. It's a quite useful acronym if you want to just lump all of those countries in one. And in a world where our research and our lives and our clinical practice is increasingly being driven by data-driven systems, we have to be very cognizant of some of the solutions we're building on the back of potentially imbalanced and biased data sets. And this, in the context of genomics, can impact everything from misdiagnosis and understanding human biology to understanding human history. And we really have ended up in a position where not all clinical insights are equally as accurate or applicable to every single type of person. Just a few statistics to see how far we've come, or how little we've come, is maybe a better way of putting it. In 2009, only 4% of precision medicine studies had individuals from non-European ancestries. And this has improved to 19%, which was a figure from 2019. And this is largely dominated by an increase in individuals of Asian ancestry, but still far behind where we should be from a global context. We've had loads of efforts in different ways over different years addressing this, things like H3 Africa or All of Us, but still we could do a lot better. Now, the slight challenge with all of this is that it's not simply as easy as increasing the number of people of different types of backgrounds in data sets. It's a much bigger problem. It's a structural problem. You've got things like the funding that determines what sorts of questions the research community poses. We've got the poor diversity amongst the actual researchers and clinicians in the sector working on these problems. We've got groups who've been historically excluded and today still excluded. We've got challenges like preferred cohort effect, i.e. it's just easier to work on the data sets that we've already and always used. And it's important to say that this is not just a genomic challenge, this is a challenge across almost all of health data. But genomics has a specifically challenging problem that certainly in the past and in many places around the world and on our home turf today, genomics has been used to justify particular abuses, particularly in eugenics. So it's a complicated problem. And our programme is going to try as hard as it can to make a tiny dent in what is a massive global challenge. And so we're doing this kind of through three main ways. So one is we're putting our hat in the ring to increase the number of individuals from underrepresented backgrounds in genomic data sets. So we're really pleased to be partnering with Born in Bradford in the first instance, who are a cohort who have many more individuals from a Pakistani background in them. Secondly, we're looking at working with different types of community groups, grassroots organisations, through novel community engagement activities. And lastly, we're cognizant of the fact that increasing the number of individuals and data sets through specific sequencing activities may take a wee bit of time. And there's a lot we can use in terms of the current tooling or statistical methods or even behaviour change right now to make sure current insights with practitioners are as fair as possible. Obviously, collaboration is a really important thing for this, and I think we all recognise we need to work together to raise a very, very low bar when it comes to health data equity. So that's been a pretty long spiel from me, and we have a ebullient panel to my left, some of whom have come not very far, and some of whom have come very far, so thank you very much for travelling all the way down to London, Sufyan. So before we kick off, one of the things that has become very clear is, and I've said it numerous times, I've talked about diversity, I've talked about underrepresented groups, and I haven't talked about who those people actually are. And what transpires is that we've got a very varied, not just in this room, all over the place, we've got a very varied definition of what we actually mean by diversity. And I really hope that none of you agree with your points when I ask you what that is now. We've got genomic diversity, ancestral diversity, environmental diversity, and wider socio-cultural factors. So what I'm going to do is I'm going to kick off by asking each of my panel to introduce themselves and also define what you mean by health data diversity, why you think it's a challenge, and what you're doing in your work to address that. Sufyan, given that we've come so much far, you can be the first one.
I can. So we're doing all the questions.
For all of them.
Okay, great.
Defining diversity, why it's a difficult challenge, what you're doing to address it.
Fantastic. So my name is Sufyan Abid-Dogra. I am an anthropologist by background, and I work as a senior research fellow in this longitudinal birth cohort based in Bradford. We call it Born in Bradford. Between 2007 to 2011, around 11,500 mothers, pregnant mothers, were recruited in this study, those who gave birth during this time frame. And now we have almost 14,000 of children and families where we have genetics data and health information, and we are tracking the health of these children and young people who are turning into young people now, because our first child this year is going to be 13, which actually for us is a journey into age of wonder. So we are trying to understand what are the causes of ill health and what are the causes of good health, and how they are linked across ethnicity, in deprived neighborhoods, and in kind of different communities with different historical, cultural, social, socioeconomic background. So that's what we do. And in terms of what is diversity in relation to genomics, for me it would be, we are talking about England and Wales here, for example, then it should be, the data should be reflective of that. And as long as that is the case, I think we will ensure and we will have genomic diversity in our data set and in our knowledge production. Now, diversity is not only numbers in data. For example, you have 500,000 sample. And then, you proportionally kind of have all ethnicities according to their population numbers. But that won't be diversity. It will be diversity if that data, when collected, when compiled, when transcribed, when translated, and when disseminated, reflect the same diversity. Otherwise, it's just a boring data set saved in some supercomputer and some big boss is running it. It has to kind of answer the question in the complications of lives of people that are linked with their kind of genetic health. And if it does not answer those questions, then it is not a diverse data set. At the same time, if, for example, this data set, when it starts informing and educating and kind of disseminating, it kind of ends up compromising different identities, although it is talking about every identity within that data set, and then somehow certain identities become superior or better or good health genetic makeup on the cost of other identities being born with defects. And then again, that data set in the process of knowledge production is not diverse. Rather, it is very linear and, as you said, very much weird. Now, that weirdness of, in abbreviation sense, of data set is a huge, massive problem and a challenge. And this is the biggest challenge we are facing in this field of knowledge production. So the second part of the question is what we are doing. So in Bradford, we have this fantastic and in a way very unique data set where we have almost 45% of South Asian population, which is kind of reflective of Bradford city's population. And then we have majority white British population and their samples, those children and mothers. And we learned a lot of things through our data, that how benefits of health are unequally distributed. And the benefits of health are there, and everybody can have, theoretically can have access and get benefit from them. But when it comes to actual practice, it doesn't happen. So what we have done in terms of data and health and genes, we kind of try to understand what are the kind of socio-economic, historical, and environmental factors that shape the genetic health and that define the genetic health of certain population groups. We also kind of learned what is the relationship of age, and particularly age of mother, with the chances or likelihood of children born with any genetic disorder. So these were the contribution that we have made in the knowledge production on genes and health. And our plus point has been, Born in Bradford has been champion of diversity in that regards. The challenge that we face is how general public perceives it. So the moment when we start talking about genes and health, and then suddenly the genes become political, and they are charged politically, and they are spoken, discussed, disseminated, and translated politically. And people with their biases and xenophobia, and people with their whole kind of, sometimes in some cases, bigotry, then they start defining it as it suits their political agendas. That has been a big challenge. For example, when we talk about consanguous, am I pronouncing it right? This is the word
I am, but I'm going to give you 20 seconds until you move on to Natalie. So
the media will suddenly have all its cameras on it, and they'll be like, oh, you see, the causes of ill health are in the marital practices. But that is only a tiny kind of proportion compared to the bigger picture, which is 19% of children born with the recessive disorder or other genetic health problems will be from the females who are giving birth after the age of 34. And of course, that reflects another population group. And that's a bigger issue which no mainstream media will be talking about. And then that, again, when people listen, media talking about, like, for example, cultural groups listen, media talking about them in a certain way, then they are not happy and they think science is their enemy, which is exactly opposite message that us researchers, academics, and people in data are trying to give. Their science is here to benefit people and improve their lives. I will shut up.
So Finn has done an excellent full tour of all the problems and solutions. Thanks. You got us covered. I don't even need to be
Let's just go.
Yeah, we should just go.
I'm going to get my train back to Bradford.
No, thank you so much, Stefan. I think the example you gave around consanguinity in South Asian families and the proportion of children born of women who are over the age of 34, I think is such a good example of the lens we take on certain types of questions that we maybe bias when we're looking at genomic research. Yeah. Natalie, do you want to remind of the question? How do you define data diversity, why it's a problem and a challenge, and what you're doing to address it?
Right, fantastic. Thank you very much, Maxine. So I'm Natalie Banner. I lead Understanding Patient Data, and our aim is to try and make the way that health data is used a bit more visible for people, more understandable and more trustworthy. So our focus is on the way that routinely collected health data can be used for purposes beyond care. And this question around diversity is hugely important for the reasons that both you and Sufyan have outlined. But I think that when we think about diversity in data, it's really important not just to focus on the kind of qualities of the data sets themselves. Because I think if we focus solely on what's in the data, as it were, we can tend to think of it as being quite static, as something that's sort of just there, it's sort of monolithic. And actually data is very dynamic. If you zoom out a little bit and think about diversity in the context of the collection of the data and how it's being used, then you get a much more complex question. And of course, I'm adding complexity to something that is already a complex problem. So if you zoom out a little and think about diversity in terms of, well, let's start with what's collected. What information is being collected, what's not? Is what we're counting all that counts? Are there things beyond what qualities we have in the data sets that are collected and that exist themselves that might matter? So there's big questions there around the sort of the diversity, the questions we're asking when we're collecting data in the first place. But then you think about the analysis of that data. Well, who's analysing that data? What assumptions are they bringing to that data? What proxies are they using for maybe certain characteristics? And in saying that there are assumptions there, that's not necessarily a criticism, because of course we all have to make assumptions when analysing and using data, whether we're talking different statistical models or other things. We are making assumptions all the time, but of course those things might be implicit. We might not realise that those assumptions are in there. So we have to ask questions about who's analysing that data, what are they doing, what kind of questions are they asking? And then further on from there, are we thinking about the sorts of the tools that might be developed off the back of that data, the software, the algorithms and so on, and how are they going to be deployed, for example, in clinical practice? And once you start thinking about that entire life cycle of data, these questions of diversity, I think, become much more challenging, much more prominent, but they go beyond, is there bias in the data set? Because there's, of course, that entire life cycle to consider when thinking about diversity. Now, one of the challenges of thinking about that much bigger picture and that more holistic perspective on questions of diversity in data is you can end up getting paralysed quite easily, right? Because you just think,
I feel seen.
wow.
I feel seen.
Yeah, you think, in order to resolve any of this, in order to do a meaningful analysis of this data or to use it in a good way or to develop a good machine learning model, I have to solve structural inequality and eliminate systemic racism, and you can just feel very stuck. So I think it's really important to be able to highlight that these are challenging questions, but it doesn't mean that it's impossible to move forward, because even becoming aware of some of these, the values that you might be bringing to an analysis, or that there might be the questions that you're asking might not actually be helping the communities that you're looking to serve when you're analysing and using data. Even just becoming aware of those things is a starting point. So I suppose my answer to the, why is this an important problem to address, is surfacing it and recognising that you can't necessarily solve for everything, or eliminate all of these biases is a starting point. In terms of what we're doing about it, understanding patient data, one of the things we saw during the pandemic was just the serious disparities in morbidity and mortality for ethnic minority groups compared to the white population in terms of COVID. But something that really struck us when we were looking at some of this data was actually how much is kind of missing. So I think one of the Open Safely studies that looked at COVID morbidity and mortality, that looked across primary health records across the population, millions of records, there was something like 26% didn't have any coding for ethnicity. There was just nothing there, right? So that is a massive amount of data that we're just not collecting and not getting. The policy response to this challenge has tended to be, we need more data. Let's get more data. We need to improve the collection of ethnicity data. Let's just go ahead and do that. Tick that box, exactly, Sufyan. But actually what hasn't been done so far, and what we're trying to do at Understanding Patient Data, is step back and say, well, have we considered maybe exploring with those populations, with particularly ethnic minority populations, what questions they might have around how this data is collected and why it's collected and what it's used for, rather than just assuming we must get more data, we must always get more data. So we're conducting a piece of work with Clearview Research, who specialise in peer engagement and community engagement, and they are working with members of the Black and South Asian communities across the UK to explore with them how they feel about the collection and use of their health data, with particular focus on ethnicity categorization and how they feel about those labels, whether they are in fact, being useful for them, whether they feel that actually research doesn't help benefit them in their life, so why on earth should they support the collection of this data? So that's a piece of work we're doing at the moment. It's kicking off right now, and we should be able to kind of report on that early in the new year. And from that work, we will then work with some health professionals as well to explore with them whether there are further questions that they hadn't really considered, drawing on this kind of deep peer community engagement work we're starting right now.
It's a good sort of tenterhook pitch for watching this space. Amazing.
Watch this space.
Dan.
Hello, I'm Dan. I'm a research...
What's your name and where did you come from?
I'm
Sorry,
a research...
I've just
disturbed my flow.
Sorry.
I've got to start all over again now. So I'm a research bioinformatician at Genomics England. So I work with Maxine on some of this diversity work. In particular, my interest is around like the genetics of this stuff. So, the conversation about diversity is something we've been having quite a lot in the office, and it's something that is quite nebulous and it is difficult to unpick. There's lots of different facets to it, as you guys have been enumerating. So, for example, I like to talk about genetically inferred ancestry. That's what I like, because we look at genes and we look at genetics, and that's what I will then try and infer, and that's how we try and do our analysis. But then that's different from self-reported ethnicity, because ethnicity isn't just your genes, it's it's your culture, your history, the way you've been brought up, your religion, all these things that do have an impact on your health as well. And then there's other elements like deprivation and so on. So yeah, it's difficult to try and define. Like I said, it is a bit nebulous. So if I try to just sort of like maybe narrow down just to the genetics, because
so you don't get too existential and paralysed with the problems, yeah.
Exactly. So yeah, I mean, at Genomics England, we're within the 100,000 genomes cohort, we're actually pretty representative of the UK population. If you look at the 2011 census, you find that really we captured the main groups in a pretty good way. With the caveat that that's comparing self-reported ancestry data from the census to genetically inferred information. But then really, it's not good enough to just be representative. You have to do more, and that's because the numbers that we have currently don't allow us to have like sufficient statistical power to do the analyses that we want to do. And so, we're looking at our data, and we're looking at various different facets of it, and we're trying to understand, are we seeing that, for example, a question we want to know, a question we want to answer for is, do people have a different experience if they're from a different ancestral background? So does that mean that they are less likely to be a solved case? So somebody who's gone through the pipeline, and then we've been able to feed back to the clinician some kind of actionable finding. And that kind of question might at first seem very simple, but really it's completely mired in so many confounders, so many things that might be influencing this, that it's really difficult to answer. But these are important questions to have answers to, and the things that we want to try and focus on. So how are we, well, what are the challenges, I guess, right? So one of the things with the genetics of it is, as has been said by everybody so far, everything has kind of been developed around this sort of white or like Western European framework, and that means that our methods are geared to work in that way. And so concretely, an example of
genetics. The way that we sequence somebody means that we basically chop up, well, not the person's DNA, but we have all of these. No chopping occurs. We basically have like lots of these millions of small reads that we have to kind of align to what's known as a reference genome. But the reference genome is largely European. And that means that we are missing like entire chapters of this book that's our genome. And so, a really interesting paper came out a couple of years ago showing that, as a human, you have about 3 billion base pairs. And they sequenced an African cohort and showed that about 10% of that was missing from the reference genome that we have. So, like really, really key information could be lost there. So the kind of things that we can do about that is try to identify the best methods to get past that. So whether that be building specific reference genomes for each subpopulation or superpopulation, or having some kind of consensus reference. But the actual solution we're not clear on, but it's important that we try and address that there is a problem there and that we do something about that. Yeah, anything else? What was the other question?
I mean,
what are you doing about it?
You've also covered, you guys are covering.
What are we doing about it? Yeah, okay. So, I mean, right now, it's, as I'm sure Maxine would attest, it's early days for diverse data within Genomics England as a program. So we're trying to get the lay of the land and really understand like what the picture is already, because you can't start fixing stuff until you know what the issues are. And I think there's a lot of work going on with the team trying to identify where biases may exist throughout the process, not just at the genetic level, but other elements such as in the healthcare records and the methods that we're applying, all that kind of stuff. So it's, like I said, early days for diverse data. So that's what we're trying to do is build the best picture possible.
Yeah, and certainly in the early days of like just cutting the data and looking at where different protected characteristics might intersect, it's showing how complicated the story is. And I know you gave the example of ethnicity and deprivation, but it's a really messy picture to work out exactly like what do we mean by underrepresented in our own data. It's taking us a long time to even get that out. Harpreet, last but not least, it better be good.
Last but not least, I'm not sure how much I have to say, but I do think there's a few things I would like to bring up. So I'm Harpreet. I'm kind of wearing three hats. I'm a GP in London. I sit on the board of Health Education England, which is a national agency that is responsible for the training and education of our workforce in the NHS. And then the third hat is I'm co-chair of the data and digital workstream at the NHS Race Observatory. So a lot of the things that we're thinking about and talking about today are relevant to that. And actually over the last 9 months or so, I've been working as a clinical advisor to the vaccine program. So how we drive up the uptake of vaccine in underrepresented communities. And so a lot of kind of learnings from there, which I think are applicable to our conversation today. So I just wanted to build on what colleagues have said on the panel. I think one of the areas from the data diversity angle is that I think the what is important, the how is important, but I also think where the data is collected is also important. And so what I mean by that is essentially in healthcare we see, health and social care, we see primary care, secondary care, mental health, community health, and I think each of those areas data is collected in a very different way. And that probably leads to many of the inconsistencies that we're talking about, especially when it comes to ethnicity coding. And I think, it was talked about, it may well be a tick box exercise, but I think it's a really important tick box exercise because we do have quite a rich set of data in primary care, for example, but it's very hard to pinpoint that on ethnicity. And if we started collecting ethnicity data more, then we can actually then do some useful work with it. But I think for me, the really important part is when we come to the challenges of it is how do we apply this in a real world setting? Now, as a GP, as a clinician, I do really struggle with the genetics agenda because there's a paucity of understanding. At medical school, it was one of those lectures that you wouldn't want to go to because, it was all sorts of reasons.
Safe space. It's not like there are thousands of people watching.
It just wasn't, in my personal experience, it wasn't very well taught. And, I think it's a complex area to really grasp. And I extrapolate that across the whole clinical community. And I think there's probably a big lack of understanding. That combined with the lack of understanding in the public, I think combines it with the issues of trust, perception, and all of that. It was interesting, I asked my mother this morning as I was coming on this panel, I said, what do you understand about genomics and your genetic data? She thought that it would require someone taking a knife, taking a biopsy of her skin and analysing that. And she's quite well educated to some extent. But my point being that it's a very different perception of what people think, and unless we really start thinking across those lines, the challenges will still remain, as well as all the structural challenges that we talked about, which I think all fit as part of the picture. I also think that there's challenges in terms of how many people come forward for clinical trials. In terms of the research agenda, we see racial differences in drug discovery, but also with the age element, how many elderly people are coming forward for clinical trials versus young, fit and healthy. So I think that also plays a part in the challenges we have with data diversity. In terms of what we're doing about it, and I think with the 3 hats I mentioned, from my GP hat, I've been helping set a clinical trial up in my primary care community in order to engage more with the primary care side of things, encourage to get more people closer to their homes in the community to understand why this matters, rather than relying on big academic, prestigious hospitals, because people are afraid of those and people don't want to engage with those, whereas they do want to engage with their GP practice. From the Race Observatory, a big focus for us is the ethnicity coding. So how do we actually make that consistent throughout all the different areas of record keeping, so that we can then join the records up, we can have a true longitudinal record. And then from the HE perspective, there's a big focus on the skills and training part. So from a genomics angle, Health Education England sponsors the Masters in Genomics, but I think this needs to spread wider than just doctors. We need to think about nursing, allied health professionals and all the other undergrad, postgrad. So there's a big piece of work that's being thought through on that element. So I will stop there, but hopefully that gives you a flavour of what we're doing.
Amazing, thank you. I think the mum test is quite a good one. Quite often when we're not sure about something at GEL, our team were like, well, we asked our mum, and this is the insert quite a specific mum response. So I quite like the mum test as a barometer. Yeah. So just, we've already got some questions coming in. I just want to remind you that we're on social media, #GE Innovation21, and also to keep your questions coming in. There's no such thing as a stupid question, just knobheads who make you feel stupid, and we are not going to be that. So please make sure you ask your question, however extensive and scientific and technical or not it is, all are welcome. So there's a couple of things that have come up that I want to just double down on that also reflects a question that's come in around categories. So one question's come in from Varun Kumar, which is around the categories of ethnic groups and the categorization that we have in the UK. And it's building on something that Natalie said about the dynamic nature of data and the fact that actually our ethnicity and our cultural identity and how we feel on the day, depending on who you are, might very much change. And also, it might not necessarily capture the things that define how you behave. And I know this is something that you've looked at extensively. So how do we marry up the fact that we've got really big questions about cultural identity with the fact that we've got the reality of we have to monitor stuff, we have to collect stuff, we have to get a lay of the land, and that requires us to have some structured forms with some labels. So in the context of genomics, we overlay an extra level of complexity with the fact that we have a scientific definition of ancestry. Mm-hmm. So how do we marry up culture, ethnicity, ancestry?
Well, go on. Well, I was going to start by saying, I think it's really important to understand what the purpose is, right? Because if you have the purpose right and where this is all going, it really matters. I mean, many of you may have come across this, but when I was coding the other day on the vaccine program, and the drop box said mixed other, right? I mean, for many people, right, you've got your white, Caucasian, black, Asian, British, etc., and you've got mixed other. I mean, who fits in mixed other? I don't know. But if you ask someone, but my point being, my point being that for the individual, my point being for the individual though, it's like, I've got an ethnicity, this is, but I can't capture that. So my point being that unless we're clear on the purpose, is it for research, is it for actually, applicability in the clinical setting, I think that needs to be clear. And I don't personally believe we are clear on that as an ecosystem. I think we could be doing more on that.
If I may. So we are human beings and we live 70, 80, 90 years as average and then leave this world. But genes are ancient. They have a long age that goes thousands of years back. What we learned in our recent study that, so 7 or 5 years ago at Bourne in Bradford, we conducted this research on, kind of marital practices and childbirth and how it shapes our genetic makeup and what kind of recessive disorders we have under certain kind of variables and conditions. But then we learned also each ethnic group has a distinct population effect. Now, that was a new concept for me because I am the non-sciencey doctor here, Doctor Who. And I kind of learned got interested in learning more about it, how we capture the population effect of different communities, cultures, and people from diverse background. And this was fascinating. Each community or each geographical human group have kind of developed their bodily kind of genetic makeup under the influence of a variety of environmental conditions. So I'm a migrant, I'm from Pakistani background, but in India and Pakistan, people are socially there are hierarchies and castes and how people kind of define themselves and clans and biradris, and based on that, how people kind of, make their identity and then use it for political, social purposes. We learned it's fascinating how marriages or how kind of family composition changed altogether over the period of 2,000 years. So this is a research that we have along with scientists from University of Cambridge and Leeds University and Bourne and Bradford. We are going to publish in Nature Communications. We learned that 2,000 years ago in South Asia, broader South Asia, there were castes and people might be having all those professional occupational identities, but marriages were not within castes. Everybody was marrying everybody in the generic sense, not that in a open relation sense. But the point is there was no caste barrier to marry from within one caste. Down the line, around kind of 1200, 1000 years back, suddenly the genetic kind of analysis informs us, and people like Data Dan are absolutely, our kind of data administrator is also
I'm
Dan.
using that after
data. So our Bourne and Bradford person who managed this big machine of data is also Dan, and we call him Data Dan.
We were all...
That's it. And we need to kind of, So when we got that data and kind of, we learned that down the line, around 1,000, 1,200 years ago, what happened suddenly, people in South Asia and India and Pakistan, they started to kind of marrying within certain clans. And then down the line, 300-400 years ago, we learned that certain clans have more kind of intermarriages, and they happen to be upper caste during Mughal Empire. So we saw Mughal kind of preserve their kind of imperialism, and then local Rajputs copied the same kind of way. But something strange happened during British Raj, and that was interesting. So
a lot of things strange happened.
So that was like something which was fascinating. We learned that
you better answer my question at the end of this story, by the way.
Absolutely, and story is very important to answer that question. Otherwise, there'll be boring answer. So during British Raj, what happened that property and revenue laws were changed. As a result, my property will be transferred to my children and sons back in the days, and then to their sons and to their sons. And that means the first casualty of that property laws that were introduced to collect more revenue, the first casualty of that was women emancipation in South Asia. So men thought, okay, if women marry within the family, the land stays within the family. So land and women were kind of equated, but that significantly shaped our genetic makeup and how kind of the diversity of genes within South Asian communities, they were like kind of compromised significantly. Now, after that, what happened is that we learned that people organized themselves on clans and braderies, and we learned, okay, if we kind of collect data and connect family and culture and ethnicity and look at the person straight away, it would not make sense. The historical population effect need to be understood in more details, which means we will have to go back and see whether grandparents are related or not. We will have to go back even further and see what was happening 1,000 years ago and how this person's kind of DNA is evolving. So that kind of knowledge and education on genes and health is the way forward. What we learned is that when we kind of focus on individual and forget about the context, it does not help very much. And this applies to all ethnicities. And by the way, we live in this country and everything is kind of, fluid identities and everything. But somehow, strangely, when it comes to ethnicity, it's never perceived as fluid. It's perceived as identified through color of skin because we live in a kind of racialized, post-racialized society. So ethnicity is very much fluid at the same time. It's not kind of...
if I could pick up on that. So, I mean, what you say is interesting in that, often you would think, right, black describes
an ethnicity, right? But I mean, it doesn't. But you would still see that as like a way of describing people.
Yeah.
And then, you know, if somebody said to me like, oh, we've got a cohort of like black people, I'd say, well, that's meaningless. Like, what does that mean, right? Like, you don't sequence black people. You sequence people from certain areas, and that is what's going to allow you to ask interesting questions. And, you know, like as Maxine said, right, I'm a mixed other, right? That doesn't mean anything. Like, what is that? You know, like I don't have anything better to put in a, right? And so, you know, if I'm self-reporting ethnicity, I'm probably just going to say, right, well, that's just a bit of an awkward bin. I'm probably not going to look at that. And then, you know, you're just going to start ignoring some data because it's not, especially, it doesn't fit into a nice box. It's not easy to kind of try and marry other features to that to try and build a more complete picture. So yes, things need to change. The one that I was actually really alarmed about the other day, I had a day 2 PCR test. I was very fortunate to be on holiday recently. And on the ethnicity thing, the option that I had to pick was white and Caucasian. And I was just like, oh, I didn't understand why Caucasian was an acceptable term to be using at that point. But yeah.
When data down becomes missing data down, we've got a problem, I guess.
Yes.
So there's, and I just want to, I'm not going to pose this as a question because I think it's just a very good challenge, but it's a question from Ava. It says, what's the value of extrapolating ethnicity genetic group when there is no clear biological or genetic racial population? How will your push for diversity not merely extend biological perceptions of race, i.e. racism? So I think we kind of touched a little bit on that, but I think that's a very, very good provocation.
Yeah.
I
just wanted to follow in that, like, you know, there is no biological basis for race, right? So, like, yeah, that's just kind of like a very uncontroversial statement within biology. There is no such thing. What people would use to describe race are things that don't really relate to your biology. So, yeah, it's not part of this conversation.
Just adding on that, we are one race, human being. And then, of course, what we kind of differentiate with each other is based on the color of skin, but we are one race. And that has to be part of mainstream perception, that we human beings are one race, and that defines our biology.
I think there's a very important point, though, that goes back to Harpreet's, which is the purpose has to be very clear. And whilst I agree with the sentiment you've just expressed, you know, the disparity in COVID outcomes is a good example that in order to understand why we had a disparity in outcomes that was seemingly emerging based on ethnic groups, in order to dig down to understand, is there a genetic mechanism, is it social determinants of health, we had to go and collect some of that data as a starting point. So I know that Natty's involved in an initiative called People Like You, and we went to a conference last week which was talking about the increasing personalisation of data. And as you become more and more granular in defining someone, you hyper-personalise, you depersonalise someone because you break them down into their constituent parts. So I guess there's a kind of fine line between defining someone based on categories and then going too far that you actually lose what it is about them. But the purpose of collecting that data, I think, is a really important starting point to allow us to get to a utopia where we are all one race, one family, not in the polyamorous open relationship marrying sense, just to be clear. Good, yeah. So, gear change. I wanted to pull on something that Harpreet said, and I know that Natty's been very involved in, and it's this idea of kind of genomic exceptionalism. Like, why is genetics any more special or different to other forms of health data out there? And I do think there are some things that make this area of science more unique, but there's also lots of things that we can take from other areas. So I wanted to particularly start with you two. What can we take from vaccine uptake work and vaccine hesitancy work for informing how we want to engage in different types of people in their populations for genomic engagement? I think this question has also come in from someone in the audience, and also when you in your work looking at sort of most other forms of health data at UPD, and particularly things like electronic health records, do you feel that genomic data is any different based on all you've done in the last few years? So what can we steal from people who've already done a lot of work on this, basically, is the question.
Yeah, no, it's a good question. And, you know, for me, it was quite eye-opening in terms of, you know, a lot of the work that has been done, for example, in vaccine hesitancy, you know, and obviously that's come to the surface of it over the last 15 months. It's more, you know, we'd have a lot of armchair experts now. But my point being that it's really well thought out space. But if I can use an analogy, which is kind of the WHO model of the 3 Cs, right, which is often helps to overcome the hesitancy. The 3 Cs standing for the first one being around convenience. So how do we ensure what you're offering is accessible, you know, it's physically accessible, but also from a kind of language, literacy, all that kind of understanding, so that people are really aware of that is convenient. You know, it's open at all hours and all that kind of stuff that we did with the vaccine program. So that's convenience is the first C. The second is confidence, and I think that's really important in terms of the trust element. And so, you know, the NHS spent a lot of time building relationships with community leaders, faith leaders, trusted individuals, using different channels of communication for different people from different backgrounds, different ages, and so on and so forth. And I think that was a really useful learning, because not only has that really understood that there was some level of paucity of how well we were doing in the health system, but I think that opens up a broader set of opportunities when we think about prevention, well-being, and how we can engage with communities more, because I don't frankly believe that the NHS was doing as well as it could have done. And the third is complacency, which is not as relevant, but that essentially stands for, you know, from a vaccine hesitancy perspective, that this won't happen to me, so I don't need to take the vaccine, I'll be okay kind of thing. But if we take the first two around confidence and convenience, I think there's a lot there that we can learn. And I think, you know, like I said, the understanding of it, the people's perception of it, the trust element of it plays a critical part, and that requires constant engagement, it requires constant communication, it requires simplifying messages so that people understand the benefit, people are aware, but it also requires time to listen, right? This is not just about one-way dialogue, it has to be two-way. And that's where I think the GPs, healthcare professionals that were doing the vaccine program spent a lot of time doing, and they're still doing, and I think we have a lot to learn from there.
Is genomic data any special? Do we need to get off our little ivory tower of thinking we're unique?
In one sense, yes, and in one sense, no. So in the sense of yes, it's interesting when I try to describe to people what I do and what I work on, and I might make reference to sort of health data, and that might include genomics. People are, oh, so you're doing like Gattaca prevention then? You know, they're just like, you're trying to stop that dystopic future, because the perception of genomics might well be that that's the direction it's heading in, right? Or that's the potential. And to be fair, that's not an entirely ill-formed view based on the history of eugenics. You know, it's not as though that's something that's entirely unreasonable to be worried about. But on the other hand, certainly we've found when you engage and talk to people about the use of health data, routine health data, and questions about genomics might come up. Actually, the sorts of questions and concerns and issues that arise are fairly similar. People want to know, you know, what's this data being used for? How's it being protected? Both in terms of my own privacy, but also in terms of any societal risks and harms or risks of discrimination against groups. How are we going to ensure that the benefits are actually coming back to patients and to the healthcare system? So those are the sorts of questions that come up all the time with regard to routine health data that apply just as much to questions in genomics. So I think, broadly speaking, once you start to engage with it, and it comes back to Harpreet's point about the two-way dialogue, really exploring the kind of questions that people might have, give them information, give them an opportunity to ask questions, to scrutinise, to challenge, then actually the sorts of issues that come up are very, very similar. And certainly we found that even if you're trying to explain and convey the potential benefits and the advantages and the way that, you know, safeguards operate, there's legitimate challenge and question there. Are you sure that the data is going to be used in that way? How safe can it be? Can you ensure that it is not going to be used for all these purposes you say it's not going to be used for? What are the assurances there? How can we scrutinise and challenge and check? Where's the accountability? All of those questions, I think, apply just as much. And the difficulty we have, I think, at the moment, certainly in the health data space, is that people feel there's good reasons not to trust what's going on. We've had the GP data debacle very recently, where, you know, it was certainly felt as though for all of the articulation of, well, here's what the data's for and how it's going to be used and how it's protected, people responded with, I just don't believe you. I think this data is going to be exploited. I don't believe that the rules that you're setting out here are going to be upheld. So I see a lot of parallels in terms of the risks and the issues around genetic data with health data more broadly. I suppose the one thing that I would say seems to create additional concern is just that sort of the future orientation of genomics and the risk that it might be used to discriminate or harm some people or benefit some at the expense of others.
Yeah, and I know that's something you've talked about a lot in terms of closing the research loop, making sure the questions that certain populations care about are the ones that are being asked, and that's being fed and communicated well in a consistent way.
I think whenever we disseminate any analysis of data from genes and health point of view, then somehow mainstream media will always narrow it down to family and culture and ethnicity, which kills the whole purpose of disseminating the message of health and its relationship with genes. What we have learned is that we need to kind of talk about genes and health more and more with young people, with children in schools, and that's what we plan to do in future through Born in Bradford, next upcoming birth cohort of 30,000 young people. But what happens is that when we say this health issue is because of the family, or because of the culture, or because of the ethnicity, then we shut that door of kind of somebody willing to give you data. Then we create those conditions in which trust is gone. Then we come up with a language that does not help communicate the message. And I mean, just one tiny example is how convenient we say COVID vaccine hesitancy, and suddenly the relationship goes to ethnic minority, as if people in ethnic minority background are hesitant to save the lives of their loved ones. But for everything else, we use the term uptake. And whenever all the health interventions or vaccines or medicine that's given to majority population, it is spoken through the language of uptake and never hesitancy. Funnily speaking, in London, in this fantastic city, when there was all the people who were anti-masks, anti-vaccine, anti-this, anti-lockdown, anti-social distancing, they were coming in thousands and they created a mess. I was looking at those images on TV and I was like, wow, those people down in London, they are like more kind of hardcore when it comes to kind of defying the law. But I then looked at how they look like. And somehow those people who were, in inverted commas, vaccine hesitant were not happen to be like ethnic minorities. And they are never informed or talked about as kind of hesitancy. The other problem with this kind of language is, okay, we say vaccine hesitancy, and then suddenly experts, and particularly, no offense, but medical experts coming, obesity is linked with prevalence of vaccine positivity. What is obesity? It is not linked. It is correlated. Obesity in itself is a reflection. I was just speaking with one of your colleagues before coming around this topic as well. Obesity is a reflection of 2,000 things going wrong back in the context, in environment, in structure. And those things are directly linked with COVID positivity and not the obesity. Suddenly, when we say obesity, we identify majority of working class people somehow because of how they look, how they appear, and what environment have created conditions in which their bodies look in a certain way. That is the reason for COVID positivity. So kind of these correlations which are like imaginary in a sense, because there is no real reasoning for that. We ignore those correlations which are real, like environmental structural factors, like food habits, like green space, like, you know, a conducive environment in which children and families can go out and eat healthy and spend healthy time and create those conditions in which they can grow up happy and healthy. Instead of talking about those real things, we talk about, I mean, things like obesity and COVID hesitancy.
I think you raised like very important points about the kind of multifactorial nature of what contributes to health and the fact we have to really recognise genetics in the context of that. Have you got a finger up or are you just tweaking?
I don't know if to put a whole hand up or just like a little,
go for it,
subtle.
Go.
I think the point about sort of ethnic minorities and vaccine hesitancy is an interesting one, and it was clear that
on track as well of the theme of the panel.
Yes, it was clear that there was a point at which uptake in those communities was lagging, but research from King's College would indicate that a large part of the reason for that is because the way it was being communicated to them wasn't culturally relevant. Right, so this is the problem of kind of like one size fits all like health policy and messaging, is that for communities that have different ethnic backgrounds, different language structures, their understanding of certain concepts might actually be different. And one of the major findings is just like the actual, the notion of risk is actually very different within a Bengali community than it would be within, like a Western European community. And also things such as, saying like, okay, well, look, you got to bubble and you're allowed to see like your core family. Well, core family can be a very different thing if you're from a South Asian community where family can include, your aunties and uncles and all this kind of thing. So actually kind of one of the things that really helped with that was, as you were saying, was it's the engagement with community leaders, people who have, deep knowledge of the communities from which they are a part, and changing the messaging so that actually we're not just translating words, we're also like translating the meaning.
Just wanted to quickly pick up on the point around whether genomics data is seen as kind of exceptionalism and it's different to other. And I think the reason why that may be perceived to be like that is partly because where it sits and its relevance of it, because you've got all the data that we have in healthcare, then you've got genomics data here somewhere, and it's never quite linked up with day-to-day practice clinical work. So I think that there's a big undertaking there that needs to be taken. And the second is, who's the champion advocate that's protecting it or helping patients understand it? You use a great example of the GP data issue that we had, and essentially it was the GPs that were speaking up on behalf of their patients. I think it's very hard for GPs to speak on behalf of genomics data because they're not very involved with it at the moment. And so there needs to be a much more grounding on how do we then interact and intersect there so that we do have advocates, we do have people who are trusted. But ultimately, because this is all patient data, but regardless of us saying that, patients always end up coming to the GPs to get their advice. And I think there's a missing link there.
I think what ties actually a lot of these points together, and there's a question that's come in from Luca, which again I'm going to be rude and actually make it more of a point because I think it's a good point. On the point of multifactoriality, that's not one to say after you've had a few GNTs. Not that we've had GNTs, but you probably have in the audience. With increasing inequalities, we will expect an increase in health inequalities. Is the increasing recourse to GWAS study and polygenic risk scores shifting responsibilities of health inequalities from society as a whole to the individual or even ethnic groups? So I think that that's quite like a nice way to tie in the challenge of if we're thinking about social determinants of health, we're thinking about communication. What does that mean for the individual in terms of the responsibility they should be taking? I'm going to bring us back to a question, this is going to be the last one from the audience before we kind of wrap up, about the science. Now, Data Dan, I'm not going to ask you to speak on behalf of all analysts everywhere, but
no, I can. I do that frequently.
Yeah, good. No. But something that Natalie said, which is around how do people still don't believe that there'll be the quality control and there's the accountability, and people actually do it in the right way. And one of the big things that we're trying to think about in the diverse data program is how do we support a whole cohort of practitioners, be them researchers or clinicians, to change their behaviours and be more aware of the fact that the data sets and the insights they're receiving are probably going to be in some way biased. And on a day-to-day basis, you'll be making loads of decisions. There's loads in the pipeline, you're just a very important cog in it, but you'll be making loads of decisions which will shape the outcome of analysis. So there's a question that's come in to say, given that the current human reference genome is European-centric, there must need to be a large and vigorous quality control process when calling variants for other ancestries. How is this handled? So I wouldn't mind you painting a picture of, you're at your computer, like, how does an analysis work like this? And what are some of the decisions that you're making that you would hope other people could be more aware of? Because for some people who are prolific bioinformaticians, this is your bread and butter. But I think for many other people, they won't necessarily know what this entails.
Yeah.
Paint us a picture of you sitting at your laptop.
So I've got my headphones on.
Good, yeah. What kind of music do you listen to?
Great, coding music.
Multiple screens?
Oh, at least three.
3 screens.
Everything in dark mode. So, yeah, I mean, it is a difficult question. Yeah, we'll start with the basic principles first. So, okay, so let's say we've got the 100,000 Genomes project and we've got 80,000 people in there. They all come through as these individual files that says, right, this is where you are different from the reference genome. And then from that, what we do is we will merge these together because if you want to try and do research on it, having to look at 80,000 different files that have been, it's really challenging. So we put it all together, and then we go through quality control. And this is because you need to have some confidence that the variant you're looking at is real. There's a lot of things that can happen in sequencing and other things as well that mean that actually it would be a spurious variant, and you wouldn't want to try and link that to disease because that would be wrong. So you have to then try and say, right, these are the good variants and these are the bad variants. But the problem is that population and genetic ancestry does actually have an impact on that, because there'll be parts of the genome that are better represented in certain ancestries, or others where there will just be a higher degree of variation. And currently what we do is, for this data set, we do it across the board for everyone. It would be nice to be able to split that. I've actually literally been discussing this today to try and integrate bringing in the superpopulation. So in genetic ancestry, we tend to talk about 5 main populations. So we've got Europeans, East Asians, South Asians, East Asians, and American, which is bit more of like a mixed group. And so the idea is that maybe what we can do is actually we can apply this site-QC to each of those populations individually, and then that gives you a better idea of whether you don't want to find that because we've got 70% of our data is European, you don't want to find that that's causing variants that actually are correct in the African subpopulation to be kicked out. So if you do it all separately, then it could give you an indication that that kind of the population structure is what is affecting that there.
I would listen to your call, Sarah.
Oh, excellent. Okay, cool. I'll set one up.
Perfect, thank you. I'm actually just going to take a question, but we've got a bit more time for more for the audience, around kind of innovative partnerships and working together and how to bring in different sectors. So the question is, how can charities and industry, etc., who fund medical research basically address this data diversity problem? And I think it'd be quite good to kind of attack this from lots of different angles. And so maybe we can start maybe on the engagement piece through innovative partnerships, if Harpreet, you want to kind of kick us off.
Yeah, absolutely. And I think, again, if I may use the example from the vaccine program, again, it was a real eye-opener in terms of where we were going and where a good proportion of the population spend their time. Just as an example, males under the age of 35 or in 40, actually, many of them don't engage with their GPs. Not many of them are coming forward. So where do they go, right? Football clubs, rugby clubs. So think about those kind of opportunities where you're creating these creative partnerships where certain cohorts of the population are going. Again, partnerships with religious institutions, right? Again, I'll use my parents as an example. My father got in touch last week and said, it's organ transplant week. I'm going to the Sikh temple to learn about organ transplant. But that's a good example of how we are using those setups where certain parts of the population are engaging. So this is not just about ethnicity, but it's also about age, and it's also about thinking different demographics and where do a lot of people spend their time. Within the NHS, for example, we developed a partnership with the Bangladeshi Restaurant Association because a lot of people go to Indian takeaways, and there's a partnership there that we developed to engage the Bangladeshi population in the vaccine uptake. So my example there is being that we can't just go mainstream and hope that everyone reads BBC, The Times, The Telegraph. Actually, a lot of people don't read those, and there's a lot of people that read different things, different modalities, and engage in different ways, and we need to think about those creative partnerships.
Yeah, one thing you talk about quite a lot is making sure that we kind of go to where people are.
Right, exactly.
Hey, I went and had my smear test in a nail bar, which I think is just going to radically change my life.
And then that's a good example because
2 birds on stone, perfect manicure.
But that's a really good example because in Africa there's good studies which show that people talk about healthcare in their hairdressers and when they're talking and getting their hair done. And I think we could be using similar examples here.
Yeah. So Natalie, your colleagues at the Wellcome, there's a lot of pennies sitting in Wellcome, and there are tiny, nuanced, minutiae decisions can move millions. And I know that something in the kind of digital equity and whole data science area is at Wellcome, you're kind of thinking a lot about actual funding and thinking about in the global context. Can you give us a sort of sneak peek in terms of the vibe at the moment, in terms of thinking about redressing the global imbalance on health data funding?
Sure, I can try. I do not necessarily speak for Wellcome, but one of the things around Wellcome's new strategy, particularly what's happening in the Data for Science and Health team, is that we have a new strand of work called digital equity, which is really trying to, I guess, address some of these questions from the perspective of a funder. And the key rationale behind that team is to look at and take into account the needs of those who are developing, using and affected by data science and digital tools. So that looks at those who might be developing machine learning models and using data to develop tools that might have an impact on health challenges that Wellcome cares about. It's about how those things are deployed and used in practice, particularly in clinical practice when it comes to health data, but also looking at how you can do sort of community engagement in a more robust and sustainable way, rather than seeing it as an add-on or a bit that you do at the end, kind of really building, thinking about who might be affected by the sorts of the data science that we're funding. And a key thing here is really trying to think quite carefully and critically about the sorts of communities that Wellcome has historically worked with and maybe where we've missed things. So at the moment, it's pretty challenging to figure out how to explore relationships with potentially new grantees or new partners that Wellcome might work with. Because we have a sort of established set of structures and processes, and it's quite a big funder and so on, it can probably be pretty intimidating to think to work with different kinds of people. So in the team, we're thinking a lot about how to create more of an on-ramp, I guess, for different types of people to come and chat, to come and talk to us, to explore with us what work they're doing, and maybe whether Wellcome could provide support for that. Also just trying to look a lot more globally than Wellcome has historically. Wellcome has funded Africa and Asia programs. We've got very established centres in different areas, but actually what's the future of those? Are we going to be able to maybe explore working with different communities? We haven't done much work in Latin America, for example. So there's a lot of thought going into ways of trying to explore more what Wellcome could do, not just as a funder, but more of a kind of, I hate the term thought leader, but a bit more of a push, a strategic push to think, actually, if we care about these sorts of health challenges, particularly around climate and health, mental health and infectious disease, how can we ensure that the science that Wellcome is funding is going to benefit everyone? And that benefiting everyone bit is what we're trying to figure out. How do you go from a slogan? How do you go from a slogan about benefiting everyone to practically speaking, ensuring that the science that Wellcome is funding and what it's supporting isn't inadvertently causing harm, for one thing, but is actually going to have more of a beneficial effect across the communities that we care about. So very early days for that team. Wellcome is kind of in the process of trying to figure out exactly how it's going to implement this strategy. But I think the digital equity piece of that is going to be really exciting in the longer run.
Absolutely. Yeah, absolutely. I think that pulls in on a couple of questions and comments that have come in around, focusing on digital equity or diversity or any permutation of these words. What do you run a risk of potentially causing more harm than good, or by kind of shining a lens on the specific challenge rather than maybe embedding it? Yep. So I think that's a good point. It's difficult to know whether to take one from the audience or to be really selfish and choose my own. Maybe I'll take one and I'll bundle it. So there's a good point that's made that are there more diverse genetics projects planned in the UK? Because this person feels that when asked about genetics diversity projects, born in Bradford, excellent as you are, is often used as the example. Surely there are more and more is needed. So yes, there are more and more is needed. But I think that this raises a very good question, that one thing that we found in this really early stages of divining and designing the diverse data program is it feels like a very, very fragmented area of research, because you've got the algorithmic fairness community, you've got the population geneticists, you've got the health data ethicists, and everyone's kind of all linked, but it's not quite talking to one another. So you're not allowed to say born in Bradford, by the way, but if you had a kind of different example of a project or a piece of work that's looking at something to do with this whole space of data diversity that you would particularly want to say, I think these guys are legends, do you have one? You can start, as long as it's not born in Bradford.
I am not aware of any, I will be honest. Maybe...
It's not competition, but Dan seems to be...
Well, so like East London Genes and Health, right, as an extension of Born in Bradford, well, so that looks at other diverse communities within East London. But I mean, also, I think one of the things that's really great is that, following on from the example, I think, really of Genomics England, there are a number of different countries now that are setting up their own similar programs. And this is across the world, whether it's like Australia or Hong Kong. There's in the States, there's the All of Us program in the States, is a very big diversity initiative there. So I think people are really mobilizing and doing these large scale type projects where diversity is, it will emanate from that.
Yeah. And I think there's a big question about how we work internationally. I think one thing that's fairly unique about certainly the data that GEL has is its linked data to administrative data sets like clinical records, which lots of other countries may have done whole genome sequencing but don't have that data linkage. And to your point, I think that there are potential massive problems and risks if we continue to look at genomic information without additional context around social determinants of health and clinical information. So I think for us that's a real challenge in the programme, that we want to be able to make use of all these international efforts, but if we don't have the same sort of data structures or linkage, or we're not framing it in the same way, it does make it a bit more challenging to collaborate. Doesn't necessarily have to be on genomic data diversity. Any other kind of interesting data diversity problems?
Well, I do want to pick up on one that is genomic data, which is the H3 Africa Consortium that you mentioned, the Human Heredity and Health Consortium in Africa, because there's more genetic diversity within Africa than there is across the rest of the world. And I think people often sort of think of Africa as a country. It's not, it's a continent. It is absolutely huge. And this is a very substantial consortium that Wellcome funded along with the NIH in the US. But that was really focused on developing capacity and leadership for genetic analysis within Africa. So I think that's kind of a nice example of something that was it was set up to try and avoid this problem of being extractive with data. This is not just people coming from the US and the UK extracting data from African populations, taking it away and doing analysis themselves and creating benefits for their own communities and populations, but it was very much focused on that really capacity building, those capacity building efforts in many of the countries across Africa. So that's the one I'd point to.
Yeah, we did start by saying how colonised the whole scientific landscape in the space is, and I think that's a good example. I'm going to just take opportunity of being the chair to answer one question from Taminda, which is, what checks are being put in place to ensure that ethnicity and other diversity metrics are built into study design and not an afterthought? Are these things being taken into account when accessing the GEL research environment? So work that Dan and I have been involved in, and it's being led by a fantastic designer in our team, Sophia Liu. She has done a thorough dissection of all the biases that exist within the GEL pipelines. Not to say that GEL is particularly bad, but I think if we were to apply the same method across many institutions in the world, I think we'd all come out, maybe being as less good as we could be when it comes to being a completely bias neutral space. And so one of the things is just finding small ways we can just nudge people's behaviours just to be a little bit better. So that could be when accessing the GEL environment, or putting a proposal or writing up your paper to make sure that you've considered equity and diversity in the analysis of that. Or the bar we've realised is set very low just for basic awareness. So we're really hitting this at lots of different levels. It's not necessarily a fancy statistical model, whilst I think a fancy statistical model is sometimes also fun to use. So we've got 5 minutes left, so I'm going to charge you with 3 calls to action. By this point, if you're sitting at home, I'd like to write everything that 12 calls to action. It's probably a long day, guys. It's a hot light and I'm sweating loads. But if you were to give 3 concrete calls to action that individuals watching could go do that would make a measurable, or at least, let's go for measurable, improvement and progress on data diversity, what would your 3 be? Let's go, Harpreet, you go first, because you're always last.
Yeah, no, okay. So
weren't expecting that way.
No, absolutely not. So I guess 3 for me. Firstly, on the engagement side, so I think try and engage your communities, do events for whether it's your local religious institutions or whatever it might be, but try and engage more with the communities, provide more education. I think that's the first thing. The second is, let's start getting more representation of people talking about this from different backgrounds, so that people can relate to each other, they can talk like each other, hear like each other, because I think that's really important, which is the second. And thirdly is, let's start collecting those stories, right? How has this benefited patients and their families? Because I think we need to ground this in reality and make it more applicable rather than it just being in the research phase. And I think we need to collect more of those. So those would be my kind of 3 things.
Nice, punchy. Dan. Data Dan. Missing data Dan, present data Dan.
So I'll probably split my first one into 2. So I think what would be really, really nice is if less research was thrown away because it's not in a European population, right? And what I was saying before about the notion that in smaller genetic ancestral groups, we don't necessarily have enough statistical power to do analysis. I think what we end up doing is, if you look at all of these different pieces of research that will be submitted by researchers, and you think about all the little bits of data they're throwing away, if you start putting that together, you could actually start to answer some of the questions that we might want to ask. And I think it's important to consider that, the role of the publishers in this as well. And we should say, we should give them a call to arms and say, you should start requiring this kind of data. That you should say, if in your analysis pipeline you have discarded people of some ethnic minority or some different genetic ancestral background, you should explain why that is. And if it's not a good enough reason, then that's the thing. So yeah, one is to try and really reduce the fear factor in working with diverse data, diverse population data. and 2 is to make that more accepted. And then lastly would be around language. As has been alluded to, genetics has a pretty checkered past when it comes to eugenics and the like, and unfortunately, to this day, genetics is still used to further racist agendas. And I think that we have a responsibility as a genetics company and as geneticists or scientists or people in the field to really clarify exactly what we mean when we say something. So to explain why I say ethnicity or why I say genetic ancestry and what the difference might be between the two. And I think that not only do we need to decide that within ourselves, as a community, we also need to then communicate that more widely so that we can't be misunderstood, so that our research can't be used in that way.
I think excellent points. Nature, New England Journal, Lancet, we're on to you. Natalie, go ahead.
Thanks. So my first point would be around the inclusion and involvement and engagement as Harpreet alluded to. Don't reinvent the wheel. There's a lot of really good resources out there. There's the NIHR Include framework. There's networks like the Equality and Diversity in Science Network. There's the Data Science for Health Equity.
I know that was my I was going to finish on that. Come on, Natalie.
I'm teeing you up. I'm teeing you up. So basically, there's a lot of existing resources for how to engage, involve different communities. I know we've focused on ethnicity, but of course there's lots of different aspects of diversity. So don't feel that you have to sort of start from scratch if you're exploring new issues around inclusion, involvement, and engagement. Secondly, for those who are responsible for budgets and for staff capacity, resource this stuff and allow people the time and the capacity to ask questions, to be curious. There's so much literature exploring these kinds of questions. There's Superior by Angela Saini, there's Race by Rua Benjamin, there's Invisible Women, Caroline Criado Perez. A lot of women and people of colour writing these books. But there's a lot of reading. And if you're curious about this and you have the capacity to explore some of these questions, and I do think that those who are responsible for budgets need to take this into account as well, that actually it takes time. You shouldn't be only exploring these questions on a sort of voluntary basis. This should be sort of built into the way you're working. So that sort of be curious and resource it. And then thirdly, I guess don't make the perfect the enemy of the good. I think it's very easy, as I said earlier, to sort of get stuck and a bit paralysed and not quite know what to do here, but small incremental steps to be curious, to raise awareness of maybe where there might be biases or assumptions that go into data and the analysis of that are steps in the right direction. And the fact is there's not going to be a clear-cut answer to how to do diversity well. It's just not something that's going to have a neat solution. So small incremental steps are beneficial.
Perfect. And I think on that note about kind of reading and consuming and educating yourself and getting out there, as a programme we bosh through a lot of videos and books and articles. Some of them academic, some of them quite light and jovial, but we are going to be releasing our resource list very soon. Last but not least, Sofiane, succinctly.
So one concept that I came across recently was reference genomics. That is, for example, for ethnic, religious, sexual minorities, if we do reference genomics, which means we take a clan, a group, a body of people, and then have their kind of knowledge about their genetic history and makeup, and then do the same with another clan or group of people within the same community, and then kind of communicate those kind of health benefits and kind of ill health factors to those people. That can be a kind of good way to communicate with those people who generally are at the margins of this kind of debates. The second thing is young people, and I am kind of realizing it more and more since past 4 years. I've been working with children and young people, and it has to be part, genes and health, the information on genes and health has to be part of school education, or where children are growing up and then they are about to end their teenage and going to start a kind of adult life. So we need to go to those children, young people, and talk more about genes and health and sensitivity related to this. And I mean, we are asking a big chunk of money from Wellcome Trust to help us do that. Thank you very much. Yeah, Don, my director, we sorted you here, and I need promotion for that. Now, the third thing is that when we're trying to help communities through the beautiful, powerful knowledge of genes, Let's not blame communities and families and ethnicities because that is counterproductive. And instead of relating it back to the cultural practices, let's look at kind of different variables that affect people's health and make them choose. Social class in Britain is a big theme that needs to be related in this broader debate of genes and health and how we basically explain to people about the benefits or consequences of certain choices they make in daily life.
Amazing.
I will shut up here.
No, perfect. I'm getting told that we need to close because we just had such a stimulating discussion that people are just overwhelmed with how interesting it is, and Harpreet needs to catch a train. But anyway, thank you so much for listening. Thank you so much for joining for the whole day or just our session. And we will hopefully see you soon. Our door is always open at the Diverse Data Programme. We are still early days. Most of the questions were raised were really knotty, complicated and horrible ones. And so we look forward to answering those with you because we recognise this is not an easy thing to solve. Thanks very much for listening.